Professor, VSS Institute of Medical Sciences, and can we please welcome the chairpersons as well with a huge round of applause. Dr. Smita Sontakke, Professor and Head, GMC Yavatmal, तालियां बजती रहें, रुकने न पाएं. Dr. Anant Patil, Associate Professor, Dr. D.Y. Patil Medical College, Mumbai. A warm welcome to all the three chairpersons. Thank you, sir. Good morning, sir. Yes, sir, it's already there. The CD is there. It's there, sir. So that's it. A warm welcome to all the three chairpersons, requesting the chairpersons to kindly take over the proceedings of the session. Thank you.
Hello.
Hello, a very good morning to this second and final day of NAPTICON 24 at IIMS Nagpur. So we are into the first session. I have the pleasure to introduce Professor Dr. M. Jayanti, Department of Pharmacology, JIPMER, Pondicherry. She has an experience of 17 years in teaching and clinical research. She has 60 publications to her credit in peer-reviewed journals. She has served as HOD of Pharmacology and Member Secretary of Institutional Ethics Committee for Interventional Studies at JIPMER. She is currently in charge of Central Pharmacy at JIPMER, and she will be delivering a talk on CRISPR-Cas9 and how the gene editing has helped in sickle cell anemia patients. Thank you very much, ma'am, and please carry on.
Thank you for the kind introduction, sir. Respected chairpersons, the dignitaries, and the delegates, whoever is present here. I thank the NAPTICON for giving me this opportunity to speak about this wonderful topic on CRISPR. So in the next 20-25 minutes, I will be covering these topics. A brief introduction to this technology as such, what is it, and its application in sickle cell disease, the therapeutic strategy behind, the preclinical study results, as well as the clinical trial results, and what is the future direction. Now this gene technology or gene therapy, if we see around 2 decades ago, it was a big hype. Okay, so a lot of things were expected from this. Thinking about this technology, gene therapy, is because of a lot of challenges involved in delivery of this technique. But if you see now again, probably it is really a hype. So this kind of an attitude, this is what a change in the outlook of this gene therapy, and this is one of those gene therapies about which we are going to talk. So I would like to again substantiate my saying that it could be a hype in future because of this. We have the evidence that these two great scientists have been awarded the Nobel Prize in 2020 in chemistry. So these are Emmanuel Charpentier and Jennifer Doudna. So they received this Nobel Prize for discovering this gene technology called CRISPR-Cas9. So what is this CRISPR-Cas9 technology? So this term, it is an abbreviation, CRISPR. It stands for clustered, regularly interspaced, short palindromic repeats. And Cas9, okay, it is CRISPR-associated protein 9. So what is this? It enables precise, customizable DNA editing. So we are aware that this editing function is such a boon to us because we use several kind of software applications, okay, whether it is Excel or Word, even Word document for that matter. Whenever there is a editing function, it makes our life easy, is it not? So when there is something which can edit the genome at a particular place, you are able to bring about a change in the DNA at the desired site. Okay, so those diseases which have mutations, gene mutations, It would be wonderful that we just replace that mutated gene with the normal gene, and it can cure a disease, genetic disease. So definitely this is going to be a great thing. It is not that this is something new. This has already been tried, as I told you, for last 2 decades. Okay? But we did not have something which really showed a very good effectiveness in curing these diseases. But CRISPR-Cas9 seems to have that potential. Let us see how. Now, how did this idea come about? Okay, so this is the scientists, they found some sequences, okay, called CRISPR, as I was telling, regularly interspaced sequences in the genome, okay, and it is like a palindrome. We know what is a palindrome, okay? When you read from 5' to 3' on the both plus and minus strand, the sequence remains the same, okay? So they started exploring this. So this was first found in the bacteria, E. coli bacteria. They found that some sequence like this is present, okay? So they, on further research, they were interested in what was present in between these sequences. So that sequence which was present in between CRISPR sequence, it matched with the DNA of virus, okay? Those viruses from which these bacteria were protected, okay? So this was interesting. So they looked at that particular sequence which was present in between the CRISPR sequences. So they found that this CRISPR RNA on transcription, so this CRISPR array, it forms CRISPR RNA. But 2 other sequences also were of importance, okay? One which formed the transacylating RNA, otherwise called tracer RNA. Another one which was transcribed into Cas9, okay? The Cas9 protein, endonuclease. So all these 3 formed a complex. So this complex acted like a soldier kind of thing. So it will, so when there is infection of this bacteria by the same virus, it will look out for the sequence because it has that viral DNA sequence with it, is it not? So it will look out for this sequence in the virus. So once it identifies that sequence, the Cas9, which is an endonuclease, goes and cuts the DNA at that particular site, thus disabling the virus. So this way it is something like a adaptive immunity in the prokaryote, okay? So this idea was very fascinating for the scientists. They thought, why not use it to edit the human genome for treating the genetic diseases? So for that, they had to do some engineering. So they had to, yes, Cas9 was already available in the bacteria, yes. Now to mimic this CRISPR RNA as well as the tracer RNA, they designed something called a single guide RNA, which will act like this. So it will guide the Cas9 towards the site where it has to make the cut. So this was the complex that was designed. So in addition, this Cas9 will also recognize the site at which it has to cut by looking at the sequence called PAM, okay? That is protospacer adjacent motive. So all these help to correctly go and localize the site where the cut has to be made. So that is how this design of this CRISPR-Cas9 for human genome edit developed. Okay, so this CRISPR-Cas9 is the, along with Cas9, that is the blue colored one, this purple colored, the single guide RNA, this will act as a complex and it will go and locate the site of interest and there it can make a cut, okay, the CRISPR-Cas9. So when there is a cut on the double-stranded DNA, so the repair process is error prone. So this will lead to non-functional, that is silencing of that particular gene. Now, coming to this repair process, there are 2 types of repair process available for whenever there is a double strand break. So these 2 repair process, one of them is called non-homologous end joining, other one is a homology directed repair, okay? This non-homologous end joining, this is what commonly occurs and it's a very simple mechanism, that is when there is a break in the double stranded DNA, so there is just resealing of the broken strands, okay? So this resealing, it occurs, so there can be, it is error prone, okay, just removal of one nucleotide, addition of one nucleotide until there is this resealing of the cut ends. So this technique is useful for knocking out of genes. Okay? Whereas what is the other one? Homology directed repair. In homology directed repair, what happens is this error does not occur because this resealing mechanism is aided by introducing a donor DNA. So this DNA is in such a way it is introduced that your desired sequence, okay, which is going to produce the desired effect, that sequence can be introduced and this will guide the repair process. So this is not error prone and this is more precise, okay. So these two mechanisms have been tapped for this further use of this CRISPR-Cas9 technology for developing treatments for the genetic diseases. Okay, so as I told you, this is the homology-directed repair. So we can introduce the desired sequence of interest. So along with the temp sequence, which matches with the homologous cut ends of the double-stranded DNA break ends. So this will result in modification of the DNA as per the requirement. Okay, so now the technology is ready. Okay, so you can make a change in the DNA for those diseases, genetic diseases which involve mutation. So how to deliver this technology into the cells? So there are several delivery methods which we are already aware of, like we have even the COVID vaccines available. So like that, we have plasmid mediated delivery, then we have and like we have the adenovirus, adeno-associated viruses, lentiviruses. All these are they are useful to deliver this technology into the cells. Then we have the preformed ribonucleoprotein between the guide RNA as well as the Cas9 protein. So all these technologies are available. In addition to this, there are also some physical methods, that is through a needle directly into the cell, like for electroporation or micro-injections. So that way also this gene editing technology can be delivered into the cells. Okay, so now is CRISPR-Cas9 the only gene editing technology? No, there are so many other technologies also available, like this zinc finger nucleases, talons. They have been researched upon for quite a long time. But there are some problems in the engineering of these things, okay? Whereas this CRISPR-Cas9, the Cas9 is already available naturally in the bacteria. So it can easily be adapted and it will do its majority of its role. Only what we have to design is the single guide RNA in order to guide the Cas9 to the site of interest. So that way, this is far easier to engineer as well as cheaper while compared to the other technologies of gene editing technologies. So if you look at its application, it has been tried in several diseases, okay? In addition to sickle cell disease and thalassemia, several other diseases where a mutation in the gene is responsible for the disease, like cancers, even infectious diseases, cardiovascular diseases, diabetes, and so on. But what has really reached the final approval stage is this, that is for sickle cell disease and for thalassemia. Now let us see how, what is its application in sickle cell disease. So we know that sickle cell disease is a hereditary blood disorder and annual incidence is around 3 lakhs worldwide, and it's a chronic debilitating condition. So the problem here is a mutation in the position 6 of the beta globin subunit of the hemoglobin. So because of this point mutation, there is replacement of glutamic acid to valine. Okay, so this results in sickling of the red blood cells in conditions of hypoxia. So these sickle RBCs, we know that they can cause blood vessel occlusions. So these occlusions can lead to the sickle cell episode, that is vaso-occlusive crisis episodes resulting in severe pain in the patient and also hemolytic episodes. And this vasculitis can involve almost any organ in the body, and this has reduced the lifespan of these patients, okay? And these people also require multiple blood transfusions and all the problems associated with this. So now if you look at the current therapeutic modalities, we do have several of them. For example, the gold standard being hydroxyurea, then several others also have come to the market, like L-glutamine is there, crinalizumab is there. So all of these different therapeutic modalities, they target that is the consequence, that is a reduction of vaso-occlusive episodes, okay? Reducing the, that is the aggregability of the sickle cell RBCs. So they look at only the symptoms and signs of the disease, but they are not targeting the source of the disease, that is the mutation. Of course, there is a potential cure also in the form of allogenic bone marrow transplant. But the problem is you have to get a HLA matched donor. So that is a problem. So it doesn't occur. Not more than 10 to 15% of the cases are able to afford or to get this matched donor. So therefore, there is a great potential for this gene therapy in curing the sickle cell disease. Introduce this CRISPR-Cas9 technology for this mitigating this mutation, okay? So they observed, like for example, hydroxyurea, I said, it also acts by increasing the production of fetal hemoglobin. So I want to reiterate that in sickle cell disease, what happens is the beta globin chain of the adult hemoglobin that is mutated. Whereas if you look at the fetal, the chain, subunit chain, there is gamma, okay? So this, when there is persistence of the fetal hemoglobin in the adult stage, they found that this kind of episodes are reduced. People do not suffer from vaso-occlusive episodes, okay? So usually what happens is at birth, this gamma globin or the fetal hemoglobin declines and the adult hemoglobin takes over, okay? So that is why these people with this disease, their symptoms start after around, you can see around 6 months, where it is almost the switchover is complete. So after 6 months of age only, there all these symptoms starts, okay? So this was an observation. Yes, they said, yes, we can do something where we can prolong the the persistence of fetal hemoglobin could help in this disease. Then they looked at the molecular factor, what is responsible for this switching over, okay? What is causing the fetal hemoglobin to decline its action? Then they found this transcription factor called BCL11A. So this is a transcription factor which represses the gamma globin expression at birth. So this was a potential target, okay? So if you are able to silence this BCL11A, so there will be persistence of fetal hemoglobin. So that will protect against the problem of the sickle cell crisis. So that is what was, this CRISPR-9 was used for engineering this single guide RNA to go and act on the erythroid enhancer region of this BCL11A transcription factor. So this single guide RNA was engineered and it was prepared and it was combined with the Cas9 and then it was tried in the preclinical study. So what they did was from 10 donors, healthy donors, the hematopoietic stem and progenitor cell was collected and this gene therapy was introduced into those cells. This is ex vivo. And they found that, yes, definitely in the edited cells, around 25 to 30% of fetal hemoglobin was there, while compared to in the controls, only 10% was there. Okay? So they thought, yes, this is a successful method. We could use it for treating these patients. And in addition, yes, we are always concerned about the adverse effects. So adverse effects, how it is possible here in this gene editing techniques is off-target effects. That is what they called. So in addition, if the targeted site is not affected, if some other site this change in the mutation occurs, then it is a problem. So they found, on sequencing, they found that there was no evidence of off-target editing. So this is a precise kind of an editing mechanism. So that is how it entered into clinical trials, and the famous trial which we know about, the CLIMB trials, okay, CLIMB SCD, thalassemia as well as the sickle cell diseases. So this was a brief report was presented 2021 in NEJM. So this was the result of single patient, one each, one for thalassemia, one for sickle cell disease. So that is what was presented here. So if you look at this left side is the patient 1 for thalassemia, right side patient 2 for sickle cell disease. So this patient who had sickle cell disease, so prior to the therapy, if you see, the amount of sickle hemoglobin was around 74%, the left side corner, okay? And the fetal hemoglobin was only 9.1%. So after therapy with this gene edited technology, CRISPR-Cas9, so gradually there is increase in the fetal hemoglobin percentage. So the hemoglobin level as such from 7.2 it increased to 12, and the fetal hemoglobin, which was only 9.1%, increased to 43% at the end of 15 months. Okay? So the lower graph shows the percentage of the circulating RBCs which had the hemoglobin F. So that if you see, around 3 to 4 months it has increased to 94, and by 5 months it's almost 100%, and this was sustained for about 15 months. Okay? So this gave a positive sign of yes, it is working really well, and they continued the trial. And in addition to that, they also looked at the vaso-occlusive episodes. So before this treatment, you can see there are several vaso-occlusive episodes, and after this treatment, it is almost nil. Okay? And looking at the adverse effects, yeah, so that was, I looked at it, it says 114 adverse events in 16 months in one patient. Okay? So I was really thinking, what is this? But they say that it is only 3 of them were serious adverse events, and one was sepsis, another one was cholelithiasis, another one was abdominal pain. Okay? And they say with treatment, all of them resolved, and so it is potentially safe, and this trial continued, and it had some interim analysis also. That is how the approval was given in December 2023, and the results were published in NEJM in May 2024 this year. Okay, so this was a phase 3. It was an open label, single group. It was a multicentric trial. It had several inclusion exclusion criteria. One of the inclusion criteria were that patients were 12 to 35 years of age, okay, and they should have had at least 2 severe vaso-occlusive crisis episodes 2 years prior to recruitment into the study. So this is what was done. Yeah. Yeah. So in these patients, the hematopoietic stem and progenitor cells, CD34 cells, were removed, and they were subjected ex vivo for this CRISPR-Cas9 technology. And once the edited cells were there, they were introduced. Okay, one single intravenous infusion into the patients. Okay, 48 to 7 days. So what was required here is myeloblastic ablation. Okay, using busulfan. So this is a requirement for introducing these stem engraftment occurs. So the primary endpoint of this trial was freedom from severe vaso-occlusive crisis for at least 12 consecutive months. And actually the sample size calculated was 45. And in the interim analysis, in 30 patients, they found that 29 of them could achieve the primary endpoint. And secondary endpoint, all of them achieved. Secondary endpoint was freedom from hospitalization due to this severe vaso-occlusive crisis, and also from freedom from that for 9 months. Okay, so that was achieved by all of the 30 patients. So you can see here in this graph on the left side, all these dots are the vaso-occlusive episodes, and also the second lower one shows the hospitalization for this. And on the right side, you can, that is the green color part, you can see that almost it is nil. Okay, so these crises did not occur, hospitalization did not occur. Okay, so this is about the increase in hemoglobin. On the top graph, the black line, it shows the increase in total hemoglobin, and below that is the increase in the fetal hemoglobin. And the lower graph, this blue color line indicates the fetal hemoglobin percentage, that is percentage of RBCs which had fetal hemoglobin. So by around 3 to 4 months itself, there is a drastic increase in that, and that is sustained. It is maintained throughout for about 15-16 months. Okay, so this is about the F cell percentage, and below is the how much of what percentage of the cells have been, this editing has been successful. Okay, on the left side, this is in the peripheral RBCs, whereas on the right side, it is about the bone marrow cells. Okay, so the substantial amount of editing has occurred, and it has also persisted throughout, and it has reduced all the incidences of VOCs. Okay, so looking at the adverse effects, yes, there were adverse effects, and grade 3 and 4 adverse events itself were around 95%, but most of them were treatable. And the adverse effects, you can see stomatitis, febrile neutropenia, platelet decrease in platelet count, appetite decrease, neutrophil count decrease, anemia, and so on. So that is what this conclusion from this study, that yes, it has a great potential for curing these patients, and almost all of the patients have achieved the primary endpoints as well as the secondary endpoints. Okay. Now, yeah, so this technology, what are its advantages over other gene editing technologies? Okay, so as I told you, the method as such, engineering this method has been, there are a lot of advantages, okay, and also it's more cost effective while compared to other zinc finger nuclease or the talent technologies, and it is more flexible, user-friendly, scalable. So that is what the experience of these scientists on the laboratory side claim. So, but there are some limitations. We know that this duration of these trials are short term. We do not know what will happen over long term. Okay, what are the off-target effects? As of now, it seems safe. Okay, but we do not know what can happen to the future, and issues of access. So this being a newer technology, whether this access will be there for everybody, because everyone have to establish this technique and really mimic whatever has the success of this reported in this trial. By the way, so who has done the trial is Vertex Pharmaceutical in association with CRISPR Therapeutics. So they are the ones who have done this, conducted this trial successfully. So access issues, the cost, and the long-term effects of this gene editing technology, all this needs to be looked at for in the future. Okay, some methods they describe for overcoming the off-target effects. That is some improvisation in the engineering techniques in such a way that it is specifically going to attack only the target, doesn't go and attack the other targets. Okay, so redesigning the single guide RNA, using the variants of these Cas9, like all these different names, okay, can help to localize the editing mechanism. So another method was, instead of instead of a single double strand break, make two different strand breaks. Okay, so that it is more this one. And yeah, what about the other diseases? Of course, I have shown in clinical trials. It is there for several other diseases, including cancer, HIV, other malignancies, and some other mechanisms are evolving with respect to Cas9 itself. What we have seen here, the basic simple one is just deletion, okay, knockout, okay, whereas there are other mechanisms like base editing without actually knocking out or without causing a cut in the double strand, just edit, okay, just introduce a point mutation without bringing about a cut. So that is also possible. That is by introducing some base editors. Then also prime editing is to cut it and replace with a larger sequence. So that is also possible. And also improvisation in the delivery techniques, using those viruses which can infect only one particular organ, and so that the other adverse events can be reduced. And also nanotechnology, instead of virus, you can use nanotechnology methods to deliver these. So those are all some of the improvisation. Okay. To summarize, I would say that this is a great technology, this CRISPR-Cas9. As of now, it has been well established in sickle cell disease. Okay, that is in bringing about a knockout of a single gene. Okay, so of course it is being tried out for several other diseases as well. So it definitely looks like it has a potential to cure those diseases where there is mutation of the genes. Thank you very much for your patient hearing.
A very good morning to all, and thank you, madam, for that excellent presentation on a discovery which would definitely change the lives of patients suffering from this debilitating disorder, sickle cell disease. But of course, it needs to be proven. Its safety as well as efficacy needs to be proven in the long term. So do we have any questions from the audience, please?
Ma'am, thank you for the excellent presentation. But my question is that this is a very, very interesting technology, and it will be very helpful for many diseases apart from the sickle cell disease. But one of your slide mentions that there is a problem in the access to this technology. So, and even I think it will be too costly, even if it is marketed. So What is your take on that? How can you improve the access to this technology, and what can be done about it, even for the cost?
Okay, thank you for that question. See, any technologies for that matter, any new drugs, new technologies, to start with, it is like that. It will be costly. Now the cost, they say, is around $2 million for one patient itself. So, but as the time evolves, there are many other people who are coming into this, who learn this technology. And so when the competition is more, the cost will come down. And access also will, when everyone knows the idea, okay, they just have to do, these people who are good at these molecular biology technologies, who do this cloning and all that. So they try with this, they are trying out now. So in future, when more people are able to do that, definitely the cost will come down and the access will also improve.
Okay, madam. Thank you for a wonderful presentation.
Thank you.
Just wanted to know, I don't know whether I have missed it, but what about the long-term side effects in animal preclinical studies?
Yeah, sorry, I didn't mention it. Thank you for that. Yes, they have also tried in immunocompetent mice, yeah, so it has found to be very effective as well as safer. In the animals, it has found to be safe, yes. Probably chronic studies like that, still it's ongoing. So still the results are yet to be expected.
Ma'am, has the CRISPR-Cas9 technology been tried in the telomere region? If so, what will be the consequences?
CRISPR-Cas9 in the telomere.
Telomere.
No, as of now, this is restricted to this single just knockout of a single gene.
Okay, ma'am.
Okay, so this is a smaller area. Okay, so this sickle cell disease is a very good candidate for this, because the problem is there in the single gene, and where there is a replacement of one nucleic acid, that is the amino acid there. So such kind of diseases, it has been useful, but of course they are trying out for other diseases where there are, in addition to this single gene mutation, there are duplications, deletions, all that. Trials are going on. We will have to look into the future for its application.
Thank you, ma'am.
Thank you.
Any more questions, please? Okay, ma'am, I would like to know, like you mentioned about 2 different gene-based therapies for sickle cell disease. So does one of them have any advantages or disadvantages over the other?
You mean to say the technology, madam? That is
No, the therapies you mentioned.
Okay, the whatever is current therapies like hydroxyurea and all that.
No,
yes.
no, not gene-based, gene-based therapies. Like even I read somewhere there are 2 different
For
gene-based
sickle
therapies
cell
approved.
disease? Is
For
it
sickle cell, for sickle cell.
Fetal
No.
hemoglobin and adult hemoglobin, I think.
No, sickle cell disease, this CRISPR-Cas9 is the therapy which has been
technology.
approved.
It's a technology.
Technology.
Genome
yes,
editing, yeah.
It is a technology, and it has been used also as a therapy. So that is
Okay, that's one therapy, ma'am. The other one also?
no, this is basically to continuation of the fetal hemoglobin. So fetal hemoglobin continues, and the bone marrow, there is replacement of the adult hemoglobin, which is mutated with the sickle hemoglobin. So this is the gene therapy for sickle cell disease is this only.
Okay, and this technology, as you said, I suppose you already said that this is used only for sickle cell as of now. For other disorders, it is still under investigation.
Yeah, this has been approved
Not
for
reached
sickle cell disease as well as transfusion-dependent thalassemia.
Okay, for others, it is still under investigation.
Yes, it is, yes.
It's time to conclude the session. So we need to thank 4 people. First, Dr. Jayanti madam for this wonderful presentation, for making it very simple.
Thank you.
3 scientists, Francis Mollica, who gave us the understanding what CRISPR is. Isabel and the Jennifer, who got the Nobel Prizes. So 4 people should be thanked today. And I think we should thank the first microorganism. We are celebrating the, not celebrating, we are observing the Antimicrobial Resistance Awareness Week. And one side we are cursing the bacteria. And today I think we have to thank the bacteria, because bacteria have helped us to understand how CRISPR can be used as a protective mechanism against the diseases. You also wonderfully explained the advantages over the zinc finger nucleases and the talents. So thank you so much for wonderful presentation. At this point of time, I think what we don't know is about off-target editing. We also do not know what are the long-term consequences. Most importantly, I think what we also have to understand is the ethical concerns, because as what we saw for the antibiotics, same thing might happen if you abuse this technology over a period of time. So I thank you once again for the wonderful presentation, and we conclude the session is over. Thank you.
Thank you, sir.
Ma'am, thank you, the chairpersons. Requesting the chairpersons to kindly felicitate our speaker for the morning, Dr. M. Jayanti, with a memento and a certificate. Thank you so much, ma'am. And to present mementos to our chairpersons, may I please call upon Dr. Sanish Davis, R&D Director at the J&J Pharmaceuticals, India. Requesting the chairpersons to please stay back for the felicitation. We have Dr. Sanish Davis, R&D Director, J&J Pharmaceuticals, India. Thank you so much. Thank you to all the three chairpersons for chairing the session. The next would be a panel discussion on decentralized clinical trials, new era of research. And to act as the moderator for the session, we are delighted to have Dr. Yashashri Shetty, the additional professor at SAIT GSMC and KEM Hospital, Mumbai. Madam has an experience of 21 years in teaching and clinical research. She and her team are the recipient of 43 awards for research in pharmacology. She has 110 research publications to her credit. Madam is the current chair of Sayan Poddar Ayurvedic College Ethics Committee of Yoga Institute, Santa Cruz, and co-chair of Bombay Hospital Ethics Committee. Please welcome Dr. Yashashri Shetty with a huge, huge round of applause. Requesting Madam to please take over. Thank you.
So now call upon Dr. Urmila Thatte, who is my mentor and teacher and everything. She has an experience of 30 plus years in teaching and clinical research. She's been cited enough to count among the top 2% scientists in the world. She has more than 300 publications for her credit, and she's the current chair of Bombay Hospital Ethics Committee and FMR Ethics Committee too. The second panelist today is Dr. Sanish Davis, who is a Research and Development Director of GCO Johnson & Johnson Innovative Medicine, Mumbai. He has an experience of 18 years in pharmaceutical industry and clinical research, worked in early development, phase 1, late stage development, regulatory medical affairs, and new product planning strategy. He's the president of Indian Society of Clinical Research, and he has 15 plus research publications to his credit. And the third panelist is Dr. Melvin George, who is a professor and head of Center for Clinical Research, SRM Medical MCH and Research Center, completed his MD and DM Clinical Pharmacology from JIPMER, Pondicherry, conducted several regulatory clinical trials as an investigator in acute coronary syndrome, primary prevention of coronary artery disease, adenomyosis, COVID-19, and he has published a total of 105 publications in various indexed national and international journals, and has an H-index of 22 with 1,300 citations. He's a chair of an ethics committee at Glenlegs Hospitals, Chettinad Hospital, and IMA Tambaram Ethics Committee. So, So this is a very difficult topic, actually, decentralized clinical trial. And if you see the Indian scenario, is it very important to discuss this decentralized clinical trial at that time point is a question to be answered. But never the say, like I feel the crowd is very less for this discussion. I don't know, they are having breakfast or they have left the place, I don't know, but said and done. We have to do a job of, getting into the panel discussion. I think so the people will creep in. So what were the challenges in traditional clinical trial because of which, DCT came into place? And this was actually a requirement during COVID times, which was because we understood like the non-COVID trials were all discontinued basically because of this pandemic, and then there was something which has to be taken out. So there were guidelines released by all regulators over the world as well as in India. But what was the real challenges were the slow recruitment, high attrition rate, and low diversity in the recruitment of the patients. And there was high burden at the sites, and there was limited flexibility for the patients because they have to come to that site at all, and limited time for the participants by the investigator. And there is limited access to the clinical trial also to the participants as well. So there were a lot of challenges of the traditional clinical trial because of which DCT had to come into play. So before I start with the panel discussion, our panelists are very renowned. They are very, experienced in their own area of research. So I only want to brief about what is exactly the DCT. So, so with the slides. So can you go to the next slide, please? So what is a decentralized clinical trial? It is making clinical trial more accessible and convenient through capabilities and technology that integrate the clinical trial into the participants' everyday life. Yeah, next slide, please. Next slide, please. Next slide. So if you see the traditional clinical trial, so if you see the traditional clinical trial, the data flow is from the brick and mortar side. And can I put a pointer here itself? Sorry. No, here, yeah. On this one. Yeah, nahi hota hai. Okay. Yeah? Okay. So if you see the traditional clinical trial, the data flow is from brick and mortar. You have to come to the site to do all the activities. Versus decentralized clinical trial data is always collected from the remote site. And if you see the hybrid trial, which actually is happening in India, where the data flow from the site is at least one data is at the site, and there is a one remote element which is actually taken care by the DCT. And there's a partial virtual type of a clinical trial where there are many, may have a few predefined sites for data collection along with data from the participant pool. So, and the virtual trial is out of question because that is not happening in India, which is actually taken from the real world data and evidence. So this slide actually was shown by Dr. Seema Pai yesterday. So what are the different types of DCT which we are going to deal with today and discuss is direct to participant medicinal product, local labs and alternate labs which are doing the sampling at the site, then home health care, physicians and home health, nursing. The next thing is the digital health technology, which is the sensors which are used, or the actigraphs which are used for monitoring the mobility activity or the sleep patterns of the patient, or the research concierge services which can happen at the remote site, which can be in a clinical aspect which has been covered, where the patient who has any queries regarding clinical setting that has been taken care by the clinical physicians. And there is a technical team also, where is a concierge services, where they are taking care of the non-technical thing, where they will look into the technology, feasibility, IT or device issues as well. So in nutshell, the optionality is, do the patient want to have that, or a site or the country or the trial want to do in this way. So exactly, if you have all the stakeholders now which we are going to discuss today, the sponsor perspective has been touched by Dr. Sanish, will deal with this. Investigator will be done by Melvin George, and this committee of course will be spoken by Dr. Thatte. And patient is a center point who is actually going to be taxed. So you have patient, if you see, he has to have a he has to have a wearable, and he should have internet connectivity. He should be savvy with the digital savvy. He has to be, and there will be web recruitment which happens through in the site, and screening and the e-consent happens at the web recruitment, and then the local MDs, they will look after the physical health of the patient, and the local labs will do the investigation and they take the medical care part, and there is a drug manufacturers who supplies the drug to the drug coordination center, and that is being supplied to the local site. So this is how the decentralized clinical trial actually looks like. Now we'll come to the panel discussion actual happening there. So I'll go to the side there, come there and sit and start asking questions to the panelists. So the first question, of course, will go to the sponsor. So you have the mic, no, sorry. So if you see the regulatory perspective, in the COVID times, the regulators were actually approving the study in a very fast-track manner. So do you feel, Sanish, from the sponsor perspective, are the DCT approvals at regulators happening at the same speed as COVID times, or is there a roadblock there?
So thank you, Dr. Yashashri, for the invitation to the panel. So jumping right into decentralized clinical trials and whether they are currently happening at the same level as that was happening during the COVID times, I would say one change which has become very distinct is the patients felt need to come to the site and meet the doctor. Now this cultural thing will not go away from India. Unlike in maybe the West, where patients are comfortable if they can actually just meet maybe the doctor once in a year or once in 6 months, etc., even if they are part of clinical trial. Unlike that in India, we have interviewed several of the sites while we were actually checking on decentralized capabilities, and they say that many of the patients, even if you tell them you don't have to come, they continue to ask that, but why? Why can't we come and meet you? So that's a cultural nuance which won't go away in India. I mean, whatever we do, whether we make technology more closer to the patient's way of life, they would still say that we would still love to come and meet the doctor. So that's one. Now, globally also, the number of clinical trials which have these capabilities in them are still as many as they were during the COVID time. So that has not drastically decreased, even though countries like ours or maybe countries in Asia Pac would have this cultural nuance added to it. Now, even if you look at regulatory agency, the DCGI or CDSCO, they do not distinguish between a DCT trial or a non-DCT trial, because even before COVID, a lot of these elements have already started to come in. Like for example, e-COA or electronic, you could say patient outcomes assessment. These outcomes assessment like PRO instruments, etc., are already moved into a tablet form or a mobile form, etc., even a few years before COVID. So that has accelerated. So in fact, all the trials that are run, which are multinational or you could say even Indian or R&D organization which runs trials, all of them have this component which is baked in. So there's no going back now. That's going to increase. There are also a lot of companies who are actually bringing in e-consents and others which are there. And if you also look at the list that you actually showed of all the different kinds of capabilities that are there, many of them are already even there in the routine clinical practice. We have telemedicine, which is a very routine thing. In many of the orthopedic hospitals, there is already a tie-up with the home health nursing and others. So they are more than happy to actually have that. So many of these are following what is there in the routine clinical practice. So from a regulatory perspective, there has been no perceptible difference in how the regulator looks at a DCT versus a non-DCT trial, partly because some of them are already been there and it is just accelerated. Some of the others are already there in clinical practice, so there is nothing which is different that is being done in a clinical trial setting.
So the next question will be to ethics committee, which actually is thought to be a hurdle by many investigators and sponsors as well. And yesterday also we heard from many of the speakers like how ethics committee delays the process. So when a DCT study is presented to an IEC, what are the critical concerns that arise from ethics committee, and what specific aspects of the decentralized element which ethics committee concentrates on?
So according to me, there's no difference, because I think that the principles of ethics remain the same wherever you go and whatever you do. Whatever trials you do, it doesn't matter. You have to follow the principles of ethics, which are justice, beneficence and autonomy. So I think that principles remain the same. So we need to remember this and follow those principles. The probably the important things that we have to remember while doing decentralized clinical trials is privacy, because it is possible that somebody, delivers a drug to a patient, and that drug is delivered to the wrong person, and that patient might have a loss of privacy in terms of his, I think it would be horrible if a person who has HIV is somebody else knows about your condition. That would be difficult, I think. It would be a gross invasion of privacy. The second thing I think is important is risk-benefit. How do you look at the risks? What are the risks? According to me, the first risk is loss of privacy. The second risk is, of course, the physical risks, because you are in a decentralized place. You are not in a site. So therefore, who is going to take responsibility? You need to look at who is going to take responsibility of the patient and how will they follow up on that patient. That is something that's very important. The other things that you will need to look at is e-consent, E-consent, the electronic consent, I think you have more experience with that. But electronic consent is, according to me, difficult, because if you're doing a thing on Zoom, then, as Melvin said, you may just lose connectivity at some point or the other, and you will be in trouble. Because if you've lost connectivity, you've lost it. So these are some of the challenges I think that you will have to face.
So Melvin, as an investigator, how is the screening and recruitment of participants is undertaken in a DCT? And what are the challenges really faced as we have actually bought DCT to include diversity? Because we don't have a diverse group of patients in non-traditional clinical trial. And how you take care of this?
Firstly, thank you, Shetty ma'am, for giving us the opportunity to be part of this panel discussion, and also to the organizers. So, firstly, I would look at the whole concept of DCT as a welcome thing, because today we are living in an era where everything is being delivered on the doorstep, be it our food, be it our groceries. So, why not even trials, if not the whole thing, at least part of it? Because the idea of DCT in the first place has come because of the many challenges in traditional trials. We see recruitment challenges. We see people who are not able to continue being part of trials. They drop out halfway because they find it difficult to come to the hospital. It's a long visit to reach the hospital, that too frequently. So because of all these things, even 20 years back, this whole concept of DCT came, and of course, COVID has made it very popular. So when it comes to DCT, as far as screening is concerned, we could use the traditional methods like using electronic health records. We could use social media advertisements to make people aware that such a kind of trial is happening, and then give them some kind of a contact detail so that they connect back to us. And once that initial connection is established, you first do the electronic consent process. And once the electronic consent process, and even for doing electronic consent, we have established systems in place. There are softwares like RedCap software, DocuSign, which give a good deal of privacy and confidentiality and all that, to ensure that there is a standard process in it, and even the validation that of the identity of the patient. Like, for example, a two-way authentication system which we have for other things, like one is to, first the patient has to put a password and followed by a OTP. All those standard processes are followed even for electronic consent. Once the consent is done, then we talk about the screening procedures. So for screening procedures, they might have to give their sample to the local lab, and those reports are in turn analyzed by the investigator at the site, and if they fit in the study, they would start the process of monitoring. Now when it comes to monitoring, again, wearable devices and all would be used. So these are some of the aspects of screening and recruitment. And of course, diversity, you spoke about the challenges. So the whole process of DCT rests under one assumption that our patients are in a position to handle all these devices, to handle all these processes that are being employed in the DCT. But we all know very well, India, there are a lot of people who would not fall under the bracket. Maybe they have a smartphone, but then they might not be very adept at using these new tools, new devices. It might take some degree of learning for them. And for some of them, they might require more than just an initial one-time training. They should be handheld throughout the study so that they are really doing it the right way.
So, from the sponsor point of view, Sanish, among all the decentralized elements, which one of this you feel is the most challenging?
Yeah, so currently the home healthcare nursing, what we call as a nurse or a doctor actually going to the patient's home, making the visit. If it is a doctor, then you can actually do many of the, you could say, physical assessments and others at the patient home. Whereas if it is a nurse who's going, I think the routine investigations can be taken, some physical examinations can be done, etc. But all said and done, I think this might be seen as a routine now in clinical practice, right? I mean, you have patients with an orthopedic condition where there is a person actually coming at home and giving physiotherapy or other things that are there. But in a clinical trial setting, one of the big challenges is how do you actually ensure that the data that is collected is appropriate? Now, there can be tablet-based things where you can actually key in data and that kind of like goes and syncs with the database for the study and then that is evaluated, etc. So all that can be there. But what if you could say when the patient is being seen at the site, at the home by the, you could say, the home healthcare practitioner or the nurse, the patient is completely okay. And probably the next minute or after the person has gone away, there is an event. And the patient does not report that event because they feel that, oh, I already come and already somebody has come and seen me, so it should not be of an importance, etc. So these are some nuances which we would probably have to kind of like give more thought in these kind of settings. Many of the other things are all technology based. So if the technology works well, it works well. But for this one, which requires a human interface, that's where I think a lot of the other factors actually come into play. And if you are not able to control those factors or understand those factors, there could also be safety issues. There could also be underreporting. There could also be perception that clinical trials are being used like a technology thing and not like a doctor-patient kind of relationship. So all these things can actually be a challenge from one capability perspective.
Shasri, can I ask one question? Melvin, I want to ask you, do you think that there is any bias, selection bias, in choosing your patients when you have to make sure that they have adequate training and things like that? Because then that would go against our principle of justice.
Absolutely, ma'am. So when we have this kind of situation where all the patients who participate in the study need to have this capability of knowing how to use all these devices, these tools, it does give some amount of bias, a lot of bias in the study. So to overcome that, there are trials which have a dual mode where we have both kinds of recruitment happening, where people who are confident with using these tools, they would be recruited through these processes. But for those who cannot use these tools, the traditional clinical trial mode would be employed for them. So this dual mode is being used, a hybrid mode, for the recruitment and for the monitoring processes, so that that part of the population is also served, is also recruited, and thereby the diversity is maintained.
So from the ethics committee perspective, like, for our own objective is to protect the rights, safety and well-being of the participants, and data credibility is a very important part of it. So how ethics committee actually does the risk benefit analysis in a DCT?
Very good question. What are the risks involved in what are the benefits? I really have no clue to the answer to that question. The risks, according to me, are loss of privacy, potential problems with remote sites. I mean, or not, event or not. And if he has it, then whether they are reporting or not. I mean, it's so different from a person who is there at the site, or there is a site where this is happening. It is different from that. I think that is one of the most difficult aspects.
So from a sponsor perspective, Sanish, like for example, you're using a lot of digital things. So digital health technology devices are all validated for research use. If not, there are many devices which are not. How reliable is the data generated from these devices, and how is the quality assured?
Yeah, so maybe let me start by giving an example. So we had several years back a study where the patients, you could say, blood sugar values were being, you could say, measured by a device. I mean, now it is very common, but several years back it was not that common. So you strap on that particular device and it kind of like has a subcutaneous element to it. It continuously samples the, you could say, the capillary or the venous, the venous blood, not the capillary blood. And then it kind of like gives the data of what is the blood sugar levels that are there. Now, when that study was run across the world, not just in India, but across the world, we found that if the values are not monitored on an ongoing basis, there are a lot of these events which are sometimes not picked up. Like the patient has sometimes events. Patient doesn't, you could say, have that perceptible understanding that, okay, this is an event. Maybe in a Western hemisphere country, those are much more commonly reported. So you had a huge skew of adverse event data across different places. Countries like India, where even sometimes people have said that they had feelings of, you could say, giddiness, etc. But they were not being reported because the patient felt that that's part and parcel of my, you could say, disease. So while the technology might be well established, etc., and validated also in many ways, but if it is not monitored in the way it is supposed to be, that can still be a challenge because it is leading to safety of the patient. So let's kind of like then go to the next level where the technology is well established, but it is not validated from the perspective of how it can still take care of patient safety. That's going to be a bigger issue, right? Because then there is no way that anybody is monitoring, and we also don't know whether it is supposed to pick up the events that it is supposed to pick up. So I think that's where I think the regulatory agencies like FDA and others, which are at the forefront of, you could say, approving many of these technology, and it is now called as software as a medical device, SAMD. So software as a medical device is now a very well established discipline within medical devices itself, because it's not just a medical device, it's a software which is acting as a medical device. So those things are becoming much more stringent in the way the guidelines are framed. A simple example, patients with Parkinsonism have a lot of tremor. Now, there are a lot of gloves which have been developed which can help the patient stabilize his or her tremor. Now, I mean, there are also, I also sometimes sit in an ethics committee where some of these questions are asked. We had people who are developing these in India, and they are just feeling that, okay, this technology is being developed elsewhere in the world, so we can also just develop a glove with magnetic weights and others, and it should still be okay. And we had to counsel them to say that it doesn't work like that. You have to have technology which can monitor the patient safety, how many times it's so. Even if it's a technology is established, the validation and monitoring for safety is paramount before it can actually be implemented for large scale use.
So for Melvin, in e-consenting actually, what are the challenges faced? Actually, for like how the verification of an identity of the investigator is done, and how the trial participants actually perform, and how it will be determined that the trial participant has, comprehended and understood the information and has voluntarily been participating in a study.
Yeah, certainly electronic consent is a challenge, but there are certain processes in place. First is to verify the identity of the individual, as I mentioned earlier. So there is a process wherein the person would be given a username and password where they log in, and after that, once they confirm their identity with by putting in their phone OTP that comes into their phone, once their identity is verified, this could be one way, or they would be asked to have their biometrics taken. So biometrics also are used sometime, or they might be asked to upload one of their ID proofs. So any one of these measures are used to confirm the identity first. And after that, the whole process of consent is based on the app that is used for the consent process. So the challenge in any application that we used to have an online meeting still remains here, because there could be people who tell us, yes, I can use the phone for a online video call. I have a net connection. But then when they are actually at home during the meeting, we find that they might get cut off at periodic intervals. And those times when they get cut off, the investigator might be telling something very crucial, which he might assume the patient has received it, understood it, but at that point, patient might not have been even there online, and he might not even know. So these kind of things could happen. And to circumvent this, we could have something like a questionnaire placed after the consent is over, where the patient has to fill certain questions, answer certain questions. So only if he has understood the consent in the first place, will he be able to answer those questions. So that kind of questions need to be put across to him. The other aspect of electronic consent is this really a place, a room? Normally when in a traditional consent, we take them to a room which has privacy. But now, when the patient is at home, the screen might appear as if there is nobody around. But we'd never know. Of course, it's their bedroom, it's their hall. There could be somebody sitting back. So maybe they are being coerced to participate because there is a benefit for the family. So those things cannot be ruled out. Or the patient might not be very forthright in telling certain things because there is somebody around them. So these kind of issues are still there. We have to figure out ways to how we can circumvent these things.
So another thing is, how is the privacy and confidentiality of the participants is maintained? So that is also the biggest concern of ethics committee as well. So when the direct to participant medicinal product is supplied, and many times the database sources which are actually accessing remotely all the sites, they are a third party altogether. Are they being there always in the agreement between the sponsor and the investigator, or the tripartite agreement, whichever?
Yeah, usually all the stakeholders who are part of the different processes of these trials, they are into the agreement to ensure that they will also ensure confidentiality throughout the process. And there are obviously data privacy laws in place in the US and the Europe, which need to be stringently followed when you're doing a DCT. At the moment in India, these laws are still in the process of, it's not fully formed. And even in the Europe, for that matter, there are a lot of ambiguous issues when it comes to providing door delivery for investigational products. I still know that there are a number of European countries which do not permit direct delivery of investigational products to the homes. So it is all a law that is in the making. So maybe we'll get more clarity in the years to come.
So Sanish, how is a DCT study monitored? And if you have monitored any of these studies, because as Satya Madam kept on telling, there is no site. So site monitoring is out of question. So what were the major findings of that monitoring?
Yeah, so our experience in India has been mainly with ECOAS, what we call as Electronic Clinical Outcomes Assessment, what we call as the PRO instruments or Patient Reported Outcome Instrument. So many of you would remember that quality of life, that or QOL, that's the commonest one that you actually record as part of patient reported outcomes. In many of the earlier studies, it all used to be paper. And what used to happen is that many a times patient would actually, while traveling to the site on the day of the visit, they would just start scribbling in that subject diary or patient diary as such. So it used to become, not only in India, but even in the Western, you could say, well-developed countries, there have been studies where it has shown that patients used to fill these in the parking lot when they were coming to the site. And it was absolutely worthless data that used to come out. But in today's time, the electronic, you could say, outcomes assessment of these kind of scales and others that are used, rating scales that are used, are all in tablet form or any very, what they call as provisioned devices, which means that it's a device which is given by the sponsor. Even if it is a smartphone, it will only have that. You can't use anything else in that smartphone. But what is the best thing is that the data actually links up as soon as the patient is within a Wi-Fi range or within, you could say, the hospital's perimeter, etc. So there is that data trail which can be tracked by the sponsor as to when it was entered. So you can't actually backdate data, etc., and still get away with it. Of course, very smart people can still maybe do all that. But I would say 99% of the time, the data can be tracked. So there is a data tracking that is possible. And you can also investigate as to why there has been a data blip or why there is no data for a particular point in time. And there can be interventions that can be used. Like, for example, the site coordinator will get an email from the central database group by saying that this particular patient doesn't seem to have entered data for these 3 occasions or 2 occasions. Can you give them a call and check as to what is happening? Is there an SAE or is there an AE which has happened at that point in time? So these are ways in which I think sponsors are now much more surer of the data that is being collected as opposed to an earlier time when these were not done. Now that's the commonest one of ECOA. I'll also talk one small thing about the home healthcare nursing, which we have implemented in a couple of studies. Again, there are not too many sponsors in India who have implemented many of these things. That's also something to note. So this particular organization, which was helping or rather has been contracted to do the home health care nursing, they have also a particular database that they can actually key in the data. And that syncs with the study database. So again, you have an opportunity to actually monitor what is happening. Sometimes in real time also. I mean, for initial few patients, because we also didn't know how this will all go, we said we will do real time monitoring. So we asked the nurse to be there, switch on the Wi-Fi, switch off the Wi-Fi, so that we can see that, okay, whether the data is coming online or not, etc. But those are like really the pilot part of how this has been implemented. But again, these are ways in which technology also enables you to help in monitoring of the data in much more real time than sometimes even when the patient is at the site. Yeah.
Can I ask a question again? Is it possible to hack into these devices?
That is the biggest threat.
I think Madam is one of my, you could say, technology aficionados that I always kind of like look forward to. Yes, I think if somebody really wants to do all this, the world is their oyster, right? But typically I think the amount of firewalls and the amount of gateways that are there to actually get into these things, I wouldn't know what a person will get by hacking into a database like that. Yeah, but that data is of no use to a person without, you could say, the other parts of the study or the other data, right? I mean, nobody can actually sell this data in an open market unless they want to actually ransom that company by saying that, look, I have all your clinical trial data for this study, and you could say, what will you, what amount of money you will get. But there, what happens is that the data is also backed onto another server. There will always be a backup server or a third server, etc., which are usually in a third country than in the country where these studies are happening. So in that way, I think many companies hedge their risk of even ransoming and all that stuff. Now you give an interesting premise for maybe an Indian movie which can be made on this, where maybe I think we can have, you could say, a third-party black actor or a deep state actually ransoming an ICMR studies data and saying that आपको छोड़ेगा नहीं जब तक आप पैसा नहीं देंगे.
How does EC, madam, maintains oversight on this type of trials?
It's very difficult, I think, because it is remote. Everything is in a remote area. So in the same way as we'll have to go to the there is no site. So we will have to go to the data sources and do a monitoring of those sources, and if it is actually done or not at the time that they have said that they have done it, these kind of things we will have to check. So this is, I think, what is, I think according to me, it is the most difficult thing to do, that is of monitoring these studies.
So Melvin, in case of an emergency, is there an effective communication plan in place? And who are the parties to be contacted if there is a delay in a device redressal? And is there a backup? For example, if there is no backup, what happens to the data lost? And are there any protocol deviations such reported by you because of the device malfunction and the redressal has not come on time?
Yes. So traditionally speaking, all the, trials which employ all these devices, they would be having a point of contact, first point of contact, second point of contact for any malfunctions. So classically speaking, they are first the CRA of the trial. Usually they handle the initial process, and if that doesn't work, there'll be a person who's directly responsible for that particular device. And many times this is a challenge because that person may not necessarily be of the same country. It might be some other country, and then connecting to them and getting them to understand what is the problem with the device, because even the site doesn't have the device is of the patient. So that connection does take a lot of time. In fact, sometimes some important outcomes might be missed because of that time duration when that problem is not solved. But a mechanism is usually established and placed to sort out these kind of malfunctions. Besides that, there are also alternatives arranged in case you are not able to get data of a particular kind from the device. An alternative for the same, maybe it might have to come physically to the site for giving some of the endpoints, so for addressing some of those outcomes. Those kind of alternative arrangements will be usually there in all clinical trial protocols.
So what are the site challenges while dealing with these technologies, devices, semantics, service providers and data flow? There are so many things you have to manage now.
Yes,
it was
certainly.
very better when it was a traditional
so
trial.
it does take a lot of learning for the site first, to be confident of handling these devices, to be confident of answering the queries that the patients will come up with. In fact, there's a lot of training that has to be done at the site level, and even the patient, it's not just a single one-time training. Multiple trainings could be needed, and even compatibility of the devices also with existing software that is present in the site. All those things are challenges which potentially have to be changed.
So Sanish, this will be the last question, then we'll put the thing open to the audience. What type of studies actually can be undertaken by using a DCT? If we say that India will be digital health in next 10 years, and which are the studies which are not to be done by DCT?
So there are, you could say, very specific kind of studies which can be run completely on DCT capabilities. Maybe if I am taking an example of maybe a hypertension study, or maybe a study where a wearable can be used. Like for example, if you want to check what is the, you could say the amount of walking a patient with a heart failure can do. Maybe that's a study which can be run as a hybrid mode where you can actually find out how many times does the patient have an issue walking, or how much amount of walking can be done. So data which can be collected easily through these devices without actually, you could say, also wanting other attributes of that data. Those kind of studies can still be done easily, and those are the studies which are commonly reported out that are there. But if you have an, you could say, maybe an infectious disease or others where you really need blood test and others to actually determine what's happening to the patient, those may not be the studies which can easily be done. You could say ICU based studies cannot be done. Acute condition studies are not those ones which might be very amenable to a DCT thing, but more like a chronic one. Parkinsonism, sleep disorders, those are studies which you can easily do using a wearable device or other devices which can help you. Yeah.
Yeah. Melvin.
I would say anything which is looking at an event-based study can be easily taken care by a DCT. Because like for example, you're giving a drug to prevent an outcome, let's say to prevent an MI. So MI is going to be a one-time event, or maybe twice it might occur during the 2 years or 3 years. So that is something which requires less monitoring as long as the drug is relatively safe, but you're only looking at finding that preventable outcomes. It is easy to do such kind of studies as in a DCT mode. But if you're having a study where you have to measure the tumor size progression, reduction of tumor size progression, they might have to come to the hospital at regular intervals for the MRI or CT scan. That cannot be done in a DCT mode.
So if you see the DCT scenario, to summarize before I put the thing for the forum, so if you see the sponsor side, sponsor actually is feeling like the cost has decreased, there is a lot of diversification of the thing, and that is accessible, like, you know, there is a lot of burden of course, but then they can have access to the patient. Ethics committee has a lot of challenges of privacy, e-consenting, digital health validation actually, because if it is not validated, we are very much concerned with the data, and also of course of keeping an oversight. An investigator has a big challenge of you know, managing everything at the site. He's there at the site, but he has to do a lot of access thing to, from labs to nursing care to, you know, patient data itself. So there is a lot of you know, juggling around between the three stakeholders. So in nutshell, I'm summarizing what has been discussed till now in panel discussion, and now the forum is open for all the questions from the audience. Please.
So any specific concerns or additional concerns for the vulnerable population for the decentralized trial, especially for informed consent?
I don't think we should do these studies in vulnerable patients. I don't think so, because especially in children, because it will be difficult to administer the e-consent without actually being in front of a patient. I think it will be very difficult.
Yeah, certainly it adds to the challenge. So like we always say, for vulnerable participants, extra measures need to be put in place. So we need to have a policy in the protocol which says what extra measures are we going to take if you're going to take a patient who's vulnerable in a DCT. So throughout the stages of the trial, that part has to be addressed.
Of course, extra measures we have to talk about, but what extra measures will you use? According to me, it is very difficult to choose extra measures. So it is better not to take these participants. It's best to do them in adults, do these DCTs in adults, in non-vulnerable population.
My question to all panelists is that, especially to the ethics committee point of view, if it is a traditional clinical trial, while going the monitoring, it is very simple to get every data at one place. But as in DCT, it is not possible. So my question to Madam is, what precaution as the ethics committee member we should take care while going ahead with the monitoring of any decentralized clinical trial?
What precautions to take? As an EC member, the precautions that you should take is that you should verify the data properly. Whatever data is collected and submitted to the ethics committee, you should verify that. There's no other way. There's no site that you can monitor. So you'll have to make sure that the data that is submitted to you is accurate. And it is, how will you verify that? I don't know, how can you verify that?
So what we have seen in some of the ethics committees that we have interacted in the Western Hemisphere again, unfortunately, is that the level of, you could say, ethics committee members' understanding of how the data flows is probably the first thing to be kind of like, you could say, taken care. If you don't know how the data is all intertwined together, then you might think that, okay, looking at only one part of the data gives you all the answers. So we actually then thought of, you could say, putting it to the Indian context, how many of our ethics committees are willing to take these additional steps to actually look at the data? In one of the ethics committee, which I was also part of, there was huge resistance to even go and monitor a site. Now this is like next level, which means that the ethics committee has to divide the work between 7-8 members, and each member has to look at, okay, what is the data that is coming from one instrument or one capability. That itself is huge. So I don't think that this is going to be any easy on the ethics committee, because you can always say, sab acha chal raha hai. Right? So
I think I agree with you that it is easier to say sab acha chal raha hai, but it is very difficult for your conscience.
Yeah. Madam, I have a question here. First of all, I must say I really enjoyed the talk and really wonderful. There was a talk about that there is limited doctor-patient interaction as far as the decentralized clinical trials are concerned. So that being the case, do you think, in your opinion, the attrition rates and dropout rates, are they any higher as far as DCTs are concerned, or are they equivalent to the traditional trials?
The whole idea of DCT is to ensure that the patient doesn't have to come to the hospital too frequently, so that there are no dropout rates, so that the dropout rates can be minimized. So as long as patients get the hang of what they are expected to do during the DCT, the dropout rate should actually reduce. That is what many of the data has shown to be. So it all depends on that particular trial that we are talking about, how much the patient really is benefited because of the drug in the first place, and then how easy it is to carry out the various procedures that a patient is required to do. I know sometimes there are some patients who, the minute you say that you have to do XYZ things, they kind of lose interest. Why are you giving me so much additional burden? Already I have a disease, and then you're giving me a lot of homework. So I don't want to participate. So this, if we have patients, a lot of patients giving this kind of response, so the number of patients are going to reduce over a period of time. So in other words, if you're doing a DCT, make it simple. Make it simple for the patient, so that the dropout rate will not be as high as what you see in a traditional trial.
So are there any more questions? Yeah, please.
I have a question.
Yeah.
So the question is to Dr. Sanesh, is it, have we seen a site-to-site variation when it comes to the conduct and then the results that we get from the individual centers for a DCT? Because of the execution related issues.
At least in India, we haven't seen it because these are all very regimental, right? I mean, there is no way that the site can use its innovation to actually do anything differently. But maybe in less structured situations, maybe that's where I think the data might pan out. So for example, if you are supposed to actually do a wearable study, what if there are sites which are in the, you could say, remote tier 3, tier 4 cities where the Wi-Fi connections are not as great, where there might be electricity cuts which might lead to wearables not being charged fully, etc. So those are situations where we would actually then see the data being teased out. But in the current situations that are there, any data gaps are immediately flagged and the site will have to find out why that has come out. And the sponsor also has to do that additional monitoring, etc. So right now I don't think we will have that answer, but yeah, when this is kind of like much more large scale, maybe those kind of differences might come up.
Melvin, I need to ask you this question. How many patients have dropped out from your trials? You've done DCTs, right?
Our experience with DCTs have been limited, but one of the things that we always get a complaint from patients is that they are not very happy with doing anything that has to be done at home with devices. That's a general complaint. So, because many of these patients are very simple people, so this extra work that is being given to them is not looked at in a very positive way. So that does affect, but because it is not too much, they are still in the trial and they're continuing to be a part of it. But I can definitely say that the more complex you create a system where they have to key in a lot of variables and all that, where they have to do something more than their usual things of the day, it is going to affect the dropout rate.
So I think so we are coming to the end of the session. Renuka, you want to ask one last question, please?
Okay, sorry. Just wanted one question. I mean, we are talking about using DCTs, but you know, like public sector hospitals where the patients are very, illiterate most of the time, or they are not very tech savvy. Aren't we the fact that we talk about diversity getting lost somewhere in this decentralized studies? And a second question, just to take forward what Madam had said about
Selection
hacking.
bias it is.
Yeah, it becomes selection bias full force. I mean, you're missing out on a large population.
No, that is what I said about ethical principles that will be compromised with the selection bias.
Exactly. And
justice is compromised, right? Because you end up in
you're choosing your patients.
Choosing your patients.
Yes.
But maybe, I'll have the last word. We kind of like have moved away from this in India at least 7-8 years back. The moment that we had this big issue with the Supreme Court, etc., breathing, many of us as sponsors have stopped having any sites to even recruit illiterate patients. We have stopped. If we have sites which say that we might be looking at patients who are not literate, we'll say don't do it. So the impact on us as a sponsor is going to be much more than the number of patients that we might get from doing that. So as sponsors, many of our companies have said no illiterate patient. So I feel that itself is leading to the diversity issue and inclusion issue. But that's the unfortunate state of how we are running clinical trials in India, because we are now more worried about legal, patient advocacy groups, Swasthya Adhikar Manch, etc., kind of like using these as a tool rather than thinking of diversity. Whereas the Western countries are looking at more as to how you can spread the diversity and inclusion, including the US, which has now clear numbers, percentages of how many you should recruit from each different type of population. And so we are moving in the other direction. where we are trying to say that we are ethical, but we are unethical now, because we have shut down a large chunk of our population from having any possibility of getting into our clinical trials.
So I think so we can end with the panel discussion. I thank all our speakers, Dr. Thatte Madam, Dr. Sanish and Dr. Melvi. And I think so we are out of time because the next session will going to start. Thank you very much for the organizers, Dr. Ganesh Dhakli, who has given us this opportunity to conduct a panel discussion. Thank you very much.
Thank you so much, ma'am. May I please call upon Dr. Meera Desai, Professor, Dr. M.K. Shah Medical College and Research Center, Ahmedabad, to please present mementos to our moderator and the panelists as well. May I please call upon Dr. C.M. Kamal to felicitate our moderators and the panelists. Thank you so much, ma'am, for the wonderful session. And now for our panelists, Dr. Urmila Thatte, Dr. Sanish Davis, and Dr. Melvin George. Thank you so much to the moderator and the panelists as well. And thank you, Dr. C.M. Kamal, for doing the honors. Now time for the plenary session 2, and the topic would be Therapeutic Drug Management and Clinical Decision Making, a Real-World Experience. The speaker would be Dr. Smita Patnaik, Professor, PGIMER, Chandigarh. And to chair the session, can we please welcome with a huge round of applause Dr. Asha Pathak, Professor, Uttar Pradesh University of Medical Sciences, Etawah. Can we please also Dr. S. Mohammad Mustafa, Professor, PhD, Institute of Medical Sciences, Coimbatore. Dr. Shashidhar Banale, Professor, MR Medical College, Kalaburagi. Please welcome all the three chairpersons with a huge round of applause. A warm welcome to our chairpersons, requesting the chairpersons to kindly take over the proceedings of the session. Thank you.
Ladies and gentlemen, good morning to everyone. Let me introduce the speaker, a famous and clinical pharmacologist and in the field of therapeutic drug management. It's my pleasure to invite Dr. Smita Patnaik. She's professor of clinical pharmacology at Department of Pharmacology, PGI Chandigarh. She has extensive experience in the therapeutic drug monitoring, in clinical pharmacology of immunosuppressants, anti-cancer, and genitourinary drugs. And she is also investigator-initiated. She has done investigator-initiated clinical trials and clinical research, methodology, biostatistics, and SRMA. Now I welcome the speaker.
Thank you very much, ma'am. Thank you so much. And at the outset, let me let me extend my heartfelt thanks to Dr. Ganesh and Dr. Kamal for inviting me to this conference. Indeed, they need a huge round of applause for conducting this in such a nice manner. I really appreciate the hospitality and the arrangements, everything. Nothing could have been better than this, I feel. And with that, since we are running late, let's start to the topic. And somehow we, as clinical pharmacologists, probably have a bit of identity crisis somewhere, where I was also wondering, medical pharmacologist, then other pharmacologists, then we have combination of pharmacologists where we have pharmacists and then PharmDs and MD pharmacology, then DM clinical pharmacology. So where are the things where somewhere we are not able to find the sense of purpose for our future journeys, or having some sort of identity crisis? Where do we stand? Do we stand in the midst of a lab delivering services, or are we in a clinical trial setting where we are delivering clinical trials, or we are in a CRO delivering these services, managing sites, conducting trials, or we are in a hospital where we are delivering the services to the patients, or we are in a lab where we are doing some biomarker research, doing some cutting edge thing where we are trying to connect the lab with the patients, etc. So having said that, we know that things are they have diversified, so huge diversification that a pharmacologist or the future generation pharmacologist have to kind of stick to one kind of stream, because the horizontal growth is so much that you need to find a niche area for yourself and then kind of grow vertically, because vertical growth will keep you remain standing on that platform or in that area for longer period, that we all know. So I have been an advocate of therapeutic drug monitoring. It may seem very funny, but it has funnily happened over a period of time, and sort of just happened. Whereas I'm not the only person who is working in therapeutic drug monitoring, and there are so many other working in this area. And I am trying to kind of reignite what used to be done in India in 1980s, and because of the logistic issues, somewhere this has become dissociated from pharmacology. And if it is dissociated, then obviously the other fraternity is going to take over, you know. And somebody, yesterday somebody, one of the madam said that, see, pharmacokinetics is our forte, is the forte of clinical pharmacologist or pharmacology. The way we understand pharmacokinetics and pharmacodynamics, possibly the other fraternity does not understand it that well. And therefore, when it comes to drug management, therapeutic drug management, where the clinician's thinking stops, probably we can add value using these principles to make a difference. So that's the mission that we are running for this advocacy. And with that, I would like to take you through this journey and try to make it very, very simple. And when I was looking through the program and I spoke to Dr. Manji, we also saw that there's a parallel session which runs, which says TCI, TDM or TCI. So I wish we should have a debate, debating here, and then would have make a case for it, and then probably would have made the audience believe that they converged to a single point. Thank you. Anyway, so let's begin. So we'll walk through all this. We'll try to understand what is therapeutic drug monitoring and what is therapeutic drug management, and why I did not say that it's monitoring but it's management. And then TCI is already being covered in the other hall, but I would briefly touch upon, because I can't go to model informed precision dosing without going through TCI. So to tell you the thing that in 1960s, therapeutic drug monitoring as a term was actually introduced, and in 90s, Nick Holford actually introduced the term TCI, which is the target concentration intervention for the MD students. Go back and read the Nick Holford series on target concentration intervention, because he tried to bring out the disadvantages or the shortcomings the therapeutic drug monitoring actually had. And then the modern time has actually sort of given us something called MIPD, which is like model informed precision dosing. To be very honest, we are not yet there. This is being done in limited centers across the world, but yeah, we are a different ethnic population. We really need data which is our population specific to generate our own precision dosing models. And the migration of a therapeutic drug monitoring into therapeutic drug management is through MIPD. And then I really would like to urge all the clinical pharmacologists to adopt TDM, because we can't look for the best of logistic. I was really amazed by Dr. Abhay Bank's talk, a very hearty talk, where he said that if you are waiting for, if you are in a situation where everything is perfect, probably you are the problem who doesn't want to do. And therefore, whatever limited resources we have, let us start doing the TDM. And it will just start, it may just start with a survey in your own hospitals, where you find out which are the areas, which are the therapeutic areas which probably need your intervention through TDM. So this is the historical perspective. Like I said, in 1932, people measured drugs where we had these only sulfonamide at that time, and people started measuring drugs through, because chromatographic methods came in the early 19s. So that's when people started measuring drugs, and as I said, in 1960s, this with the anti-epileptic drugs, where we saw that the therapeutic efficacy and these, especially the toxicity, which could be aborted only by doing the monitoring. So that's when the term was coined, and this is the period where I say that in this depicts this in historical perspective, basically 1932 to 1981, where the therapeutic drug monitoring as such got established. You see introduction of HPLC, correlation of serum saliva concentrations. Then in between we also have the ELISA techniques coming into monitoring. Then in the meantime, we also have the development of the free drug concentration, then the drug-drug interaction, first structured review on therapeutic drug monitoring that was published in 1965. So with this legacy in pharmacology, we moved on with the therapeutic drug monitoring. And there we go, is the modern therapeutic drug monitoring, where we know that it's a multidisciplinary science and it's a clinical science, where we are individually adjusting the doses of the drug, which is in conjunction with the clinical experience or the clinical trial data, which has shown to improve the outcomes. Now, if you see, it's a full circle, you know, it's a full circle. It starts from the patient and comes back to the patient. starting from the sample collection, sample analysis, result interpretation, and then you use your medical judgment to adjust the dose. And somewhat it's kind of personalized dosing, but it is not true personalized dosing. Why? Because we have defined the therapeutic ranges for the drugs, and these drugs has not individually been tested for this same patient, but this is derived over a population. So, TDM essentially would say that you can do a priori kind of a TDM or an a posteriori kind of a TDM. By saying this, I just mean to say that the a priori means you already have some information about the drug, you already have the pharmacogenetic markers that would affect the levels, and the demographic and the clinical information of the individual patient. Now, a posteriori would mean that you are actually also considering the PK and the PD monitoring, and then adjusting the dose levels as per your requirements. So, it can be practiced either ways. Either it's a priori thing where you are looking at the initial doses that has to be used, or it's a posteriori thing where you are modifying the dose based on certain information. So, that's the transition that we have made now. We are monitoring itself is a very sort of a passive term, and we no longer want to be passive in terms of our approach to TDM, and we really want to do a reactive kind of a thing rather than from a reactive mode we actually want to go to a proactive mode. So, that's and then we were talking about the decentralized trials. You look at this. So, now we also have the sensors and wearables for drug monitoring. So, you can actually have a real time monitoring and then a real time feedback for dose modifications. So, the therapeutic drug monitoring remains becomes therapeutic drug management, but please remember the TDM remains as TDM only. So, again this is some Krishna consciousness says that we all change, only we change our bodies, our soul remains the same. So, the essence of therapeutic drug management is like this. I would say it's an old wine but a new bottle, right? Or is it a new wine in an old bottle? Any differing thoughts? Whatever it has just changed its name and the way it is delivered. Okay, with this when we wrote this manuscript, we were asked by the reviewer that is TDM, therapeutic drug management, a standard terminology? So, we were arrogant enough to say that no, this is the terminology we coined to say that it's no more monitoring, and we are no more these passive people who would just give you the level and whether it is in range or not in range, whether you should sit tight when this is in range, that any biochemistry lab will also do, right? So, we were arrogant enough to say that no, this is not, this is probably an upcoming term, and we probably need to rephrase the TDM as therapeutic drug management. So, the dictum in TDM has changed. So, we no more treat the blood concentration, we treat the patient. So, therefore, our advice is directed towards the patient being in the center. Like we heard a lot of now the PROMs are the biggest instrument that we use, because that's how we have progressed. And similarly, here the patient is the center, and in my case studies I would make a case why the patient is in the center but not the drug level. The number that we get is not the center around which we are functioning. So, this is a traditional approach. You prescribe a drug, you give the dose, and then you know, most of the you name any drug, it starts with a say that okay, steady state has to be reached before you do a TDM, right? So even if you do it in critical illnesses, the earlier thinking was, oh, if the steady state has not reached, how can I comment on the drug level, or how can I say that whether it is going to be effective or not, or whether I can comment whether it is in range to treat the infection or treat the condition or not. Now, this traditional conventional TDM actually waits for a steady state. It samples the drug at the trough and measures the drug concentration and compares with a target range with a dose adaptation. See, this works fine when you are treating a chronic disease where the effect comes in 4 to 6 weeks and you can afford to wait for, let us say, 3 days or 4 days for the steady state to arrive, or for you to do the drug levels and comment. But it does not work in critical illnesses, or it does not work in situation where there is a fluctuating renal function, where you need to be more precise, because that golden time to treat a patient is gone till you wait for the steady state. So these are certain disadvantages our traditional TDM method had. So therefore, I mean, I would like to delve a little deep into it, because we have a upper and the lower therapeutic window that we are given for all the drugs, the conventional upper and lower. So the upper level says that okay, this is for the toxicity, the lower says okay, you need to achieve this concentration to have some therapeutic or the intended therapeutic benefit. However, this seems to be an oversimplified approach, because this considers that there is a linear relationship between the dose and the levels that you are achieving, and a linear relationship between the dose and the effect that you are reaching. Now, and this needs to wait till steady state, because you can't comment on it otherwise. Now, the timing of the sample collection is not flexible, because you have to wait. And then this does not integrate the pharmacodynamic aspects. We are only looking at the blood concentration and a certain therapeutic effect. Now as we move on, I wish we had the TCI introduction integrated into this presentation, but for the purposes of non-conflicting things, so that the other speaker also gets to gets his share on the TCI, I did not purposefully include it. However, the TCI actually says a target concentration intervention, but I have to mention this, as if I don't mention this, I probably won't be able to move to the MIPD. So TCI actually says that, like I gave you an example of critical illness, and I am giving a drug like vancomycin, where I have to reach a certain concentration to kill the bacteria which is causing the septicemia. In that case, my first dose when I started is very crucial, and my first dose has to predict that I have to reach that level which is optimal, which is the AUC by MIC level for the bacteria itself. So therefore, in that case, my predictability has to be more. My predictability of a concentration that is going to be bringing about the desirable pharmacodynamic effect, it's more demanding, you know, than the normal conventional TDM. So therefore, we need an optimized initial dose. So this optimized initial dose is not achieved by the TDM itself, the normal therapeutic drug monitoring approach. Therefore, my sampling time has to be flexible, and the measurement of drug concentration, obviously we call it as the lead-in time, or the time that is turnaround time for this. So this lead-in time or the turnaround time has to be very small. The Bayesian estimation of the PK and the target dose calculation has to be integrated, which gives you a dose adaptation strategy that helps you achieve the second dose, which is going to achieve the TCI or the target concentration which is needed for this condition. So if I give you this approach, it's a little complicated. Having said that, it is for the postgraduate students to just take away those words and look up, and without this I would not have probably been doing a justice to you. So this uses a non-linear mixed effect approach, and this non-linear mixed effect approach actually has a structural model which outlines the PK, and it has a covariate model, and it also uses a mathematical expression to do this thing. Now, it can be a parametric approach, it can also be a non-parametric approach, and it starts with a population model, and that can be employed before the first dose is administered. Now, the last thing that I would pay attention, request you to pay attention to is the probability of target attainment. So, every time you use a model informed precision dosing instrument, it gives you the probability of target attainment. That means, if you use this dose, what is the probability that you are going to achieve a level which is important for the condition you are treating, right? So, and there is something called Bayesian forecasting. This is what the models are integrated in. So, having said that, despite many years of doing this, we are yet to develop the model for our population. We are using the Western models, which are obviously, we suffer from various criticisms for using these models, and for all practical purposes, we should not be using these models, but should be developing our own models. Things take time, and as we also yesterday discussed during the career avenues, that we are probably not as greatly equipped for doing this modeling thing, and we need the allied specialty to come in to help us out, and probably this is one area where the young pharmacologists can take the lead. So, these are the progresses in the last few decades, and I mentioned before that, before I make this case for TDM, I thought I'll give you this random drug as a concept, which is nothing but what the other labs do and give you a number. So, this is the question for all of us: whose domain is TDM? Probably this I would have given I mean, this would have been a talk for 30 minutes or 40 minutes itself, but it's a food for thought for everyone, that whose domain is it? Are we going to leave it like that for others to take over? Right. So, because in the interest of time, I'll just discuss one case which came yesterday. So, this is a pediatric nephrotic syndrome patient with who came for dose optimization. He is a 3-year-old female child, and hospitalized for frequently relapsing nephrotic syndrome, treated with prednisolone and cyclosporine A, and it is clinically labeled as steroid refractory nephrotic syndrome. So, the child needed periodic prednisolone pulse therapy to get rid of the overcome the periodic episode of relapse, and then the cyclosporine level came as 31.3, which is the current level that we measured in our lab. We have a previous report of 68. So, what did we do? So, we requested them to collect the samples at 0, 1, 2, 4 and 6, because the level was 38 and the recommended level is not very clearly defined in nephrotic syndrome, because cyclosporine is still very off-labelly prescribed in this condition. Now, the child was on 7.5 mg per kg, the prescribed dose is 5. So, the clinician is also apprehensive how to do the, how should we manage this case, because we are getting only 38, maybe 100 to 200 is the therapeutic range, and the child is having no response. Then we checked for the compliance was good. Sometimes you do not function the way it should function, in the sense that you think something and then something else happens. You ask them to do a sampling on 0, 1, 2 and 4 hours to see whether the drug is getting absorbed or not, but the resident is bindas. He collects the sample at 1, 2 and 4. Now what do? Your patient has been pricked thrice. You have to salvage yourself in this situation and give some recommendation which is helpful for this clinician. Then what do we do? So, what we did was, we did a imputation kind of a thing giving 38, which is the previous value available to us, and also used the same technique to do the, I'll take 2-3 minutes more. So, and used also 68 as a value which is available. Both the times we got a level, then we calculated the AUC. Now, we looked at the literature, what is the evidence available on this, and this study, which is an old study, 2005 publication, it says around 100 to 360, 160 to 364 they got the value. And the mean was around 250, right? Now, we went back and did the calculation. So, when we imputed these values, our CSA was coming somewhere around 2665. And if I impute it to 68, it's nearly 3000. So, we thought that we are nearly in range. Then what's the problem? Is CSA, the cyclosporine absorption is adequate? What could be the issue? Then I went back and checked the up-to-date, where they are saying the 80 to 120 is the trough. So, now the big question is, could this be a formulation related issue, which is a reality with all of us. The patient was actually on a formulation which is not the innovative formulation. Now, you will end up in situations like this day in and day out. So, even if we are not able to do the MIPD, we are trying to salvage ourselves as much as being we are trying to be as much useful to the clinician by doing our small bit. So, finally, we decided to sort of change the formulation to the other and use the same dose to see whether our AUC is improving. And the whole idea was to induce remission to this patient and prevent the relapses. So, I have 6-7 more cases to show, but in the interest of time, anybody can take my slides and take a look on that. And these are the real cases that we do every day. And so, I'll skip through them. Or can I do one more case? Can I do one more case? I think it's okay to skip. I'll do some advocacy rather. Okay. So, before I end, I would like to bring it to your attention about this organization. This is International Association of Therapeutic Drug Monitoring and Clinical Toxicology. When I started my career as a faculty about 12 years back, I came across this organization, and then it sort of clicked, and I thought. And at that time, I was below 40 years, so I went and did this for the signing of this as a member, and I found that it's a free membership. So, I took the membership, and then everything is a history. Now I am a counselor of this society, working actively into this, and I am really proud to say that we have highest number of participants from the Indian subcontinent, and then we should not stop there. My urge to all of you who are not members and who are really wishing to have careers in a hospital setting in TDM, or in a setting where you can actually deliver these MIPD based services to the patients, because these days a lot of hospitals other than in the government sector are also interested in delivering the services where they could be a useful value addition to the patients. Now, if you go to this website, you would find the scientific committees. When you go to the scientific committees, you would probably get those areas where the organization works. It has alternative sampling strategy where you explore saliva and other alternative matrices. Then you have TDM of biologics, clinical toxicology and drugs of misuse, immunosuppressant drugs. This is where I work, and I also work in the oncology space. Then you also have toxicology, and there's a corner you would find as young scientist committee, whoever is less than 40 years is eligible for this young scientist committee, and the membership is free for Indian students. And even if you are more than 40, the membership fee is about $20 to $30 for 2 years. So it's really, really reasonable. And we conduct webinars on TDM, which is a bi-monthly affair. And this has been very widely appreciated, and we try to circulate it as much possible. This is a free webinar, whether you are a IATDM city member or not. And we try to post it on LinkedIn and other platforms. And we were doing it since 2012, and now this legacy is being carried over by our younger colleagues from India. And so that's all. Then the second thing that I would like to draw your attention is to the newer program, which is the mentor-mentee program. It's not working. So please join. There is a mentor-mentee program where you can actually. So this is the upcoming webinar. Just mark your calendars for 4th December. You might. There is a good opportunity that this is a very convenient time, 4:30 p.m. in the afternoon. So join this, register for this and join this. And to summarize, I would say that TDM really needs a push in India, and the younger generation and the people who are in the position of command are the ones who will have to take this forward, because making another society would probably not be as useful as then working actively in it. And these are my sincere acknowledgements from the IITDM city and the colleagues at home, my department colleagues, and my institute. And this is the team at PGI Chandigarh, who is actually they are the ones who run the program, all my students. And a very, very sincere thanks to all of them for being a part of this. Thank you, everyone.
Good morning to all. Thank you, madam, for your wonderful lecture. It is the eye-opening for the post-graduation young pharmacologist, because this TDM is one branch which is going to be very important in the future because of the immunosuppressant and the anti-cancer therapy, and the therapies the list is going on. So many drugs, monoclonal antibodies, and the targeted therapy. Definitely it will be a future target for the pharmacologist for the job, as far as the job is oriented. Now the session is open to question. Any questions from the audience side?
Yes, I have a question, ma'am. Thank you for an elaborate presentation on the TDM and non-linear mixed effect modeling. So what is the current status of physiologically based pharmacokinetic pharmacodynamic modeling in TDM in India and in the world as a whole?
Thank you so much for asking that question. I am sure you are an advanced learner. So the PBPK modeling is important, definitely important, but most of the PBPK modelings are, PBPK is mostly looking at the different compartment level, right? So when we say, because the TDM itself recognizes the body as one compartment, central compartment. We give the blood in the, we give the drug, and it goes to the central compartment, and we don't take into consideration all the peripheral compartment, which are the different organs. So the PBPK modeling generally is complex because it takes into account a lot of other compartments in the body. So the primary use of PBPK model is mostly in the drug development space when we are developing new chemical entities. However, the usefulness to TDM as such, or MIPD as such, is very limited, because the concept that Bayesian estimations are superior because it takes into account the a priori probabilities of the organ functions, body weight, the other covariates, as well as the drug dose and the concentration, sort of overpowers the predictability that is there in the PBPK model.
Is there any other question?
Good morning, sir. Thank you, ma'am, for this wonderful talk. This is not actually a question. When the minimum standard requirements of NMC was recently released, they have listed so many labs, and unfortunately the therapeutic drug monitoring lab is nowhere seen, and the role of pharmacologist is not nowhere seen. So as this is a forum for national pharmacologists, and ma'am has told that is one of the areas on which we should work, because I am working in a government medical college in Kerala. We have given the proposal in 2017 and still it is nowhere seen. Unless it is mandated from a higher side, the peripheral centers will find it very difficult to run a program like this.
I can't tell you how much I agree with you. I mean, it's neglected because people think it's expensive. We buy other equipments, but buying an HPLC or other analyzer becomes a secondary thing. You know, long back, the Phil Walshan is one of the founder of the IATDM city. He said the greatest barrier actually comes from the administrators, right? But here what you have pointed out is a very, very fundamental question or fundamental thing which we all should ponder over. If it is the case that the NMC does not think that it is mandatory to or it's not even desirable to have a TDM kind of a facility for a pharmacology, then we must push it forward as a society. That's the way I would look at it. And having said that, we all have struggled in our own ways to make things work in smaller pockets. It has to catch a movement which is widespread.
Thank you, madam. We conclude the session by giving concluding remarks.
Thank you, madam, for a lucid and informative session. As you said, we are rather than passively measuring the levels, we can shift over to the therapeutic drug management, and we can be a bridging. We can bridge the gap between a theory knowledge of our clinical pharmacology knowledge, pharmacokinetics knowledge, to the practical patient-based clinical approach. So maybe in future we can add AI and precision pharmacogenomics into this, so that it'll add more value to
Absolutely. We do a bit of genomics also. One of my cases had that. In the interest of time, I had to skip that.
Yeah, sure.
And you're very right, because AI has actually come in already.
Yeah.
So instead of the Bayesian forecasting, now they are using machine learning based forecasting for the drug levels.
Okay.
Thank
Once
you
again,
so much
thank
for
you,
that
ma'am.
comment.
Thank you, everyone. We'll conclude the session. Thank you, all the organizers.
Thank you, everyone.
Thank you so much, ma'am. May I please request the chairpersons to present a memento to our speaker. Thank you so much, ma'am, for enlightening us on the topic. Can we please have a huge, huge round of applause for our speaker, Dr. Patnaik. Requesting Dr. Renuka Munshi, Professor and Head Clinical Pharmacology, TN Medical College and BYL Nair Hospital, Mumbai, to please come up to the dais to felicitate our chairpersons. Can we please have Dr. Renuka Munshi on the stage? Requesting Madam to please felicitate Dr. Asha Pathak, Dr. S. Muhammad Mustafa and Dr. Shashidhar Barnale. A big thank you to all the three chairpersons. We all can definitely clap. Also for Dr. S. Muhammad Mustafa and Dr. Shashidhar Barnale. Thank you to all the three chairpersons and thank you to Dr. Renuka Munshi as well for doing the honors. The plenary session 3 talks about the single-use medical device sunrise sector for Indian healthcare, and the speaker would be Dr. Varun Gupta, the CEO of MedVital Ventures Private Limited. And to chair the session, may I please invite Dr. Vijay Motkare, former professor and head, Government Medical College, Nagpur. Please welcome, sir, with a huge round of applause. Dr. Julius Anthony, pharmaceutical consultant, Bangalore. Dr. Reshma Quattros, medical advisor, Sun Pharma, Mumbai. We shall have Dr. Vijay Motkare, Dr. Julius Anthony, Dr. Reshma Quattros to chair the session. A warm welcome to the chairpersons, requesting you to kindly take over the proceedings of the session. Thank you.
Good morning. Warm welcome. Thank you, the organizers of NAPTICON, for allowing me to chair this session. It is my privilege to introduce Dr. Varun Gupta, who is the CEO of MedVital Ventures Private Limited. Sir has extensive expertise in creating consumer-focused healthcare solutions and scaling innovations. So as the CEO of MedVital, he leads innovations in wound care, wound closure, and skin regeneration. So Sir has also launched various drugs in emerging markets such as Russia, Kuwait, French West Africa, and Southeast Asia. So previously, he was also the Senior Vice President at Tata 1mg. He's also the Chief Business Officer at Antara Max India with over 15 years of experience. He has many accolades under his name, as you can see. A very warm welcome to you, sir.
Thank you. Thank you very much for the kind introduction. And I think, thank you very much for at the outset, I would want to thank Dr. Kamal, Dr. Ganesh for inviting me for this. So I think I'll start with Dr. Smita's word that as pharmacologists, we find identity crisis, right? So what is next for us? And Dr. Abey's points yesterday that you have to find your own apple that is falling. And then that's the problem you find and you kind of go after that problem. So my journey has been slightly in that way. I mean, I started with Elsevier doing medical education at South Asia market. then was part of the 1mg, that is creating healthcare platform. Now the current looking at is single-use medical device. And the reason we picked up this when Dr. Kamal and Dr. Ganesh was talking, I thought pharmacology conference में medical device की बात करना तो थोड़ा सा it's different. But I thought I see a lot of similarities between pharma of 25 years back, pharma industry in general, and where we are in medical device sector. So I see a lot of similarities. That, for example, in India, 25 years back, we were importing a lot of pharmaceuticals, right? Now we are the pharmacy of the world. Medical device, similarly, 70% is being imported. We can't even produce. I was with a manufacturer yesterday. Do you know micropore? We can't even, at this point, we don't have a facility to produce a micropore. Sutures. All the simple things, we don't have a manufacturing facility. We are dependent on the China, Taiwan, other countries to import those products. And the government policies have been supportive to say that make in India and for the globe. That's why I picked up this as a sector. So the kind of cover in next 25-30 minutes would be the bird's eye view of how the healthcare sector for us as an entrepreneur looks like. What are the important components which we have, last 15 months we have been working on this sector. Some insights and interesting insights. Please feel interrupt, we can make it more interactive. And I will keep a lot of questions for the. So I see this healthcare as this is a pharmacologist from a business lens, right? That's the perspective. If I divide that healthcare, I see this from a perspective, it's a $100 billion market. And these are the components. Hospital services, healthcare services, 30 billion, health insurance, 3 billion, pharmaceuticals, 20, and diagnostic, 5 billion. The important piece was this 10 billion of medical device. What we thought was last 30 years, if you look at this, innovations have happened in pharmaceuticals, diagnostics, lot of new models, lot of acquisitions, mergers, but medical devices at the cusp where innovation is going to happen a lot in next 20 years. So that's one. If you see the CAGR, it is expected to go from 11 billion to 50 billion at a CAGR of 16%. And India is being one of the fastest growing medical device markets. And what we see is that in this, all the long-term growth drivers are very, very strong. Rising incomes, we need quality products at affordable price, insurance penetration is increasing, and we have got access to newer markets, and government policy support is overall there, right? Schemes, if you look at imports of a lot of these regulated are now restricted, and medical device parks are being produced. So we see the fundamentals are very, very strong for this as a sector, and there is an opportunity to innovate in this sector. And the reason I picked up, this is the course slide, the reason I picked up in a pharma conference, I picked up a medical device because I see deja vu. 90s pharma sector, 2024 medical device sector, absolutely on the same trajectory. We were kind of dependent on imports. There was a TRIPS agreement, an unlock that happened at a policy level. We started exploring export markets, started cracking US, Europe markets. Indian pharma sector research started growing. So we see that is absolutely the case for medical device industry. So this is where, if you look at pharma industry, it is now 130 billion, expected to reach 130 billion by 2030. Pharma medical device is still very, very nascent, small, but expected to become a $50 billion. And we see a lot of, it is at this point, 80% market is fragmented, dominated by SMEs, right? Then there's a lot of organized effort would happen. And interestingly, a lot of vibrant ecosystem. I met 3 years back, I met an entrepreneur in Ahmedabad. He said, I said, "Kya kar rahe hain aap?" He was MBBS junior, right? So he did his Stanford innovation program, and he was innovating on some medical device for fracture. मैंने कहा, very nice, where did you innovate? Surat. What is your primary market? US. He has acquired 5% of market in US. Super. That's a very interesting innovator based out of Surat, was able to create a product, get a US FDA certification, exporting the product. Met another entrepreneur who was making implants, and he was challenging all the big MNCs on ortho implants. So what I could see was that in silos, a lot of things are happening. A lot of manufacturers are out there, innovators are out there, distributors are out there. But this is all in silos. But there is an opportunity to integrate them into a single model, right? So that's the vibrant ecosystem we see. And within the medical device, if you look at, 30% of medical device globally as well as domestically is single-use medical device. Single-use means your syringes, your suture materials, your wound dressings, everything which is consumable, classified as consumable. And it is unfortunate, but that is the reality, that at this point, 70 to 80% of consumable we do not make in India, right? That is, and there is a lot of unorganized import that is happening. Unorganized import, when I say, so for example, let's take a surgical glove. All the rejected gloves of Malaysia and Indonesia are pumped in the country in containers. They get sterilized, packed, and kind of go into the market. Similarly, it happens for a lot of other things. So there's an opportunity for us to take a stab at it and say that we can make in India, we can innovate in Indian product, create high quality products at a world standard at an affordable price, right? That's the kind of opportunity out there. And there are, in any sector, there are leaders who have already done it, and then other innovators or other entrepreneurs kind of follow them. So in Indian companies, 5-6 Indian companies have done really well in last 10 years, right, in the medical device space. I'm just giving some examples. Say that Polymed is a market listed company, 1.7 billion, 2 billion kind of market cap. Very, very good on IV cannulas. Healthium, it just got recently acquired by a private equity fund. It was a suture in India, very high quality sutures. So they have been able to challenge MNCs on how to create high quality sutures. Sajanand Medical, I met the senior members, and I was surprised to hear that in 2026, world's 25% cardiac stents be produced by this company. Sajanand Medical would produce 25% world's cardiac stents. What all this indicates is that there is some bright light. Some Indian companies that is less than 5% have reached that level where they're challenging the status quo, creating high quality product for the globe. But it also creates a vacuum to say that multiple people are needed at the bottom to create this, replicate these models in multiple categories. And that's the basic point. Merrill, I mean, I didn't know when I started working on medical device that Merrill is an Indian company. It's a high quality, again, one of the top Indian companies. So to the point, going back to that pharma and medical device comparison, if you look at pharmaceutical industry at this point, there are 60 to 800 companies which have got turnover of more than 500 crores, right? In medical device, there are 6 to 8 companies, right? So that's the difference. The scale, a lot of these companies are subscale, 10 crore, 20 crore, 30 crore top line. Only 6 to 8 companies are above 500 crore. But because of the macro growth factors, we see that next 10 years, there would be, like pharma, there would be 50, 100 companies in that particular category of 500 crore, more than 500 crore top line. And then we started, what we started mapping was that what are the factors where Indian companies have gone more than 500 crores? Where are the companies which have remained subscale? What are the patterns out there? We realized that there are larger categories, cardiac stent, implants, cannula, where there was already a leading multinational company, and Indian innovators were able to produce high quality products at a competitive prices, started disrupting the pricing. So in each of these categories, there were kind of competition, and multiple categories, there is no challenger Indian brand. And one of them was wound care, and if you look at wound care, we looked at this market that 80% of wound care market, the kind of dressings, the kind of bandages, the kind of all these things that we're getting is unorganized, is from the kind of hyper-local market. This is a wound dressing tray of a multi-specialty hospital. If you look at this, not provided by a single brand, multiple brand. You can see for a simple thing like a wound dressing, there are 8 to 10 brands supplying different parts of wound dressing. So that's where we thought it's a good place to be in. And in general, there is either very, very high premium products that are available, or there are low quality products which are available, right? The gap is between finding a high quality product at a competitive price. That's broadly the white space or the gap. Because for lot of Indian population taking a 1,500, 2,000 rupees per day dressing is unaffordable, right? So can we create a high quality product at this point? So we see that there is opportunity to innovate in this space, unorganized market, healthcare spend. I wanted to spend time on this, and this is just for the youngsters to look at a problem and then how do you create new age models. So when we looked at these silos, we said that, look, there are manufacturing capacity that is available. There are innovators that are available. There are distribution networks that are available. Can we create a new age model in this? New age model means that you utilize manufacturing facility capacity that is available by taking a loan license and becoming a manufacturer without owning the asset. So you can have, so for example, at this point we have 5 manufacturing facilities. We don't own the asset. We own the license. And in some time we were able to create those 5 facilities of different products. So that's the manufacturing facility. But the challenge was what should be produced and how do you measure the quality? That is the value addition we wanted to make in this manufacturing facility. The thinking process of manufacturing and the output. Can we use new AI tools to measure the quality? Can you use AI tools to measure do the visual inspection, right? All those things. That was the first part. Second part was that fulfillment network. So the R&D in a new age model for us was to find the micro needs of doctors and nurses. That smaller needs which are not addressed by the global players, can we kind of find a commercial model that it works. And second part which we did was that there are innovators which are available who have done a good significant work for last 8-10 years. Can we partner with them, become their go-to market partner, in license with them, take the technology from them, in license, build a royalty model, and take that model so it becomes a common team for multiple innovators. So there is an efficiency of sales, marketing, and fulfillment networks. So that is an in-licensing model. Shop-in-shop manufacturing, and of course, focusing on demand generation. So this is what we set for ourselves. This is just to say that when you start something new, this is second, third time I'm starting something from scratch. Very, very important, somebody said in the, is that define the purpose statement for yourself. Define the purpose statement for yourself, define that purpose statement for the organization, and keep hammering yourself and saying, this is the purpose, this is where we exist. And if you feel, there are ups and downs which comes up in the journey, right? Purpose statement helps you to navigate those ups and downs. I think that was the fundamental point when we started. We said, let's first write down our purpose statement and define who am I, why by me, right? Purpose, and this is what we did for ourselves, is to identify the micro needs of doctors and nurses, innovate smart products to empower them to deliver better care at affordable price. And focus area for us is to innovate smart products in wound care, wound closure, and scar management. And we kind of launched few products. Interesting example, 2 examples I would want to take. When we started meeting a lot of players, market, we realized that we passed MBBS MD before 2000, right? And we realized that a lot of technologies have changed. There is something called as negative pressure wound therapy, which is which has become the mainstream in wound therapy. It creates a vacuum, device creates a vacuum, and that vacuum is put for a closed system for 5 days, and the healing is equivalent to, it accelerates the healing, and there's a lot of scientific evidence to say that this healing is much, much better than traditional wound dressing. But it is at this point is limited to only super specialists, plastic surgeons. A 5-day treatment costs around 20,000 rupees, and a patient has to take 3 5 days treatment. So for a patient, it is 50K to 60K. We said, wow, there is a treatment which is effective, globally acceptable, but at this point, not accessible and affordable for Indian population, right? Opportunity for us to reverse engineer, find that how can we make it affordable and accessible, right? And also add a lot of, second thing we realized is patient घर पे चला जाता है, patient और doctor का interaction break हो जाता है. So we said, can we use WhatsApp business to kind of help them communicate that this is the wound? Can we create a normal remote monitoring call center to help them? So we started building all the digital ecosystem around that. So this was the first example. Second example, and these are all innovators which were there in the market, and we were helping them to commercialize this. This is an innovator based out of, he's a French scientist. 15, 20 years he has been working on biopolymers, and within the biopolymers he has identified chitosan as one of the biopolymers which can help wound healing. So he collaborated, the source raw material of chitosan comes from crustacean shell. Now to produce 1 kg of high quality chitosan, you require 1 ton of food waste. Now how do you get 1 ton of seafood waste to produce a high quality? So then there was ecosystem enablement, I would say. A seafood exporter collaborated with this innovator, and we being the commercial partner, we were able to produce this high quality chitosan and stabilize this biopolymer into liquid form so that it can go deep into the wounds, right? So all I'm saying is, what we are trying to do is just a thought process for people to think is that do not reinvent the wheel. Don't reinvent the wheel. There is a value to also create a connected ecosystem so that the end user gets the benefit of the innovation. So that's, and finding which innovation can be commercialized or can be scaled, because multiple innovations are ahead of the time, or sometimes they are me too. So finding those right innovations which have also got a commercial opportunity out there, and then creating that ecosystem. That's, I'll just take a pause at this point. So we are calling ourselves as, and again, multiple journeys, finding a new name. When we started our 1mg journey, I remember that we were in office, pre-funding, pre-everything, finding a new name. So somebody said, यार कुछ नाम अच्छा सा ढूंढते हैं. तो ऑफिस में बैठे हुए किसी ने नाम बोला कुछ कि अपना ऑफिस का एड्रेस जो है वो 1mg रोड गुड़गांव है. तो don't we give 1mg as a name? So 1mg was our office address. 1 Mahatma Gandhi Road, Gurgaon. And that is how 1mg became kind of conceived, and it got a different go. So similarly, we were finding a name, and it was, we called as No Wound. So thank you very much. Open for questions.
Thank you for a very insightful presentation. Before I hand it over to the audience, I want to ask you a question which I think is very fundamental to everybody in this room. Single-use devices, as the term states, should not be used more than once. Okay, but across the world, even in the developed world, single-use devices are being used 30 to 50%, even in US, even in Western Europe. Okay. Now the thing is, linked to that, there are multiplicity of issues, and all of us face that problem. I'm sure you would face that problem as well. There are issues on regulation, there are issues on ethics, there's medical issues, okay? There are legal issues, there are environmental issues, okay? Now, I discuss everything or state your points if you can, but at least tell us the legal issues. Because in many hospitals across, I have worked initially, okay, as a physician, and in hospitals, reuse of the single-use devices have been happening. The legal implications are going up. Just want your perspective on it. There may not be an answer, but
Yeah, I think it's a problem, as you rightly said. My perspective would be that it has to be, there are some fundamental user needs that are not being addressed by the companies. And that is the starting point of people are not at peace. Let's say doctor, nurses who are at the helm of things, they're not at peace. They say that, look, this particular, let's say for example, I do remember of this suture removal or a stapler pin removal device is 10,000 rupees, is not worth using it for multiple use. So fundamentally there are times when they are kind of for a business model or a price that pushes them to use it multiple times. That's the first fundamental thing. So as economy grows, as it becomes more affordable, then people say it is not worth it. It is the price versus of reuse versus a single use makes it the risk they take or the legal regulatory policy risk they take is not, patient safety risk they take is not worth the price. So first solving for that price / quality imbalance, right? Because doctors or nurses or administrator at the helm of it, they know it, that this is may or may not be the right thing. That's the first piece. And somehow they have that piece that medical device industry or pharma industry in general is profiteering, is not profiting, they're not giving at the right price. So solving for that is the first piece. Second is, of course, I don't believe that top-down regulation blockage would stop a lot of these things, right? Because people find loopholes and ways to solve for that. There are, of course, policy level changes which government is working on. A lot of these policy level changes because medical device industry in general got organized. They started putting this together and this is evidence together that this is why particular things which are happening at mass scale for a particular thing, and they started solving for the 80% of the problems. So I think my approach would have been, my perspective would be that make it so simple for people not to do that in accessibility, affordability, and quality. When I was I remember an example, I was at Elsevier and we had this global conference. One of our MD said, go to Google. It was easy to download a plagiarized book versus buying a normal copyrighted product, because friction, as Elsevier, the friction we had created so many frictions and touch points that it was easier to do download a plagiarized book. So I think that was.
Thank you. Thank you for a very well. Just a follow-up question, which is probably even more tougher. Okay, because I was also working in industry, and I know that. Do you think that there is a need for single-use devices in countries like ours, rather than having a single-use device which needs to be reprocessed and reused n number of times, which is directed by a manufacturer itself? Obviously, it is far more complex for the manufacturer, but it helps society. Just your perspective.
Absolutely, absolutely. There are absolutely clear areas where industry push is to make it single use to make the economics work. And a lot of these times, so for example, in our case, I'll just give our case example. In negative pressure wound therapy, the dressings are single use and canister is a single use. See, a simple canister costs you 3,000, 2,000 rupees. For a same patient, if the treatment is for 15 days, why can't be the design of cleaning the canister or sterilizing the canister be so simple that it can be reused? You don't need single-use canister for collection of the exudate or the pus, right? So those components, definitely there is opportunity for industry to do that, I think.
Thank you, thank you. I leave it up to the audience. Yes.
I think so this is another forum where, you know, pharmacologists are not very much trained, but we are supposed to be jack of all trades, but a master of none. So I also feel the same thing. So when we actually in KM have a lot of medical device studies, and we as a pharmacologists are very untrained in knowing the challenges of this trial. And as we approve, like, and when we try to find out expertise in this field, and there is a lot of deficiency in the expertise as well as in the field. And people who want to venture out in medical device industry as well, pharmacologists also have a lot of challenges because they are not trained enough in their curriculum. That is another deficit in our curriculum. We don't train our pharmacologists in medical device, how medical device actually works in our this thing. So when there was a wall study in KM, we actually could not make out how we should approve this study, because if you see the preclinical setting from where these trials come in, I don't know how much of the preclinical setting these devices are undergoing through of single term device, but I'm talking about intricate device where mortality is a big issue. And we did not get into preclinical trials at all. There are 2 pig studies, and in 1 pig dies, it is 50% mortality, and you have to take a decision that first time patient, first time trial in and that comes to an institute like us, and then we are very much in a dilemma to get approval of such studies. So what my question is, who are the experts? Like when I went into this expertise, to ask questions to the experts, I found all the pharmacy people are experts in medical device. I could not find a medical doctor to convince me, like, how should I approve this device in our setting. And that was a big challenge for me, a pharmacist convincing me. He was talking a perspective of medical device, but he could not convince me how it actually works in a human structure. So for me, the challenge for you I'm asking is, who is actually an expert who actually advises on medical device? Because again, regulators are also that gray, like as we are.
Yeah, and I think, ma'am, you have raised a very, very valid point. Last few years, we all knew that devices were part of Drug and Cosmetic Act, right? And then this change happened when Medical Device Act and rules are separate from Drug and Cosmetic Act. Now the same regulators, everybody in the pharma value chain are also expected to have the competency of medical devices, right? That's across the value chain. That is, I speak to a lot of regulators. They also say that we find this challenging. We come from a pharma background, and there's a lot of background on that, but is medical device a similar structure like a pharma? Absolutely not, right? So this is in general, this is the transition phase as an industry, as a country we are going through. This is a new sector for us. Now to your specific question, I think in pockets, a lot of good work has been done by AIIMS Stanford Biodesign Program. That's a collaboration between AIIMS Stanford and IIT Delhi to develop this expertise in medical device. For last 8-10 years, they have been doing a lot of innovation around that. So that's one area of center of excellence out there. Then from the similar, there is a London Institute of Healthcare Engineering, which is with one of the largest hospitals. They have done absolutes. Dr. Prashant heads that healthcare engineering. He was at AIIMS and he went there. A medical doctor specializing into medical device. And they have an institute, given West some ahead of curve, they have created an entire institute on healthcare engineering. So that's another area of excellence that is happening. So there are such pockets of excellence where you would get these expertise. And there, I mean, in my journey, I met a lot of surgeons who are now the medical device manufacturer. Because what they realized was कि जो वो stapler use कर रहे हैं, उसमें कुछ challenges आ रहे हैं, वो MNC को बताते हैं, वो solve नहीं हो रहा क्योंकि product life cycle बहुत है, तो उन्होंने कहा मैं innovation करके, मैं mold बना के अपनी manufacturing facility में खुद ही अपने लिए बना लेता हूँ, and then I can also distribute and sell it to others. So there are such pockets of excellence across, and within ICMR also there is a division on medical device. So the reason I also picked up medical devices in this forum is, as you said, all of us are expected to be also at some level to be at least appreciate that what are commonalities between medical device and pharma, where they differ, because there is no expertise and people would come to us for such guidance / consultations.
Sir, is there any committee which approves this?
Yeah. So drug,
it is just like I am on that drug approval committee. On
absolutely.
DCTs, yeah. It is a committee like that.
Absolutely same structure. There is in the Medical Device Act and rules. There are the committees under the DCGI there are committees on the on the medical device, and it has to go through the same process. Just what they've done well in this Medical Device Act and rules is they define the levels of medical device based on their risk assessment, A, B, C, and D. A being the non-sterile, non-invasive, B being the sterile, non-invasive, C being the sterile, invasive, and D being the highest level of being invasive. So regulations and the registration and the committee approvals are different for all of these 4 categories.
Secondly, there is a control over the price? It is some drugs are controlled. The price control is there?
Yes. So medical device like implants and stents under the same national NPPA, National Pharmaceutical Pricing Agency, there is a body to look at the pricing of medical device. So
there is fixed the price, because if you go from hospital to hospital and other place, the prices they varies, like stent and all these, the dressing material also.
So now few of these medical device have come under price control. So of course there is always a business model to find a way to price higher, but in general they're under price control and they're expected to show you transparently that the stents pricing cannot be beyond this price control price, ceiling price. Yeah, please.
Good afternoon, sir. Thank you for the session. I don't have a question. I want to ask for your advice. The new generation doctor who want to be a future entrepreneur, if I want to start a business at this hour with medical device in India, as you said, the market is huge because we are not manufacturing anything at this time. We are asking other countries to send it to our country. So if today as a fresher with no knowledge about business, as I think medicos lack that, so what should I do? Like what first 2 steps I should start to start a medical device business and get into the market as a MD pharmacologist?
Just start. Don't think.
You can start.
You can start. Just start. I mean, I like that Dr. Abey's point that find a problem, at least find an unmet need which you are passionate about. You will meet 10 people, you will interview 10 doctors, 10 nurses, 10 patients, whatever, X number of patients, find unmet need. And unmet need should kind of also drive your passion that this is the problem I would want to solve for. And then rest all is start and keep doing. I mean, find that problem, then you will get support from the ecosystem in terms of how do you make a business case, how do you start small, and you will find people to support you in your journey. And another practical way to do it would be that be part of some startups ecosystem while you are solving for your problem, to just see that what are the basics, hygiene of fundraise, making a company, hiring the team, all those fundamentals and basics are in to be placed. But all these don't make it too complex. वो मतलब इतना complex है नहीं, वो journey में अपने आप solve होता रहता है, मतलब just so that's basics are there in place, so it is okay to start. Family and friends से छोटा सा round create करिए, एक problem identify करिए, वहाँ से आपने शुरू करिए, product बनाया और sales and marketing करनी शुरू की, rest all you will keep evolving.
Thank you so much.
You can start your business right from just now.
Yeah.
Before passing your MD, and once you pass it, you can start business.
And sir, that's the difference I see. I get to engage with a lot of engineers, MBAs across my journey, right? So I interviewed a person who was 22-year-old. I said him, this is my 3rd job. I failed in 2 startups. When did you start your startup? First startup, 2nd year. It scaled and it didn't work, but he learned the basics. 2nd startup, he learned the basics. 3rd, he said that I will want to work with you guys, and then I'm here, we call them as short shelf life hirings, right? They come for 12 to 14 months. They know that they have to jump back, right? So it is okay. I mean, it's a good kind of, at least there's a clarity of thought on the both sides. Yeah.
Thank you, sir. Thank you so much.
Thank you, sir, for your wonderful presentation. It was a new topic for us. Thank you. Thank you once again.
Thank you.
Thank you.
Ma'am, no, ma'am, please. Yeah.
So, Dr. Varun, thank you so much for that. A lot of new things I learned. I was wondering, once we had a discussion with a cardiologist, they, forever are dissatisfied with the quality of the Indian stents that they use for their patients. And unfortunately, like you prescribe generic, so it's by default choice in an institute. So any comments on that, the quality issue is something which probably prevents the clinician to use these products?
Very, very valid point. I think there are two parts to it. I think one is, of course, over a period of time, in general, as an industry, as a country, we have to increase our bar for quality, right? Setting up the quality processes, setting up those infrastructures in place to check the quality. But over the last 5-10 years, I've seen that evolution. Second part which I see is somehow there is some inherent bias sometimes. They do not start with, Indian companies do not start with when the neutral mindset. They kind of, that's also a bias. So I'm not saying it's a one linear problem. So there are times products are very, very high quality. But there is a bias that because it's an Indian brand, Indian product, so the quality might be poor. But I think still at the stage where we are in medical device in general, for example, pharma, the quality standards are gone up, right? Still, US FDA would say that India में वो quality के products नहीं बन रहे, pharma के भी, pharma के quality के products नहीं बन रहे हैं, but pockets में excellence के regions भी हैं, तो कहीं ना कहीं, and we have seen that example कि एक ही manufacturing facility में there are facilities for export and there are facilities for domestic market. Because the price control is out there.
And then, and the products that roll out from an export section and those for the in-country section, there would be a difference.
Yes. So, I mean, just to summarize, what I see is the competency-wise, we don't have a competency challenge of making high-quality products. I met a manufacturer in Gurgaon who was the contract manufacturer for the largest 4 global companies. For IV cannulas and for all the central venous cannulas. I asked him, do you apply in domestic market? Because the quality which I produce, at the price which I produce, we can't afford this. So I do not do a single product for India, right? So this means that the entire manufacturing facility or the entire competency of engineers, production managers, they are built is of highest quality. But something other than the quality, there's something is not working for them. So I think it's a complex problem.
I know. It's pretty intangible also. I mean, we can only perceive it, and the solution towards it is a complex path.
Yeah.
Thank you.
Thank you.
Thank you so much, sir. Dr. Varun Gupta, may I please request the chairpersons to felicitate sir with a memento. And can we have a huge, huge round of applause for Dr. Gupta. The doctor entrepreneur for all of us, showing us the new ways. Thank you so much, sir, for being here with us and guiding us through. Thank you so much. And to felicitate our chairpersons, to present a memento to our chairpersons each, may I please call upon Dr. Saroj Kothari, Professor, Sudha Medical College, Kota, Rajasthan. Felicitating our chairpersons, Dr. Vijay Motkare, former professor and head, Government Medical College, Nagpur. Dr. Julius Anthony, pharmaceutical consultant from Bengaluru, and Dr. Reshma Quadros, medical advisor at Sun Pharma, Mumbai. Thank you, chairpersons, and thank you, Dr. Saroj Kothari, for doing the honors. Thank you. Moving ahead, it is time for a deliberation on AI in drug development and clinical research, boon or bane. And to moderate the session, may I please call upon Dr. Ashish Kakkar, who is an associate professor at the PGIMER, Chandigarh. Sir has an experience of more than 15 years in teaching and clinical research, is also the member secretary of Institute Ethics Committee, PGIMER, the associate editor and editorial board member for various peer-reviewed indexed journals, also the principal investigator, extramural funded studies from Department of Science and Technology, Indian Council of Medical Research, and other funding agencies. We welcome you, sir, and requesting you to kindly take over the proceedings of this session. The session would last for a total of 40 minutes. Thank you.
Thank you. So at the outset, I'd like to thank the organizers, especially Professor Ganesh Dakle, for choosing to hold this important panel discussion at the National Forum of NAPTICON, and to focus not just on the opportunities, but also the challenges associated with the use of AI for clinical research. So we are actually continuing from yesterday's panel discussion, where we discussed the various career avenues which are available for pharmacologists, and even following up from the previous session by Dr. Varun. So so this one is also right up that alley. So when we are looking at career avenues available for budding and young medical pharmacologists. So going on further, I, on behalf of the entire panel, would like to thank the organizers for giving us the opportunity to engage in this interesting discussion, and we sincerely hope that you would find it to be worthwhile. So without any further ado, let me invite the panelists to the stage. So first up is Professor Sneha Ambani. She is the professor and head of department of pharmacology at AIIMS Jodhpur. She has an experience of 30 years in teaching and clinical research. She has received the Best Paper Award by Indian Pharmacological Society in the year 2000. She is current chair of the Animal Ethics Committee of AIIMS Jodhpur and also the Institutional Ethics Committee of IIT Jodhpur. She has more than 90 research publications to her credit in national and international journals. Ma'am, may I invite you to the panel here? Thank you. So next up is Dr. Santosh. He's a senior clinical research scientist for Cure AI Bangalore. He has an experience of 14 years patient care and clinical research, and an extensive experience with the conduct of regulatory clinical trials. He has clinical research expertise with the use of AI tools in healthcare. He has won several awards at conferences, including the one with Indian Society of Clinical Research, and he has research publications in both national as well as international journals. So may I invite Dr. Santosh to the panel here. And finally, Dr. Sharik. He is an assistant professor and deputy medical superintendent. He's from Department of Pharmacology, JNMC, Aligarh. He is an experienced teacher, has 14 years of experience in both teaching and clinical research, has more than 30 research publications to his credit, and he has managed various intramural and extramural projects. He has filed 2 patents and has copyright of 2 apps related to pharmacovigilance. He's also a member of the Clinical Research Committee of CCRUM, and additionally works as the Deputy Coordinator of the Clinical Trial Unit at AMU. So thank you for accepting our invitation, Dr. Sharik, and may I invite you to the panel. So this is when I'll hand over the mics to our panelists and give them an opportunity to just let us know what interests them about AI and how they are connected to this topic, i.e. AI, and what are their experiences and their thoughts on the role of AI in clinical research as well as drug development. So can we have a mic, ma'am? I think we have a mic here. Yeah, I'll join you, ma'am, there. Yes.
Thank you, Dr. Ashish. And at the outset, I thank you, the organizers, to give me the opportunity to conduct this panel discussion. As we said now, currently this AI is, of course, definitely play a very important key role in extending from drug development to diagnostic and monitoring and all that. But as all the techniques, all the new era have certain advantages and disadvantages. So same thing happened with the AI. So that's why we are here to discuss whether this AI is using in clinical trial and drug development, whether it is a boon or ban.
Thank you. At the outset, I would like to thank the organizers for inviting me. It's a great privilege and an opportunity to be here. For me, how am I related to AI? Like my everyday work involves, working. I work at an AI health tech company. So for me, the way I see it is, whatever work which we are doing, what is the patient impact it's creating? Either in terms of like, irrespective of what the work is. So for me, I think the work which I do, which mainly concerns health tech AI, and which is kind of helping democratize access to AI tools across the globe. So I think that's where my personal interest lies in. And also, of course, as ma'am mentioned, rightly we have the challenges as well. So I think the time has come for us to kind of embrace the challenges. It's not just embracing the one technology, or it's about the whole gamut of challenges, both its positives and the negatives, and work through it. So that's where I say it. Thank you. Over to you.
Thank you, Dr. Ashish, and the organizers of NAPTICON. For me, AI is something which a lazy person, can have. And to make the life of a lazy person easy, that's what AI is to me. I was introduced to AI as an automation of my work, some of the work in clinical research. I also organized an online workshop which has more than 170 participants, and this workshop was on AI tools for research writing. And that gave me a lot of insights about what the students want and what is the future of AI. And I was more of inclined to an idea that AI is here to stay, and AI will be used as an augmentation of our work. It will not replace us, it will be used as an augmentation of this work. And this is the idea, this is the main idea on which I will be discussing more of my topics.
Yeah, thank you, Dr. Sharik. So, and to your point, a lazy person's life is anywhere easy. Yeah. So the way we have planned this session is that we are initially going to have two questions, a couple of questions directed at each of our panelists to set the stage for today's discussion. And later on, we are going to open the floor for audience participation. And we encourage you to come up with your comments, your queries, and to raise your questions at that time. So we'll just kickstart the discussion right now. So we are having a couple of questions for each of our panelists. So our first question is that coming from artificial intelligence, to Dr. Sharik's point, what he has just discussed. So there have been success stories with artificial intelligence, not too far ago, in COVID-19. So one of the well-known platforms is the Benevolent AI that actually repurposed baricitinib, if you would know, for management of COVID-19, which was later on approved for emergency use. And also there are reports where AI could find successful treatments, means potentially useful treatments for Ebola within a day. So given this background, do you see AI as a revolutionary tool which is capable of replacing discovery and development paradigm, or is it better viewed as a aid which complements the current process? So that would be our very first question, and as Dr. Sharik has just alluded to that, we would direct this question to Dr. Sharik.
So I think the answer to this question is more complex. It's not just a black and white answer. It's not just a yes and no answer, whether it is a boon or bane. It lies somewhere in between. I think we are at a movement which is a calculator movement for it may be. What happened with the calculators initially, my grandfather used to say that don't work with the calculator, don't do your maths with the calculator, because it will degrade your mind. And what happened, the calculator stayed. And I used calculator. And he was right that it degraded my mind that I can't do 17 into 19 in a jiffy, like he was able to do it. But what it did was it freed the mind. It freed the mind for competition of other things. What other things? That we have now, with the help of these computations, we have now sent the Chandrayaan to the far side of the moon. That is a very big feat of computation. That was only possible if we use these computation skills. And similarly with the AI, there are two aspects of AI. One is a hype, that AI can solve all the challenges, that it can generate all kinds of drugs. It may have generated drugs for COVID-19, but it is not possible that it can generate drugs for each and everything. If the data is not there, the AI actually works on data. The better the data, the better will be its generation. If there is no data how to generate a drug, it will not be able to generate that molecule for you. You have to work for it. So that is a hype for it. But on the contrary, there is also the people who underplay AI. Of course, AI is going to revolutionize the idea of how we do clinical research, how we do clinical or preclinical research. It is already doing it. So when I was in MD, there was a software called as Autodoc Vina for the first years who have just joined pharmacology. It's a docking software. You can see it as a virtual lock and key mechanism software where you dock your substrate with your ligands, and you can see whether it fits or not. So while doing that, I was very, very intrigued by that idea that the things that we have to do on rats or in vitro, we can do it virtually, which was very interesting. But what happened after that, I found out that there was advancement, automation. So there was automation of those software by several companies, and then now with the AI revolution and the GPTs available, we have machine learning tools which can actually do, for example, like iDoc software or NovaDoc software. These softwares are available, and they can actually do, you don't have to do, any single thing without for generating the softwares, generating the good molecules for that. So I think the AI is here to stay. We have to look at the challenges. There is a lot of challenges with the AI, but I think one challenge of AI is worth mentioning here, and that is the black box of AI. So what happens? So let's understand what is the black box of AI. So what GPT is that you give a prompt, and then it will prosecute, it will execute some commands, and then it will give us an outcome. For example, if you give some symptoms, it will give you that the symptom might be of this disease. Or if you give 5 molecules, it will tell you that this molecule, the one molecule or the B molecule is the best molecule for going forward. The black box is how it comes to this decision. That is the opaque part. And we have to understand that this black box is black box is the problematic part of the AI, that we do not know how it is going to affect the judgments and whether the judgment call that the AI is making is correct or not. So that is the main issue with the AI. Apart from the hype and apart from all these undermining things, this is the main issue that we should be focusing on to make it more transparent, the AI. I think that is...
So would you like to add?
Yeah, so I was just wondering, like Dr. Sharik mentioned many points about the challenges. So when it comes to AI in healthcare, it's a very, very exhaust topic, very broad topic. So I think for the sake of like that is going to impact going forward in terms of health care. So when there humans will computer vision and so where guys use it's not about replacement. When we talk about patient care, there is a first you have to kind of look at the prompt to a certain direction, then there is further, then at last in the whole gamut of this process points we will have to evaluate of AI in how it can supplement. So it's not about AI is going to play a role from end to end. It's the at which critical juncture AI can really, because at many times there are subjectivity involved in how we deliver care and also how we deliver how we conduct research. So AI to an extent, because it is algorithm based, it can reduce subjectivity to an extent. So that's where I see it.
So in true sense, it should be a complementary, not the replacement of all the existence system. That's
so
it should be complementary.
So we use the term augmented intelligence.
Yeah.
So that is better, where we have human intelligence and that is integrated with machine-derived outputs so as to improve healthcare. So that is the I think the take-home point for the discussion that we have right now. And of course, there are certain areas where AI is still not very capable, particularly, for example, understanding the complex biology of such as diseases like COVID-19 and also certain contextual insights. For example, the baricitinib example that I gave. So it was found to be useful, but then there could have been a possibility that baricitinib could interfere with the immunological responses, making patients more vulnerable to secondary infections. So therefore, taking those kind of decisions will always require human inputs.
So it cannot replace the human trials.
Yeah, so
definitely.
You need not worry, the postgraduates. Yeah, okay, so in the interest of time, we'll move to our second question here. So many of the AI tools in clinical research, as we understand, they are based on data derived mainly from the West and the Caucasian populations, right? So for example, there is an AI tool from IBM Watson for oncology. So some of the outputs that this tool gave was about drugs which were not approved in other countries, or it was giving out outputs based on a different set of staging processes and diagnostic algorithms which were not suited for maybe the lower middle income countries. So my next question is, how can we ensure that these AI systems which are used in clinical research, they are more inclusive and they are more reliable for our own patient populations? And I would like to direct this question to Professor Sneha Ambani.
So it's more related to avoid the bias while selecting all these, because in LMIC, we have limitations in our patients and techniques. As Dr. Sai said, that we have to collect the data for the proper implementation of the or use of AI. So while there are bias while you are collecting the data, so just not to follow the data which are given by the Western country, we have to generate our own data. And while collecting those data, we should not be biased in getting the demographic, as we discussed in the morning also. Here in the Western countries, they have fixed set of population to collect the data. Here it is not. So same thing here, we should collect the data unbiasedly from the demographic, which I think address the same problem which are going. So before that, I must say that we should generate the, don't bias with the in generation of problem. So you first collect data related with the problem, then collect the data correctly without or unbiased by taking this demographically data as compared to in relation with the race, age, vulnerable population and all that. So we have to collect the data accordingly. Second point is, while this collecting data, assessing these data, we should take care of all these missing and outliers that should be there. And you collect the data from the population which relate with that particular problem, not the generalized population. So that we should address that particular population which you can relate with that particular problem. Another thing is during analysis as well. Another thing is while doing analysis, and as I mentioned, it should not be the replacement. It should be complementary. So human loop must be taken in while doing all these research. So just to avoid this bias and while you're collecting these data.
So bias is a pertinent thing. I think it's not only restricted to LMICs, it's across the globe. Even when you talk about an FDA approved, AI algorithm, even the FDA looks at it in terms of what is the bias. Like they are very particular about certain introduction of bias, especially with regard to the data from which ethnicity the data was collected. So the one way to kind of overcome is to have different data sets. That is the training, testing, and validation data set, different data sets, and also prior to the deployment and prior to the clinical validation studies, the data pertaining to that study, usually the algorithms will also be fine-tuned, or a calibration exercise would be carried out. So, and another thing, just I wanted to make a quick point here is, I think lack of data from LMS is a challenge. I agree, but at the same time, it's also an opportunities for many of the diseases. If you can, gather data from those countries and use AI, it can truly democratize the access to the care. So that's a potential there, which I truly see. Yeah.
When you said it should follow the algorithm, so there are three algorithms whether we will collect the data, whether it is supervised, semi-supervised, or unsupervised. So it is better we collect all these data supervised to avoid all the bias and all that. So to get the good data for a particular problem.
So the summary is that we need to have our own data sets, because our models can only be as good as the data they are trained on. So we do need good quality data, but at the same time we also need to contextualize these tools to our own settings.
I think I want to add one more point to that. I think the data of pharmacogenomics is very sparse. And if the FDA is saying that you have to prescribe the drugs based on pharmacogenomics of the patient, then it becomes really difficult for the AI to decide whether we should give the drug or not based on the sparse data. Sparse data is a problem for AI. The training data set is very small, and we cannot put this data, we cannot impose this data on small data on the whole population. That is the main issue with the sparse data. And what ma'am is saying is very correct, that it will definitely have some bias on the trials, even in the real world scenario.
Okay, I think we'll move further, and I'd like to invite Dr. Santosh to share his thoughts on the areas where AI is being currently used and what are the challenges being faced in those areas.
Yeah. So just as you mentioned at the outset, AI is already here. It's not something of the future. Like AI is being used as we speak and it's being deployed. So one of the challenges with regard to AI is not only about research and also adoption and implementation. That's one of the challenges as well. So in terms of real world examples, if you can refer to the previous talk, wonderful talk by the way by Dr. Varun Gupta. So a lot of things are happening in the AI medical device segment. So in fact, our health tech company Q.AI, works on this medical images. So even within the medical images, there are different kinds of support which the AI can bring in. One is it can triage, like which are of the lesions or the images with some lesion, without lesion. That is known as the CAD-T. And then we have the CAD-E, where it can mark a lesion, detect a circle around an area of interest. That is known as CAD-E. And it, in some cases, it can not only mark, it can also say what is the lesion. Say for example, QXR, that Q.AI has automated solutions for X-ray, CT, and CT brain. So in the X-ray, it can mark a lesion and say, is it a nodule, or is it a opacity, is it a, is there a pneumothorax, is there a fibrohilar enlargement. So these are the aspects of medical image based AI inputs, where it can be a CAD-T or CAD-E or CAD-X device. And these are already in place. In fact, if you look at the FDA website, there are more than maybe 500 AI-based software as a medical device, and also which have gone up recently in the last 2 to 3 years. And with use case, particularly if you see, you mentioned about LMSs. Now, automated interpretation of chest X-ray is being used worldwide. In fact, WHO has also endorsed this in screening for TB. It's being, in fact, QXR is deployed in more than 80 countries. And if you take the another use case, detection of a nodule, a suspicious nodule with a certain feature on an X-ray, any X-ray taken for any chest X-ray taken for any reason, it can flag a nodule, lung nodule, and which can diagnose and detect lung cancer early. Here, the AI is not saying it's a lung cancer, it's prompting the patient to the right pathway. Say, for example, lung cancer is very aggressive, we know, stage 4, and majority of, in fact, in India, more than 90% of the lung cancers are diagnosed in an advanced stage, where the treatment is, again, the options are less and also prognosis is poor. But if an incidental X-ray taken for any reason, if AI can prompt a nodule, and if the patient is directed to the appropriate pathway for that CT and then the diagnosis, and early stage cancers have better prognosis. And another use case is stroke, where the AI is able to read an image, CT image, and immediately prompt and the patient for a therapeutic intervention, be it be operative interventional intervention like mechanical thrombectomy, where the golden time matters a lot. So these are some of the use cases which AI is already being used. In fact, there are many other like clinical prediction models and multimodal data, risk prediction models, and also personalized medicine and precision medicine. So these are, and in addition to, these are the real world use cases. If we talk about the challenges, again, there are challenges with regard to the deployment. If you want to deploy at a particular site, maybe a remote site, what are the certain infrastructures to be available beforehand? So those are certain challenges we will have to go through.
So I agree with this. So another popular area is diabetic retinopathy.
Yes.
Scanning the retinal scans, looking for, especially in the periphery when we don't have specialists available, and approximately 20 to 30% patients with diabetes are going to have some form of retinopathy.
It's in fact used widely in the UK NHS for diabetic retinopathy.
So one thing we realize is that majority of them are rightly are focused on diagnostics. So is it like therapeutics, particularly with some from India? Do we have many examples, or for that matter, for other
in terms of oncology,
well?
not exactly. I can give you an example from maybe US based, where they are looking at, for a cancer patients, there would be multiple options in terms of regimes. Say, for example, colorectal carcinoma, patient with colorectal carcinoma, you have the FOLFOX regime, FOLFIRI regime, different regimes are there. So which one to choose? So AI with the prediction, algorithm based on particular patient characteristics can give you the exact, in terms of probability of which one would suit better. Multimodal, points, different data points into consideration. Those are some of the areas where AI is being also used as a therapeutic, therapeutic support as well.
So I'll move on further since we are running short of time somewhat. So I'll direct this to again Dr. Sharik, who has previously discussed that the way to go forward is having a collaborative approach between both humans as well as the AI. So we understand they help us with speed, efficiency, and even cost effectiveness, but then again, they lack the nuance understanding which is needed for clinical judgment. So what do you foresee is the ideal partnership between AI and humans in clinical decision making? So would you like to comment, Dr. Sharik?
So before that, we have to understand what AI can do and what AI cannot do. So the AI can have lot of data. It can analyze, can read a lot of data, a big data file. It can read, then it can analyze the data, and it can produce some answers for you based on that data file. Now, what do it? It will not give you answers simply just asking, "Okay, give me a drug for AIDS." It will not give that answer. What will give? It will show you a pathway. Pathway with which you will be going to have on that particular pathway, and then you can get a drug on AIDS. but it will only give the answers based on the data which is there. Secondly, and which is very, very important, is that in a dynamic clinical environment where the doctor has to make decisions on the point, there are two things which are lacking here. One is the situational awareness, and second is the empathy of the doctor. These both things are actually lacking, and therefore the right decision making for the patient might not be that easy for the AI. Therefore, human interface will have to be there. The human intelligence, the human experience, they have to be there. The AI will augment them. The AI will help them in achieving the right decision, but the decision has to be taken by a human being, by a human doctor. If we see, if what we are talking about the data, let me give you an example that it's not that, see, people were saying that the AI will make a very huge impact on various jobs, profile, and various fields. It may have a very high, impact on different fields. But I believe that the impact of AI in the field of drug research will be limited. Why it will be limited? Because we were doing this, doing a part of AI, we are doing part of automation. We were doing it for decades. So it's not rational drug designing. So it's a part of AI-based automation. So what rational drug designing has achieved, it is the same data that will be treated as a test data for the AI to produce something. Now, there lies the problem. The problem is that AI will give you drugs like zanamivir, or it will give you drugs like the pamivir drug. So these drugs basically are the template of the previous drugs like neuroaminidase inhibitors like oseltamivir. So if the template is there, then it will create that drug for you. But the problem is that if the template is not there, then it will not create the drug for you. You have to make that template. Once you have made the template, then you can ask the AI to give a drug molecule or research pathway. That is the only way for the AI. That is the data that you want to give to the AI. So in all, AI will have an impact on clinical research and decision making in clinical scenarios, but the impact will be limited.
Right. So we know AI is actually doing well as well as target identification, as well as the screening of compounds is concerned. But of course, one way to address the trust deficit that we may have in a healthcare professional is to have something which is called as XAI, explainable AI, to what Dr. Santosh refers, that the AI can highlight scans based on which the decisions have been taken. So they can create heat maps based on what how the AI classified that. For example, those chest X-rays into tumor, or CT scans into tumors, or the retinal or the fundus scans into the type of diabetic retinopathy. So my next question would be for Professor Sneha Ambani. So we already discussed that we need both good quality data as well as large volumes of data to have better models for AI. So at the same time, this creates concerns regarding privacy because we are now dealing with large amount of data. So how do we achieve a balance between the need and requirements for need of good quality and quantity of data versus privacy concerns, especially in resource limited settings like ours?
As with other clinical trials, autonomy lies always with patients. So we should ensure the autonomy of the patient even while using AI. So while, as I already mentioned, we should take this human in loop while collecting these results and analyzing the analysis. So when we are talking about when we are going to use these data, and that is the major risk. Dr. Said that we should have a good template as well as good data to compare or to get the answer. If you don't have any good data, and if somehow the other validation of those data doesn't occur, and you have some or some dilemma related with patient's care, better you go for the clinician response rather than going to the or rely on the AI. So that is the, I am talking about the risk which is related with. So you have to maintain all these things while assessing or while generating these data. So as I mentioned, autonomy is always lying with that. And if patient wants that I should not go or I should not means go for these treatment or diagnosis given through AI, and I will go for the clinician concern, that autonomy lies with them. So that is there. So privacy as well as autonomy is always lies with the patient, even while using AI. As you know, all these are technical based, so privacy is at the risk, as you know, as the technology advanced, everything is on the clouds. So we should have encrypted data, so that only the person who is authorized or have copyright can access those data. And ideally, they should be, while you are collecting the data, they should be anonymous. So that is how you can save this privacy of the patient is there. Again, same thing, this encrypted, high techniques which which generate the technical issues, technical needs that are there while using data, they should be ensured. And same ethical consideration and all these, that is always there with this. So privacy should be maintained while using these data.
So yeah, so I agree with your points, and there are some techniques like federated learning also available, so which can also help us ensuring that. But in the interest of time, we'll move forward to our last question. That since we are continuing for yesterday's panel discussion on career avenues, so I would like to pose this question to Dr. Santosh: that what competency and skills do pharmacologists, especially the ones who are currently in training, they need to develop to thrive in this rapidly evolving field? I think all the MD residents are paying close attention to this answer.
Yeah. So before that, I just wanted to add to whatever ma'am said. But when we talk about AI research, we should always have two things in mind. That is the patient interest and the innovation. If we drop off innovation altogether, then there is a missed opportunity as well. So patient interest on one hand and innovation on the other, we should try to balance either at the ethics committee level, regulatory level, even as an individual investigators, researchers. So that's a key thing going forward. So in terms of skills, I would say that clinical, sound foundation in clinical research design and clinical research methodology is a valuable asset even with AI, because we are going to see a lot of clinical performance assessment of all the AI, whatever AI be discovered or the generated. So a lot of clinical assessment, how are you going to conduct the study, what are the study designs, and a strong foundation in clinical research design, not only RCTs, across the various study designs, that's a must, I would say, and a strong foundation in biostatistics. So we spoke about different concerns like explainability, transparency, and all those things. When we can put that exact error, this is the measurement error, this is the confidence interval, and we have the strong, a strong foundation in biostatistics would help. And programming, I would not say a must, but valuable asset to have. Strong, basic knowledge of AI, ML models and algorithms. And to an extent, I would also add, since all our MD pharmacologists, clinical practice and clinical experience is also a valuable asset. Yeah.
Right. So I think we have an AI bell here who's not paying attention to the gravity of the question. Yeah. So I think we can open up the floor. I think we can have 5 minutes. Professor Ganesh Dakle sir, can we have 5 minutes? 3 minutes, okay. So we can have 1 or 2 pressing questions here.
So I was looking up the FDA site and we have nearly 1,000 devices or AI-enabled devices approved or tools approved. And to my surprise that there were many, many recalls of these. So Dr. Santosh, you were, very confident about the validation procedures. So may I ask you why they are so frequent compared to drugs?
If we look at how the medical devices are, the regulation and the drugs, there is something called 501K submission, and also there is de novo submission. Many of them are based on the predicated devices. So in terms of recalls, again, it depends on the risk assessment. It is not exactly in what patient care pathway that is being used. So like I mentioned, if it is in the different categories of triage or assessment, and in terms of clinical procedures, when you talk about the validation methods, FDA has got 501K summaries which clearly detail, which is detailed enough to provide the evidence with regard to how the device or the drug was approved. And when it comes to continued performance or algorithm change plan, the landscape is still evolving. If there is an algorithm change plan and it has to be documented, and if there is a change in the decision or the way the device behaves, so there will be certain consequences of that. The manufacturer might recall that and may replace it with another improved or updated algorithm.
I agree with you. It depends on the gravity of the situation or the risk that most of these AI-enabled tools are radiology-based tools.
Yeah.
And probably we trust our clinical acumen more than the AI which has given us the diagnosis.
So, and there also it is different paradigms which are approved. Like it is concurrent paradigm, like where AI is also reading and the clinician is also reading. Or it is second reader paradigm, where clinician reads and AI reads and clinician can see what AI has read. He can agree or disagree. And and there is automated read that is rarely approved or this thing. So most of it now is second reader paradigm. The ultimate responsibility is with the clinician, and they have the liberty or the choice to either accept or reject. So thereby the patient safety is ensured.
So the third level comes with the molecular diagnostic, which is again you put a needle and see. So you really don't trust the AI much for doing everything for you.
So in terms of, you can definitely take the advice, because the main thing is you don't want to miss a critical diagnosis. So even if there is one case which is missed, and if you're able to detect it, and that's a value addition.
So I think we can have one more question.
This is for Dr. Santosh. What is the role of AI in personalized medicine or precision medicine as it is known nowadays? Is there any role or any existing things are going on there?
I don't have personal experience with that. I have not worked in that field. But just to summarize whatever I mentioned earlier, in many of the cancer treatments, the multimodal mode of approach is being explored a lot, where they take into consideration a lot of genomic data, not only at the level of key mutations, but even additional mutations as well, and adopt other feed into the data other risk factors and the patient factors, and see how the patient would respond. So that is one area where it is being keenly explored, the precision medicine.
Thank you.
Yeah, and especially also in clinical research. So we have AI currently matching patients to the available trials. So like the basket and the umbrella trials where we have based on disease heterogeneity. So there also AI is playing key roles in matching the patients to the appropriate trials which are being held. Yeah. Okay, I think, thank you. I thank the organizers and I thank the entire panel for this engaging discussion. Thank you.
Thank you.
Thank you so much. That was indeed a wonderful brainstorming session, requesting the moderator and the panelists to stay back for the felicitation. And to do the honors, may I call upon Dr. Deepak Kumar Langre, Professor and Head, D.Y. Patil University School of Medicine, Navi Mumbai. Requesting sir to please felicitate our moderator as well as the panelists for this wonderful brainstorming session. Thank you so much, sir. Thank you so much, Dr. Ashish Kakkar, Dr. Sneha Ambani, Dr. Santosh, and Dr. Syed Naeem. And thank you so much, Dr. Deepak Kumar Langde, for doing the honors. The next one would be one of the most awaited parts of the NAPTICON, that is the NAPTI quiz. We shall have the stage set up in the next few moments, requesting all of you to please be seated for this really, really exciting quiz, that is the NAPTI quiz. Console, please help us with the laptop setup here. Somebody from the console, please. इसमें आपका इसमें कुछ नहीं है ना सर अभी ये वाले लैपटॉप में some really, really serious arrangements for the NAPTI quiz. Seems more serious even than the Bonvita quiz contest and the Kaun Banega Crorepati on the TV. It is really going to be something very exciting and unique. The NAPTI quiz has always been one of the most interesting parts of the NAPTICON, and we're looking forward to the NAPTI quiz 2024 at the NAPTICON 2024.
स्लाइड चेंजर भी लगेगा उनको देना पड़ेगा मेरे वाले में नहीं सर्विस सर को दे दिया इसको हटा के नीचे रख लो शायद तब तक ये वाले को
जजेस को आप कुछ दे रहे हैं शीट वगैरह लिखने के लिए स्कोरर कौन है सर अपना
स्कोरर है तो पारुल
कहाँ बैठेंगे पारुल जी
साइड में बैठेंगे
ओके ठीक है और सर जजेस को कोई शीट वगैरह देनी है हमने
ये पार्टिसिपेंट को कहाँ देते हैं वहाँ पे क्वेश्चन
सर आपके लिए 2 बोतल
हेलो हेलो
सर आपका लैपटॉप नहीं आया नहीं आप इधर ले लीजिए लैपटॉप ठीक है अभी आपके लिए
हेलो 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 डन हेलो हेलो
वो नहीं है क्या
check
हेलो हेलो ready for the NAPTIC quiz please raise your hands I would like to make request those who are sitting behind they can come forward please join us because we are having exciting prizes you will be able comfortably to participate please come forward those who are sitting behind please occupy the front chairs for past 1 month we are putting very hard efforts and this is my guarantee you will enjoy this session and it is going to be different in what sense you will gain a lot of knowledge there will be excitement there will be winning of the prizes with the participants and we are considering audience also a team okay ready thank you all and this is our guarantee that in between there will be lighter moments also you will really enjoy along with the gaining excitement knowledge we know it pharmacology is a very little bit boring subject but we have made it very interesting in between you will keep on enjoying we are having commercial breaks also in between okay so thank you all for joining us for this NAPTIC quiz I really thanks to our anchor, Dr. Richa, for putting such a hard work since yesterday. I am making request again that those who are sitting behind, they please come forward if they can join, because we are having prizes and it will be difficult for us to shift the prizes from here to the back row. Okay.
Thank you so much to the very dynamic Dr. C.M. Kamal. And the quiz is again going to be bemisal, because Dr. Kamal will be our prime quiz master for the day. And reading out the CV of Dr. C.M. Kamal, though everybody knows him and everybody knows about his achievements, but as the protocol goes. Currently, the Professor of Pharmacology at the SMMH Government Medical College, Saharanpur, founder and national coordinator and founder of National Association of Pharmacology and Therapeutics. He is the UG and PG Assessor, National Medical Commission, Delhi, certified GCP trainer, certified trainer in medical education, and certified UG and PG teacher. Professional experience of more than 15 years in pharmacology and 18 years of in pediatrics. Sir has worked in CRO as clinical research physician, former director and founder, Department of Clinical Research, Sharda University Hospital, Greater Noida. Represented the country at International Pediatrics Pharmacology Symposium organized by the IUPHAR. Please, please, please put a huge round of applause to Dr. C.M. Kamal. And we have a special garment, a special gown for Dr. C.M. Kamal. And to do the honors for Dr. C.M. Kamal, I would request Dr. R.K. Dixit to please come up to the stage. There's a special outfit designed for the quiz masters of this NAPTI quiz. आपने कभी भी किसी भी कॉन्फ्रेंस के इतिहास में ऐसा नहीं देखा होगा, ना ही भविष्य में देखेंगे, जो आज यहाँ पर NAPTICON 2024 में एम्स नागपुर की इस धरा पर होने जा रहा है. It's going to be very, very interesting and exciting for all of us. And we all can clap. And here comes the hat for the quiz master. Here comes the hat. तालियां बजती रहें. We're also delighted to have Dr. Sarvesh Singh, Professor, Department of Pharmacology, KGMU, Lucknow, as our second quiz master. And
thank you very much, Dr. Richa. Thank you. We are just doing this tam jam to make it an interesting event so that you may not get bored. And believe me, you will keep on enjoying also. It very, very stressful event. You will gain a lot of knowledge also. You will really getting the prizes also. But there will be a lot of fun in between. Now please help me and let me introduce my other quiz master.
please put a huge round of applause.
Dr. Rahul Kumar.
Can we please change the mic for sir? किसी अच्छी चीज के लिए थोड़ा करना ही पड़ता है और सब्र का फल मीठा होता है.
Thank you very much. Thank you, Dr. Sarvesh. Thank you, Dr. Rahul Kumar. They both have hard efforts behind us on making this event successful. Unfortunately, our other quiz master was Pritpal Singh Matija, but somebody from his family got admitted in ICU. He could not make it today. So Rahul Kumar have happily replaced him. I would like to ask you, please give a big round of applause to both of my colleagues, Rahul Kumar, Dr. Sarvesh Singh. Thank you. So we are going to start the quiz round. Before I can start, let me ask the team that have been selected for this purpose. I would like to request Dr. Sarvesh Singh, please announce the team and please ask them to join the stage. And before I can go ahead, let me introduce the judges for today. Dr. Ritu Shatak. Madam, please, if you can come forward. Yes. Hira Bhalla. Dr. Ritu Shatak from Himachal Pradesh. Dr. Hira Bhalla from All India Institute of Medical Sciences, Gorakhpur, will be the judges for this event. Thank you, sir. Thank you for accepting our request to be a judge for this event. And let me request Dr. Sarvesh Singh to please ask the team to the stage. Dr. Sarvesh Singh.
All the team.
Please explain them how we have organized the prelims rounds also.
All the teams are requested to sit, come together. All the teams are requested to take. This is the for team A. Team A. Introduce himself.
Team A, GMC Kannauj. Please give a big round of applause.
Myself, Dr. Akashdeep Vishwakarma, junior resident from GMC Kannauj.
Myself, Dr. Ritesh Kumari, JR3, and I'm from GMC Kannauj.
Team A, Kannauj.
Now request to team B to take the seat. Introduction.
Myself, Dr. Kishore from SET Medical College and KM Hospital, Mumbai. Good afternoon all. I'm Dr. Avilash from SET Medical College and KM Mumbai.
Team C. Team C. Yes. Introduce yourself.
Myself, Dr. Pranav Dike, final year resident, MGIMS Sevagram.
Good afternoon, everyone. I am Dr. Gautam, JR1 from MGIMS Sevagram.
Then team D. Yes. Complete. Introduction.
Good afternoon, everyone. I am Dr. Renu Thakur, PGJR Academic, AIMS Raipur.
Good afternoon, everyone. Myself, Dr. Nikunj Agrawal, PGJR Academic, AIMS Raipur.
Have a seat, please. Yesterday we conducted a MCQ test, 50 MCQ test, and the 4 highest scorer are selected. There are 17 teams. 17 टीम थी जिनमें से हम लोगों ने हाईएस्ट 4 को लिया, बाकी एलिमिनेट हो गए हैं. And welcome. Now we have 4 rounds. We have 4 rounds, and sir will explain.
Let me introduce the rules and regulations of the NAPTIC quiz 2024, the 3rd national grand quiz of by National Association of Pharmacology and Therapeutics.
Hello.
Welcome to the 3rd National Grand Quiz of National Association of Pharmacology and Therapeutics, exploring the frontiers in pharmacology. These are the quiz final rules. We are having the round 1, round 2, round 3 and round 4. First round will be the MCQ round. There is one correct option for that. Audio below. Audio. Second round, audio visual round. Video image will be shown. Question related to that. Third round, explain why, rational and justification. In fourth round, there will be rapid fire. 4 times of round for the final 4 teams. Questions to the audience in each round of the quiz. Rules for each round will drift at the beginning of each round. One team with the lowest score will be eliminated after the first 3 rounds, and in the last round, top 3 teams will participate or compete in rapid fire round.
Then first round is very easy, just MCQ round, and one question will carry 10 marks for correct answer, and for wrong answer 0 marks. If team is not able to answer, then question goes to audience. And we consider audience also 5th teams. So every time one question from your side, and at every round, and we have some gift for you. So who will raise hand your first? They have opportunity to answer. So first question for round 1 for team A.
Let me explain the, ask Dr. Sarvesh Singh to explain the rules for this round.
Welcome NAPTICON 2024 by the 3rd National Conference of National Association of Pharmacology and Therapeutics. There are 5 teams, A, B, C, D, and audience. 2 questions per team. Each question has 4 options, out of which only 1 is correct. Each correct answer carries 10 marks, no negative points, and if the team does not answer, the question goes to the audience. Here you can see first question from team A, then question 2, team B, question 3, team C, and so on. And this is very easy round, and all the team can score 20 marks, maximum 20 marks.
So please note that there is no negative marking, and the person who is going to give the right answer will get plus 10 marks, and there is no negative marking.
Time for each
Timer will appear in the screen, on the screen. 30 seconds. Rules and regulations for the audience are always manipulative. We would like that the maximum number of the audience should take the prizes. Let me show the special cap that we have designed. Can somebody show me the cap?
एक cap दिखा दो.
For audience, this is my cap and this is audience cap. Along with this sweets also. मुंह मीठा कराया जाए. Thank you.
Then first question for team A. Only one candidate will answer. Your right side partner. You will answer.
So
you will answer.
Team A is the Kannauj team.
You will answer.
Team B
and here you will answer.
Is the KEM team from Mumbai.
You can discuss.
Team C is
answer you have to give.
Sevagram team, and team D is the Raipur, AIMS Raipur team.
And
so we are going to start the round.
1 minute. Dr. Parul from Kannauj, she is my scorer. She will score the marks.
Welcome, Dr. Parul. Thank you for being accepting request on such a short notice to be agreed for this. Please keep an honesty in the numbering process. Should I start, sir?
Yes, sir. Yes, sir.
So here we are starting the NAPTIC quiz 2024, and we are going to start with the questions, and the questions will be appearing in your screen.
NAPTICON 2024, the third national grand quiz of NPT, exploring frontiers in modern pharmacology. Question is on your screen.
This is for Team A. Team A.
No prompting, please. 8 seconds remaining. आप पहले भी जवाब दे सकते हैं.
सर 80 मियोसिस.
Meiosis, wrong answer. Wrong answer. Actually, scopolamine crosses the blood-brain barrier and may produce sedation. Then drowsiness is the correct answer. Okay, sorry.
Hello.
Next question, sir, next question.
Sir, we are having the question again separately. So don't worry, in between you will be giving the opportunity to answer. And thank you very much, and please give a big round of applause to the spirit of pharmacology and therapeutics. It should be louder in the sense that it should be heard in each and every part of the country, even louder than the clinical subjects. And say proud to be a pharmacologist. Say proud to be a pharmacologist. Thank you.
Second question, sir. Second question for team B. You can see here on this screen only. 30 seconds. Welcome NAPTICON 2024, the third national grand quiz of National Association of Pharmacology and Therapeutics. And question is on your screen.
No prompting, please.
Option A, enalapril.
Correct answer.
Please give a big round of applause. Thank you. Your clappings will increase the morale of the participants. And if you agree with us that we have put some efforts, then please keep on clapping. Because we have made a lot of hard work. You can probably, it is quite evident from the way. And let me promise that it is going to be altogether a different experience, from including the clinical branches also. Okay. Thank you.
For team C. Welcome to NAPTICON 2024, the third national grand quiz of National Association of Pharmacology and Therapeutics. Question is on screen.
Option A, naloxone is a competitive antagonist.
Right answer.
Congratulations. You are rocking all.
There are so many questions, sir, सब balance हो जाएगा. Sometime easy and sometime hard. Next question for team D. बोल सकते हो बेटा, 10 सेकेंड ओनली, 8 सेकेंड.
Answer is C, largely hydrophobic.
Correct answer. Next question for audience. Next question for audience.
Audience are getting impatient. Rahul, Sarvesh sir, please note.
See, first you have to raise hand.
So I'm the audience coordinator for the quiz. You have to raise your hand and I come to you. Okay, so I can see that sir has raised his hand first. Sir.
Audience, please.
Yeah.
Raise your hand.
I think we found.
Yes, sir. One. Here's one. अभी सर रुक जाइए, बस आप खड़े हो जाइए सर, खड़े हो जाइए. Anyone else? Yes, second. Tushar. Yes.
Decrease heart rate.
Pardon?
Decrease heart.
Decrease blood pressure.
Reduce heart rate. B.
Yes, correct answer.
Congratulations, sir.
One cap and one chocolate for him. अगल बगल वालों को भी दीजिए, डॉक्टर विपुल बैठे हुए हैं. Now next question again for team A. Team A, 30 seconds only. You have to answer within 30 seconds. Welcome to NAPTICON 2024, the third national grand quiz of NPT, exploring frontiers in modern pharmacology. Question is on screen now.
Sir, D, niacin.
Niacin? Wrong answer. Sir, please stand up. Gem fibrozil. Wrong answer.
Cholestyramine.
Yes, sir. Ma'am and sir, please give. नहीं मैम ने भी बोला था सर. One topi.
Thank you for answering.
Fat soluble vitamins are reduced, vitamin K, and so bleeding occurs. Okay.
Next question, sir.
Next question for team B. Welcome to NAPTICON 2024, the third national grand quiz of NPT, exploring frontiers in modern pharmacology. Question is on your screen now. Don't prompt, please.
Option D, clozapine.
Right answer. Now third question from team C. You can score 20 marks. This is the easiest. सबसे आसान है. ये आपको जीतने में हेल्प कर सकता है. Team C. Question for team C. Welcome NAPTICON 2024, the third national grand quiz of NPT. Question is on your screen now.
C, inhibiting the acetylcholinesterase enzyme in the brain.
Right answer, right answer.
Please give a big round of applause. Thank you.
Next question for team D. Welcome to NAPTICON 2024, the third national grand quiz of NPT. Question is on your screen now.
B. Flucytosine.
Yes, correct answer. Next question for audience.
Okay.
Please give a big round of applause to the spirit of the audience. It should be loud and loud. Thank you. So, at the end of the round one, we are.
Sir, one question for audience.
ठीक है, ठीक है, ठीक है.
आसान है, काफी आसान है. सबसे पहले कौन बताएगा? आसान है?
Okay, I can see one hand has been raised.
Dr. Tushar?
D. Ipratropium.
Yes, yes, correct answer.
Here's your cap, sir.
And chocolate also.
Congratulations.
मुंह मीठा कराइए.
Then, sir, score please.
So, at the end of round one, we are having, thank you Parul for helping us. Team A from Kannauj have scored no marks, 0 marks. And Team B from KEA Mumbai have scored 20 marks. Team C from Wardha Sevagram have scored 20 marks. And Team D from All India Institute Raipur have scored 20 marks. So with this round at the end of the round one, Team B, C and D are running equally and Team A is running behind. Maybe they will catch up with the passage of time. You all, please give again a big round of the applause to the successful completion of round one. Thank you.
Round 2 will be the image round. One image will be shown. Dr. Kamal will conduct round 2. These are the rules of round 2.
These are the rules of the round 2 that are appearing on your screen. सर कौन बनेगा करोड़पति की फीलिंग आ रही कि नहीं आ रही है? So these are the audio visual rounds rule. A scientist name will be given. Then in the second question adverse effect and you need to choose the drug and you need to name the scientist. There will be options. You will be naming the scientist. Again there are the 2 questions from each team and there is no negative marking. Plus 10 for putting the right answer, and if the team not able to answer, it will get a 0 marks. Then question will pass to the next team, but next team will not be getting full marks. They will be getting half marks. Okay, and we will be giving the slips. Okay, you have all got slips. You need to write answer on the slips. Okay, so all teams will be in action. It is not that we are going to ask the team, they are only thinking. All are noting down the answers.
Equal opportunity for all team.
Equal opportunity for all the team. So question 1 will be picture, question 2 will be ADR, and team who has its turn has to answer, other will get the paper slip. Wrong answer, I have already explained. So if no team answer, then the questions goes to the audience, our 5th team. Thank you.
इससे पहले सर आप लोगों के लिए एक ब्रेक भी है. टीम 1 के बाद एक ब्रेक भी है. So one break is little break, 2-minute break.
Sound, please. Sound.
Abracadabra.
ट्विंस ऐसे ही मजेदार ट्विस्ट है अनमोल ट्विंस में। बटर क्रैकर में चीनी और मसाले का नया ट्विस्ट।
अनमोल ट्विंस, टेस्ट में नया ट्विस्ट।
Thank you very much.
Then question यहाँ पे team B से start होगा. First question for team B.
So first question is going to team B, and the question is appearing on the screen.
Time up.
Time up. Pass. Sleep.
No.
Your answer?
Yes.
So Vincent
Name, no
Van
answer.
Gogh. Vincent Van Gogh.
Wrong answer?
No.
Wrong answer.
Answer for
William Withering.
Yeah, right answer. William Withering. Plus 5 marks.
for team C. For team C.
So they have not given the answer. They have given the answer. Second questions.
Next question for team C. Next question for team C. Welcome to NAPTICON 2024, the third national grand quiz of NPT, exploring frontiers in modern pharmacology.
Question is for team C. Team C.
बाकी लोग स्लिप पे लिख लो. बाकी लोग स्लिप पे लिख लो.
Other team can round answer on this slip.
B2UU.
Please repeat.
B2UU.
B2U. Right answer. You're rocking. Give a big round of applause. Great. Third question is from team D. Team D, Raipur. All India Institute of Medical Sciences, Raipur. Question is appearing on the screen now.
बाकी लोग स्लिप पे लिख ले. बाकी लोग स्लिप पे लिख ले.
Can you name the scientist?
Have you written?
Okay, I am giving the clue. First is the David Carlson, second is the Paul Ehrlich, C is the Gerald Dogmack, D is the Alexander Fleming. No?
Gerald Domick.
No, it's a wrong answer. The
Audience.
right answer.
Sir, audience. Audience. और लास्ट में
David Carlson is the right answer.
ट्रू है
मुंह मीठा कराइए. मुंह मीठा कराइए और कैप दीजिए. जबरदस्त आपकी नॉलेज. Thank you.
So this is the response for the other teams. James Parkins. Okay.
Next question for our next team,
No,
that
not
is
for the
audience.
teams, all are wrong.
Audience, be ready. हैंड रेंज करना है, जो सबसे पहले रेज करेगा उसको मौका दिया जाएगा.
Are you enjoying? Give a big round of applause to the spirit of the pharmacology.
Welcome to NAPTICON 2024. Question is on screen. खड़े हो जाएं अपनी सीट से बल्कि.
Audience, please.
Audience, अपनी सीट पे खड़े हो जाएं. Yes. First. Yes. Stand up. Second. अभी पूछो मत, अभी रुक जाओ. सेकंड भी आ जाने दो. अभी रुक जाओ. आंसर मत बताना अभी.
Hello, please wait for a minute.
Yes, time up. Okay. Yes.
No. I'm giving the options: Jean Stern, Alexander Fleming, Frederick Banting, Paul Ehrlich.
Frederick.
ऐसे नहीं चलेगा सर. ए भी बता रहे हैं, बी भी बता रहे हैं.
Answer C, Frederick.
वही एक कैप दे दो सर को नहीं मानेंगे. Okay, this is the right answer.
Please give the cap.
Give the cap and मुंह मीठा कराया जाए.
मुंह भी मीठा कराइए डेयरी मिल्क से.
सर आप मुंह बंद नहीं करेंगे जब तक मीठा नहीं मिलेगा.
Now next question for team A. Yes, ready? Team A, ready? Welcome to NAPTICON 2024, the third national grand quiz of NPT, exploring frontiers in modern pharmacology. Don't prompt, please. Don't prompt.
No prompting, please.
आपकी बारी आएगी. यह टीम ए के लिए था. Please collect the slip. Please collect the slip.
This is for
No, wrong. I am giving the options and for audience again.
Danny Bowett.
Yes, sir.
We
Sir,
have
stand up,
somebody
please, sir.
in
Stand up,
audience
sir.
to answer.
Yeah,
Sir, one, two.
Bowett. Very good.
Sir, we have
Dr. Rajan Nerurkar, sir, please. Daniel Bowett is the correct answer.
Please give
He
the.
discovered antihistaminics in 1937, and his work focused on drugs that block histamine receptors and are widely used in the treatment of the allergic disorder. Congratulations, sir. Thank you.
Next question for team B. Yes, ready?
Dr. Parul, are you noting down the... Okay, thank you.
Welcome NAPTICON 2024, the third national grand quiz of NPT, exploring frontiers in modern pharmacology.
So your question is appearing on the screen now. is really very interesting questions.
Don't prompt.
No prompting, please.
Please बात ना करें.
Which team? Sir, कौन सी team से? Team B. B.
Team B.
Would like to answer? No, unfortunately, this is the wrong answer.
Collect the slip.
Yes.
Yes, other teams had
Collect the
chance.
slip.
Option A, pyroxicam. It is the wrong answer.
Give me. Penicillin and pyroxicam. Penicillin and pyroxicam.
Wrong answer. Pyroxicam is the... Audience, please. Dr. Kapil. Azithromycin is the right answer. मुंह मीठा कराइए. आपका नंबर भी आएगा सर. And your participation, your interest is reflecting that our efforts are successful of making this event in an interesting way probably. And those who are agreeing, please give a big round of applause. Do you think it's different? It's different or it is the same? I can guarantee you from the clinical branches also, nobody have ever made such efforts in making quiz an interesting event. Okay? And we can challenge, and from this dice, with the kind permission of our President Sir, if he agrees that our President Sir can announce the prize of 10,000 if any institution is organizing the quiz like this. Are you permitting us, sir? So, sir will declare this also. Please declare, sir. So, his blessings is always on our heads.
Two announcement, very important announcement. One is this 10,000 prize, and the second is that we have to be more, family with more members. So the one special drive for 7 days, and that is presidential drive for the membership, and that is a special drive, and that will be having a discount of 1,000 rupees if anybody joins within these 7 days. So please make the most useful of this particular drive for the making number of more members. Thank you.
Thank you, Dixit sir, for your kind blessings always. And let me explain again. Those who are joining the life membership or student membership of National Association of Pharmacology and Therapeutics, please take the advantage of the presidential discount, 1,000 rupees. So you need to give only 1,500 rupees for joining National Association of Pharmacology and Therapeutics, and the offer will remain open for the 7 days only, but you need to have a registration number of the NAPTICON. Okay? So those who are willing to become the member, please raise the hand. Thank you very much. Next question, please.
Next question for Team C.
Team C.
Welcome.
Ready? Your question is appearing on the screen now. Third National Grand Quiz of National Association of Pharmacology and Therapeutics.
Don't prompt. Write on slip. Other teams write on slip. Dr. Rahul, collect. Time up होने वाला है.
C. Sulfasalazine.
Unfortunately, this is the wrong answer.
मैडम ने उठाया था.
नहीं, sir, here.
Team B, right answer, Amiodaron.
Amiodaron also of this team.
Team D, right answer, Amiodaron.
ये आंसर है.
right answer for two teams.
Team B and Team D. Team B and Team D, right answer. है ना? You have to give +5.
Team B and Team D.
Dr.
Will gain
Parul,
5
please
extra
note
marks.
it down.
You have to give +5. है ना?
Next question, please. 8th question, probably.
This is for Team D. Next question is for Team D.
Team D. Next questions. Third national quiz of National Association of Pharmacology and Therapeutics. Exploring frontiers in modern pharmacology. Your question is appearing now.
Answer is B, ACE inhibitors. Answer is B, ACE inhibitors.
ACE inhibitor is the right answer. Congratulations. Next question, please.
Next question for audience.
Audience. Welcome to the third National Grand Quiz of National Association of Pharmacology and Therapeutics, exploring frontiers in modern pharmacology. Your question is appearing now.
Okay, I identified somebody who raised their hand.
Yes, please stand.
बोल दो. Doxycycline.
No, ऐसे नहीं बताना है. जब हम आए आपके पास तभी बताना है. ऐसे कंसीडर नहीं करेंगे हम. इन्होंने प्रॉम्प्ट किया या आपको हाथ उठाया? ठीक है. What is your answer, sir? Doxycycline is the right answer. मुंह मीठा कराइए. कैप दीजिए. Thank you. Sir, can you take his introduction also, please, in between? सर रैगिंग तो बैन है ना, मगर इंट्रोडक्शन तो देने दीजिए कम से कम.
Ashok Dubey from AMC Himachal.
Yeah, big round of applause for the right answer, Dr. Ashok Dubey. Thank you.
Now question for Team A again.
10th question, probably, for Team A. Your question is appearing on the screen now. अब कुछ मुश्किल क्वेश्चन आ रहा है, अभी भी आसानी आ रही है.
Please submit your slip if you have right. Within 30 seconds you have to submit. Yes, answer. B. Answer B, sir. Answer B.
Which team?
Team A.
Team A. What is the answer? B. B, unfortunately, is the wrong answer.
So this is D. The answer is A and D.
D, इनका आंसर ठीक है. इनका आंसर ठीक है. And
no, sir, B only. B.
आपका आंसर?
Sir, this one. Thalidomide A.
थैलिडोमाइड थोड़ा ही बताना, ये बताना, इनमें से ऑप्शन बताना था ना उसमें से. थैलिडोमाइड तो सबको पता है. क्वेश्चन फिर से दिखा दीजिए, लास्ट पार्ट क्वेश्चन का. बच्चे की फोटो लगा के थैलिडोमाइड का नाम थोड़ी पूछेंगे आपसे.
Sir, option is A लिखा हुआ है.
A?
Yes, sir.
A, unfortunately, is the wrong answer.
Audience. From audience. There are so many hands.
Which answer? So Team B has given the right answer. The answer is the risk evaluation and mitigation strategy. Team B, rocking. Thank you.
Parul, Team B, 5 marks. Other 0. Yes, so it is the end of round 2 and a small break. Just a small break.
पहले ब्रेक लेना चाहेंगे कि रिजल्ट बता दूं अभी तक का? हैं? हैं?
कितने लोग चाहते हैं रिजल्ट पहले बताया जाए? कितने लोग चाहते हैं ब्रेक पहले लिया जाए? सबको रिजल्ट चाहिए सर।
So at the end of the round 2, team A is 0, team B 30, team C 35, 35. So with this, team C and team D are leading with 35 and 35. Give a break. Sound please. Sound please.
तुम यहां नए आए हो। मेरी चाय इस तरह नहीं आएगी। ये चम्मच सीधे हाथ पर उल्टा करके रखेंगे, समझ गए?
सीधी फेस।
और ये कप का हैंडल भी इधर उधर होने के बजाय सीधी तरफ एन चम्मच के दरमियान में ऐसे रखा जाएगा। और ये बिस्किट भी ऐसे नहीं रखे जाएंगे। मीठे वाले बिस्किट राइट साइड पर ऐसे,
ऐसे।
और नमकीन वाले
Follow the protocol.
लेफ्ट साइड पर ऐसे। और हर लाइन में तीसरा वाला बिस्किट उल्टा होगा, ऐसे। और ये ट्रे भी इस तरह से नहीं।
दिमाग के कीड़े के अलावा हर कीड़े को मारे, सिर्फ एक हुस में किंग टॉक्स छोड़ेगा नहीं, मारेगा चुन चुन के।
छोड़ेगा नहीं, मारेगा चुन चुन के। दिमाग के कीड़े के अलावा सारे कीड़े मार दे। डॉक्टर सुकांत सिंह, मेरे प्रोटोकॉल तो फिर भी बहुत इजी टू फॉलो है। डॉक्टर ढाकले के प्रोटोकॉल तो बहुत सख्त है, मुझसे भी ज्यादा सख्त। सर कड़क है, कड़क। प्लीज गिव ए बिग राउंड ऑफ अप्लॉज टू दी ऑर्गेनाइजिंग सेक्रेटरी डॉक्टर गणेश ढाकले। He is rocking. Please stand up, sir. All India Institute of Medical Sciences, Nagpur, is rocking with each and every event. Probably you will agree that this was very, very boring event, and probably we have made little bit efforts to make it interesting. We are going to start next round.
Hello. Next round will be carried forward by Dr. Rahul, our third quiz master. Explain why. And first rule of round 3.
Now, welcome to round 3 for explain why and justification rules. The rules are same. Each team will be asked 2 questions. Only slight change is there. Time is given extra time. First 30 seconds for understanding the question and next 60 seconds for your response. Correct answer, you will get 10 marks, and if wrong, then other teams has a chance to gain 5 marks. So team same. Rules all these are same as in the round 2. Now, this time for team C. Question first of round 3 will be given to team C. Please display the question. Welcome to the NAPTICON Quiz 2024, the third national grand quiz of NPT, exploring frontiers in modern pharmacology. Team C, here is the question. Now you will be given some more time. Do you need more time?
30 seconds we can give.
We are giving 30 seconds. Would like to attempt?
Are you ready with your answer? No? Okay, so this is chance for other teams. Please collect the slips. Your slip? This one. Okay.
किसी का आंसर सही है क्या?
Yes, sir.
Dr. Parul,
All other teams gave the right answer. So 5 marks to all of them. Congratulations, other teams.
please explain. Explain the question.
Okay. This is chelation of aminocycline by the calcium. So that's the reason for reduced response.
Thank you, sir. Next question.
Next question for team D. Welcome to the NAPTIC quiz 2024. And the question is on the screen for team D.
We are giving again 30 seconds to think.
30 seconds.
Yes, that's good. You have submitted your response. Yes, answer is correct. So,
Please explain,
no
sir.
chance for other teams.
Please explain the answer.
Primaquine was missing from the treatment. Okay?
Thank you, sir. Should I go for next
Yes.
question?
Question 3 is for audience. Welcome to the NAPTIC quiz 2024, the third national grand quiz. This is the question for audience.
Don't answer yet. Don't answer yet.
Yes,
We have-
raise your hand.
We have two people. We have identified, sir.
Tobramycin causes skeletal muscle relaxation.
Yes, next, sir. Your chance.
I already gave. Tobramycin causes skeletal
Next,
muscle
okay.
relaxation.
Anyone else?
It is a nephrotoxic drug. Looking at the age of the patient, 76 years, I guess it should be lesser.
Okay, yes, you are right. The condition of the patient, like immobility and the breakdown of the muscles, will lead to the rise creatine phosphokinase.
Is
and
it the
the
right
drug is
answer,
also
sir?
nephrotoxic.
Is
Yes,
it the right answer? मुंह मीठा कराइए.
answer is right.
कैप
Now-
दीजिए, मुंह मीठा कराइए.
Now, second cap.
Should I move to the next question?
Yes, sir. Next question for team A. Yes, the question is on the screen. Team A.
We are giving some time to think. Please see whether somebody have given the answer.
Post collect these clips from other teams. Are you ready with your answer? Yes, explain.
Can you explain?
सर क्लोरेटिलिन जो है दैट इज अ माओ इनहिबिटर सो एमिट्रिप्टिलिन जो मेटाबॉलिज्म है दैट इज इनहिबिटेड सो देयर इज नो रीअब्जॉर्प्शन ऑफ द नॉरएपिनेफ्रिन सो नॉरएपिनेफ्रिन विल बी इंक्रीज्ड इन द सिनेप्टिक लेफ्ट एंड देयर विल बी हाइपरटेंसिव क्राइसिस
So,
Is it the right answer, sir?
sir, partly correct.
But
What is the ultimate reason for this hypertensive crisis?
sir, partly correct. We won't consider the right answer. He has.
Do you want to add anything in your answer?
सर इट इज अ माओ इनहिबिटर तो उसकी मेटाबॉलिज्म
Either is it right or wrong? Sir, you made two decisions. Is it right or wrong?
Not, it is wrong.
Wrong. Wrong answer.
Yes, now the chance for other teams.
So both the drugs if given together will cause serotonin syndrome, and that is what will precipitate the hypertensive crisis in this patient.
Mainly, other teams? Show me the answer of other teams. Cheese reactions. Any other? Serotonin syndrome has occurred. Okay.
Did somebody have the answer question?
Yes, from audience.
No question from the audience. We are moving because the time is also moving
Okay.
fast.
So no.
We are still skipping.
So no marks for any team.
No marks for any team. I am moving to the next questions. Question 5. We will be giving a break in between for the audience.
Answer. Sir. Okay.
Answer.
Okay, let me explain the answer. There are two drugs, amitriptyline and clozapine. For the previous question, I am explaining the answer for. So. Okay, so there are two drugs, amitriptyline, tricyclic antidepressants. This decreases the reuptake of Na into neurons, and other drug is clozapine MAO inhibitors, which decreases the metabolism of catecholamines. So both drug potentiate each other action and increase the Na activity. So that's the reason. Okay.
Yeah, they have given probably the answer, right answer. So you can consider this.
Okay.
सर कड़क प्रोफेसर
Okay.
मत बनिए, थोड़ा रहम करिए बच्चों पे.
Okay, to give full marks to team A.
I am moving with the next question.
Now, next question is for team B. Welcome to the NAPTICON quiz 2024. For team B, here is question on the screen.
You are lucky enough, professor is considered enough, not the correct professor.
Reason is bradycardia.
Yes.
Additional bradycardia, hence the patient is gone.
Okay, answer is right.
Big round of applause.
Both drugs produce cardiodepression, so additional cardiodepression.
Should I move
Okay,
to the
next. Next question is for team C. Here is the question.
giving some time to think.
You will be given extra 30 seconds if you need. And if you're ready, you can give your response. Team C, are you ready? There is no negative marking. You can try.
Due to excess release of neutrophils.
No. Please collect the slips of other teams. Slip. You don't have slip? Okay, yes. Your response? Give the answer.
Sir, aspirin causes hyperuricemia, so it is contraindicated.
Okay, that's correct. And from team B? Okay, was the slip? Campus? What have you written? Accretion of probenecid is increased by no. Okay, so answer by team A is correct.
Round of applause.
Actually, there are two reasons for this. One is aspirin is
Please
antagonizing.
leave the decision on the judges.
As aspirin is antagonizing the uricosuric
Judges
effect
can
of
decide
probenecid.
whether the answer
and
was right or wrong. Marks to be given. Dr. Balla, Dr.
And
Ritu Shatak.
there are two reasons. Aspirin inhibits the tubular
You can
secretion of
assist out
uric
as an
acid.
umpire. Please assist us
Yes.
in deciding whether marks to be given or not. Only one decision, please. Right or wrong? Right or wrong? Right or wrong? Sir, please, can you
Please repeat your answer.
What's your answer?
Sir, I said that the aspirin causes
Not cause
hyperuricemia,
hyperuricemia
so
in
it is
such a low dose.
Sir, you need not repeat the answer. They will make the answer. You need to decide right or wrong.
Okay, wrong.
because it causes
Wrong?
hyperuricemia.
This is the wrong answer as decided by the judges. Please
Okay, so answer is wrong.
take the
Answer is
honor of the
wrong.
judges.
Okay,
Thank you.
as per the judges, no marks is given to any team.
Yes, this is correct.
Next question, I am moving.
Next question is for team D.
Take the judges' decision into consideration.
Here is your question.
सर एक ऊपर नीचे हो रहा है थोड़ा तो बखशिए we have kept the judges to make this final decision and we need to honor them.
Please collect the slips of other teams. Are you ready? Yes. Explain.
So, sir, the patient has been added spironolactone, which is a potassium-sparing diuretic, and thus the patient has landed in hyperkalemia, and the ECG changes are typical of.
Yes, answer is correct. Full
Big
10
round
marks
of
to
applause.
team D.
Next question.
Next question for audience. Here is the question on screen for audience.
Please give the answer quickly. Any hand, raise of the hand, please notice. Those who are volunteering of helping us, please note, raise of the hand. No sir, आपको एक बार मिल गया ऐसे थोड़ी ना और दूसरों को भी मौका दीजिए.
Because of inhibition of this metabolism.
Rahul sir, please note the answer.
Yes, please.
It is because of the accumulation of norpethidine.
Yes, answer is correct.
मुंह मीठा कराइए.
The metabolism is
दीजिए.
shifted towards norpethidine, which has excite reactions. Okay. Next question is for team A. Team A, please get ready. The question is on screen.
And after that, a rapid fire round is coming. That is the final round.
Don't worry, you will get extra 30 seconds. Are you ready with your response? Team A? Please collect slips from other teams. Yes. B. Yes, team A.
Anybody has given the answer, right answer?
Yes, team A, please explain the answer.
Sir, propranolol is a beta blocker which causes the which masks the hypoglycemic action, which masks the warning symptoms of the hypoglycemia.
Okay, answer is right.
Right answer!
Full marks
Big
to
round
team
of
A.
applause.
Next question for team B. Team B, are you ready? Please display the question for team B. Welcome to the NAPTICON 2024. Here is the question for team B.
Last question of this round.
Explain the reason for the symptoms. There are certain symptoms. So what's the reason for the appearance of these symptoms? Tell the reason for the symptoms in this case.
Some extra time also.
Are you ready? Are you ready for your response? Yes? Explain. Abdominal colic is a symptom. What's the reason behind this?
Antacids will disturb the absorption of calcium.
And what's the name of this condition or syndrome?
Rahul sir, quickly decide whether the team has given the right answer or not. Or you can move.
Sir, we may consider it right.
Okay, this is the right answer.
It will increase the calcium absorption and it will lead to metabolic acidosis and is called the milk alkaline syndrome. So thus they tell the reason. So they have rightly described the reason. So full marks to team B. That's the end of round 3. A big applause to all the teams.
Now we are calculating the marks and तब तक आप लोग एक advertisement देख लें. And now the highest 3 team will be selected.
इन बच्चों का बाप कौन है? रंजन, मनु, बेटी जो एक बार चला जाता है वो कभी वापस आता है? तुम्हें दूसरी शादी
नहीं काका, रंजन और मनु के बाप आएंगे।
रात को करवट बदलने की बहुत आवाजें आती हैं। चरत मरत।
हमारे बाबू जरूर आएंगे।
सरला, मैं इन बच्चों को एक बाप का प्यार देना चाहता हूँ।
अकिल, हमारे बाबू आएंगे।
कब आएगा तुम्हारा बाप?
आंसू के कड़वे घुट पी ले रंजन, हमारा बाप आएगा।
कब आएंगे रंजन मनु के बाबी?
ए लाला, हमारे बाबू आएंगे।
कब तक छुपेगी कैरी पत्तों की आड़ में? कभी ना कभी तो आएगी भिंडी बाजार में।
ए बिस्कुट।
रंजन, मनु, कब आएगा तुम्हारा बाप?
कब आएगा मनु? रंजन का बाप?
कब आएगा तुम्हारा बाप? कब आएगा रे तुम्हारा बाप?
कब
आएगा तुम्हारा बाप? कब आएगा तुम्हारा बाप?
आ गया मनोरंजन का बाप। DLF इंडियन प्रीमियर लीग सिर्फ मैक्स पर।
Thank you for enjoying the session with us. We have kept this to make the environment lighter. I am here to announce the result also. So this was the 3rd round and with this round, I am going to announce. Any guess which team is leading? We have already told that with this round we will be going to eliminate one team. So unfortunately, we are going to eliminate one team. But our best wishes with the team who is being eliminated. Let me announce the team who is being eliminated. Team BA is being eliminated with the lowest score, 25 marks. Thank you for your cooperation. Team Kannauj, thank you. Our best wishes, thank you for the participation. And participation is much more important than the winning. Let me announce the scoreboard. With this end of this round, Team B have scored 55 marks. Team C have scored 35 marks. And with this end of this round, Team D is leading with the 60 marks. Team D is leading with the 60 marks. Congratulations, Team D, and that we are going to start last round of this NAPTICON 2024. Round 4.
Rules लगा दीजिए सर. तो round 4, true and false. True and false. So 1 minute, total duration is 1 minute, and team D, है ना? Total duration is 1 minute. True and false you have to tell. Okay? How you can attempt? You can attempt. If you don't know, then
this
you
is
can
going
pass.
to be very quick round. Please note it carefully.
Total 15 question we can ask. But if time is finished, then it's okay. Timer है ना? Timer. हाँ.
Timer will also appear.
जब बोलेंगे तब ऑन करना है. जब पहला क्वेश्चन आएगा तब से ऑन करना है. अभी 0 कर दीजिएगा.
सर जल्दी से राउंड के टीम ये रूल एक्सप्लेन कर दीजिए फटाफट.
The top 3 team will be selected at the end of round 3. The team with the lowest score at the round 3 will be eliminated. The top 3 teams will be complete rapid fire round. Each team will be given 1 minute, maximum 15 questions.
Or 1 minute.
If 1 minute is over, then questions will be stopped. Each correct answer will be given +6 marks and wrong answer will be given -3 marks. The team will maximum score, and with previous all scores, they will be added and then final result will be declared. Right? तो आप लोगों के पास मौका है यहाँ से चमचमाती हुई ट्रॉफी ले जाने का. Right? तो रैपिड फायर आपको.
Are you ready with the rapid fire?
Okay. Then we have the 4th set of questions. Team D, you have to first start, and you can choose which set you want to answer. Set 1, set 2, set 3, set 4. +6 marks, -3. Right question, right answer, +6.
This is having a different marking system. Yes.
Yes.
Should I start, sir?
अभी सर सेट पूछ लेते हैं. Which set you will prefer? 1, 2, 3 or 4?
4, sir.
4 set, sir. 4 set.
Timer, please. Please start, sir.
फिर से start.
सर फिर से जब बोलेंगे तब. जब बोलें. अभी आप 0 कर दें. अच्छा 1 कर दिया. ठीक है. You are ready? The question will appear on this blue screen. You have to tell only right, true or false. Dr. Rahul and Dr. Parul will give the marks. Answer we will not disclose, right? And as many as you can attempt, you can attempt.
Should I start, sir?
Start, yes. Blood-brain barrier is deficient
True.
at CTZ.
Start.
True.
Atropine is used for treatment of bradyarrhythmias.
True.
Methotrexate is most commonly used disease-modifying agent. Next. Sampatrilat has additional inhibitory activity on neprilysin apart from inhibitory effect on ACE.
True.
Next. Thiazides are also used in nephrogenic diabetes insipidus.
True.
Next. Acarbose inhibits intestinal absorption of carbohydrates.
True.
Next. Olanzapine is associated with weight gain.
True.
Next, propofol is used for daycare surgery.
No prompting,
True.
please.
Thrombin inhibitor.
True.
Salbutamol is a short-acting beta-2 agonist.
True.
Time and here. 10 questions you have attempted.
Please note down the Dr. Parul.
No, 10 seconds. Yes.
Dr. Parul. Have you noted down?
Yes, sir.
Should I proceed for further, sir?
Yes. Team C. Which set you will prefer? Set 1, 2 or 3? Second set. Sir, second set. टाइमर जब बोलेंगे तभी स्टार्ट करिएगा. ठीक है, तो आप तो देख लिया ना सारा? Understood? Yes. Start the time. Sir, start question.
Yes.
Basic drugs are generally bound to plasma albumin.
False.
Next. Atropine is drug of choice for producing psychoplegia in children.
True.
Abatacept is a co-stimulation inhibitor used in rheumatoid arthritis.
Pass.
Next. Losartan inhibits the formation of angiotensin
Please
2.
speak bit louder.
True.
Acetazolamide is used for prophylaxis of mountain sickness.
True.
Next. Metformin causes weight gain.
False.
Next. Don't prompt, please.
No prompting,
Haloperidol
please.
can cause tardive dyskinesia and long-term use.
Yes. True.
Xenon has very high blood gas partition coefficient.
Pass.
Edoxaban acts as a direct factor 10A inhibitor.
True.
Formoterol can be used for acute attack as well as for prophylaxis of asthma.
True.
Enalapril can cause cough and angioedema.
True.
Time ends. Dr. Parul, have you noted down? Should I proceed further?
Then team A. Now 2 sets we have. Set 1 and 3. Which set do you prefer? Set 1. Sir, set 1. Mike.
Please speak a bit louder. Timer, please.
Hello.
No, जब सर कहें तभी करना start.
Start, sir. Question start. Timer start. Most drugs are absorbed in ionized form.
True.
Next. Atropine is used for treatment of belladonna poisoning.
False.
Serulumab is a monoclonal antibody against IL-6. Next. Sir, next. Aliskirin acts by inhibiting the secretion of renin.
False.
Please speak bit louder.
Thiazides are first-line drug in hypertension.
True.
Glipizide can cause hypoglycemia. Next. Clozapine can cause a granulocytosis. Next. Halothane is preferred anesthetic for pheochromocytoma. Rivaroxaban is an oral. Steroids alone are most effective drugs for acute attack of asthma.
True.
Next, atropine is first-line drug in angle closure glaucoma.
True.
Insulin is preferred in most patients with type 2. Next. Sodium chromoglycate is ineffective in acute attack of asthma. Silostazol is PDE4 inhibitor.
Time ends now. Dr. Parul, are you ready with the marking? Should I proceed further?
Time लगेगा.
Team B कर.
So we are calculating the marks. Please bear with us. तब तक एक छोटा सा ब्रेक ले लेते हैं.
पानी जिसके लाल लाल नाम है उसका चुटकी. मम्मा, क्या फिर से रेड हो गए?
अब क्या बताऊं, ऐसे ही है सूरज. गुस्से में लाल नहीं, आग का गोला हो जाता है.
ये कहानी है गर्म सूरज की, उसकी गर्मी से परेशान उसके अपनों की. ये कहानी है इस गर्म शरीर के ठंडे सपनों की.
एक टॉय से नहीं खेल पाया मेरा बच्चा.
मंगलयान पर बैठ के धरती पर आया ये एलियन. बाल देखे इसके लाल.
सूरज की अम्मा सूर्य वंशम देखी रही 150 बार, तभी ना पैदा हुआ है सूर्य वंशम.
मासूम सूरज अपनी गर्मी से अनजाना था. आइसक्रीम खाने के लिए दीवाना था. सॉफ्टी पकड़ता तो जल जाती और दूध की कुल्फी पिघल जाती. घर की चीजों में भी गर्मी दिखती रही।
तू हमारा हाथ जला देता है।
पेरेंट्स की टेंशन बढ़, पर भाई हौसला तो रखना था। भाई बच्चा भी तो अपना था। ठंडे को गर्म करना, बाल्टी के पानी को बॉईल करना, बहन के बालों को स्ट्रेट करना और कभी कभी मस्ती में डेयर डेविल बनना।
मम्मा, सूरज ने मेरा रॉकेट जला दिया।
फेस पे कल्लन के खेत की मुल्तानी मिट्टी, हेड पे बच्चन की दुकान का ठंडा तेल, पीठ पे शाहरुख का कूल कूल पाउडर भी उड़ गया और मुन्ना बर्फ डिपो का आइडिया भी भाप बन गया। माँ बाप ने क्या क्या नहीं किया। मंदिर की घंटी, चर्च में फादर बंटी, गुरुद्वारे की सीढ़ियां और दरगाह की कवालियां, पर सूरज ज्यों का त्यों रहा मियां।
बस अब लास्ट रिजॉर्ट है इसकी शादी।
शायद इसी से इसकी गर्मी निकल जाए।
लड़की के घर चाय की चुस्की चल रही थी। सूरजमुखी शर्माते हुए भजिया तल रही थी। इतने में लड़की के पप्पा छिल्लर जी बोले,
तो चक्रवर्ती साहब रिश्ता पक्का समझे? हाँ हाँ जरूर। पर हम आपको एक बात बताना तो भूल ही गए।
चक्रवर्ती जी ने किया साइलेंट मोड। छिल्लर फैमिली के सामने की सूरज की गर्मी डाउनलोड। सुनकर छिल्लर जी फिर से बोले,
बस इतनी सी बात। बेटा सूरजमुखी, अरे सबका मुंह मीठा कराओ भाई।
सूरजमुखी झट से कुल्फी ले आई, एक एक करके सबको थमाई।
थैंक यू, थैंक यू।
ले लीजिए।
बट इस बार सूरज की कुल्फी ना पिघल पाई और ये देख छुटकी चिल्लाई,
मम्मा, भैया ठीक हो गए।
रिश्ता पक्का हो गया। सूरज वाकई चंगा हो गया। इस तरह हमारी ठंडी गरम स्टोरी का हैप्पी एंड हो गया।
Thank you. Are you enjoying? Did you enjoy it? This is we are ending with the quiz. Thank you very much. And your participation is much more important than our participants themselves. We are getting too late. It is the time for the lunch also. And I can see on the faces that you are taking a lot more interest. Thank you very much from the bottom of my heart that you have given preference to the NAPTIC quiz and you are waiting for the lunch. We are going to announce the result, and with this I would like request to the dignitaries to please sit. Let me announce the result. We are going to give the awards also. Okay, so any guess? So with this, the team C unfortunately has got the minimum marks, 77, and second team is, any guess? B is with the 94 marks, and team D is the winner, 120 marks. Team D is the winner. Please give your introduction again. From which medical college? Please give your introduction. And I would like to invite the President sir, and if General Secretary is also here, to please come on the dais. And I would like to request other teams, they can take their seats. We are going to felicitate the winner team. Thank you for your active participation. Other teams. Team can take their seats. Yeah. Other teams can leave the stage. Thank you for your participation. Team D, please come forward. गाउन लाए जाए। एक मेंबर को प्रेसिडेंट गाउन पहनाएंगे, दूसरे को जनरल सेक्रेटरी गाउन फेलिसिटेट करने के लिए। कांग्रेचुलेशंस, ए बिग राउंड ऑफ अप्लॉज। इसको रहने दीजिए। एंड ट्रॉफी लाई जाए। So with this, NAPTICON 2024, we are going to announce the winner. They are on the stage. Introduction again, please. From which medical institution? AIMS Raipur. Big round of applause. Winning trophy is being given. Thank you. Please come forward, all the judges and quiz master, for a photo session quickly. Thank you.
बता देते हैं अच्छा
Dr. Ritu Shatak and Dr. Hira Balla, thank you very much for accepting our request. And you can give them the trophy. Both judges, please give them the trophy. Dr. Hira Balla to one candidate, Dr. Ritu Shatak madam to another candidate. Madam can give the trophy to one team and Dr. One person and Dr. Hira Balla to another person. Dr. Thank you very much being associated with us. Don't leave the stage, please stay with us. Yeah. All India Institute of Medical Sciences, Raipur is rocking with the winner of the NAPTICON 2024, third national quiz of National Association of Pharmacology and Therapeutics. Thank you for your active participation.
One thing more, same college has won second position in Bangalore. Second position in Bangalore, same college, students are different.
And congratulations to all the teachers and mentors behind this successful team. A big round of applause for the team, All India Institute of Medical Sciences, Raipur. Congratulations from National Association of Pharmacology and Therapeutics. With this, I am going to conclude. Dr. Sarvesh.
And also winner of NAPTICON 23 in Lucknow, KGMU Medical, KGMU Lucknow. तो लगातार 3 बार से ये लोग जीत रहे हैं, सेम कॉलेज. मैं जो आपके कॉलेज के अगर प्रोफेसर हैं यहाँ पे तो वो लोग भी स्टेज पे आ जाएं फॉर फोटो सेशन. सेम कॉलेज के अगर प्रोफेसर्स हैं तो स्टेज पे आ जाएं. सेम कॉलेज रायपुर कॉलेज के अगर प्रोफेसर्स या अदर बैचमेट्स दे कैन कम टू स्टेज. Same college wins, runner team in Bangalore, winner team Lucknow NAPTICON.
We are really proud on the mentors and the teachers for producing the successful students. And always teacher is the behind the success of all their students. And this is the trophy for All India Institute of Medical Sciences. Congratulations to both the teachers and faculty members from All India Institute of Medical Sciences. Raipur, hip hip! Hip hip! Thank you very much for joining us. Thank you.
Please give a big round of applause to all the today's quiz master, Mr. Rahul, Dr. Sarvesh, and most important the person of the quiz is Dr. Kamal sir. Once more, give a big round of applause. Thank you, sir. Thank you.
Thank you so much,
Now
sir. It
we
is
can go ahead with the lunch and then we will meet after 15 minutes.
सर से ये पूछ लीजिए 10,000 का हम अनाउंस किया है, क्या कोई जीत पाएगा? ऐसा रेप्लिकेट करना है इसको।
जो नहीं हो पाएगा उसकी बात छोड़ो, अभी जो हो रहा है पारस पब्लिकेशन की तरफ से, आ जाओ बेटा, तुम लोग आ जाओ, पारस पब्लिकेशन की तरफ से एक स्पेशल प्राइज दिया गया है। बच्चे अरे ले के आओ भाई।
And before we take this ahead, can we please have the mementos for
There
our
are
quiz
other
masters and the
prizes
judges?
also. Book from Paras Medical Publisher. I would like to request Dr. R.K. Dixit sir and Dr. Hira Balla sir, please come forward to give other prizes to the winners. Book from Paras Medical Publisher.
Congratulations to the winners. And may I please call upon Dr. Alok Dikshit to the dais to felicitate our quiz masters as well as the judges for this NAPTI quiz.
Publisher, these books are being given. Thank you very much.
Quiz masters and judges. We're delighted to have Dr. Alok Dikshit to felicitate our quiz masters and the judges for the NAPTI quiz 2024. And just after this, we shall disperse for lunch and come back in the next 30 minutes. We shall have the valedictory ceremony to begin at sharp 3:15. Thank you so much. Mementos for all the 3 quiz masters. For the felicitation. A huge round of applause for all the quiz masters. And it is now time to felicitate our judges for the NAPTI quiz. Can we please have the mementos for the judges to be felicitated by Dr. Alok Dikshit? Heartiest congratulations to the winning team and thank you so much to the entire organizing team of the NAPTI quiz. We shall now disperse for lunch and come back in the next 30 minutes. We shall have a pink cap ceremony and a spotlight felicitation as well, followed by the valedictory. Thank you so much. We shall be back in the next 15 minutes. Thank you. We shall begin with the pink cap ceremony and the spotlight in the next 15 minutes to be followed by the valedictory ceremony. Thank you so much.
the upcoming launch of a DM in clinical pharmacology, the introduction of therapeutic drug monitoring services, and the establishment of a dedicated clinical pharmacology unit. At AIIMS Nagpur's Department of Pharmacology, we are shaping the future of healthcare through innovation, collaboration, and a relentless commitment to excellence. Thank you for joining us.
Welcome to Canvas Clinical Research Services Private Limited, India's leading site management organization for global clinical trials. Founded on February 25, 2019, CCRSPL stands tall on four unshakable pillars: quality, integrity, empathy, and modesty. At the helm of this powerhouse is founder Vijaya Bakate, a trailblazer with over 18 years of unmatched expertise in site management, setting a benchmark in addressing and resolving site-level challenges with precision. CCRSPL is the epitome of domain expertise, revolutionizing clinical trials by significantly reducing costs and timelines. We boast the fastest patient recruitment rates in the industry and an unparalleled 99% patient retention across diverse global studies. No other organization matches our agility and excellence in driving trial success. Our digital presence is unmatched, actively engaging life science students, clinical research stakeholders, and professionals, spreading awareness and fostering a connected research community. Our sites consistently dominate rankings across India for patient recruitment, data quality, and integrity, making us the go-to partner for global sponsors, CRO, and investigators. Bridge between hospitals, cross, and sponsor companies, CCRSPL ensures the seamless execution of clinical trials. We are the first choice for those seeking efficient, cost-effective, and results-driven site management solutions, backed by a proven track record of excellence. Our vision is bold and transformative, leveraging AI and advanced technologies to elevate India's global contribution to clinical research. Beyond trials, CCRSPL is shaping the future through specialized courses for life science students, empowering them to stand out in the competitive clinical research industry. Today, our alumni are excelling at top-tier organizations like PPD, IQVIA, LabCorp, Accenture, and TCS, cementing CCRSPL's reputation as a leader in education and professional development. In just 5 years, CCRSPL has established a monopoly in ophthalmology clinical trials in central India, conducting 9 successful studies and building strong, enduring partnerships with sites and investigators. Our expertise also spans a wide array of therapeutic areas, including cardiology, oncology, psychiatry, nephrology, gastroenterology, and rare diseases. This success is no accident. It's driven by a highly motivated, growth-oriented team perfectly aligned with CCRSPL's mission to redefine clinical research excellence. Recognized as a certified startup by the Government of India, CCRSPL is accredited by the Department for Promotion of Industry and Internal Trade, DPIIT. We proudly hold the Udyam certificate from the Ministry of Micro, Small, and Medium Enterprises, along with ISO 9001:2015 certification, underscoring our relentless commitment to quality, innovation, and industry leadership. Canvas Clinical Research Services Private Limited, transforming clinical research, empowering progress, and building a healthier tomorrow.
Indian Institute of Clinical Research and Management, ICRM, is a premier institution dedicated to advancing education, research, and training in clinical research and healthcare management, established in 2002, affiliated with Savitribai Phule Pune University. ICRM aims to create a global workforce equipped with cutting-edge knowledge and skills in clinical trials, clinical data management, clinical SAS and regulatory affairs, and healthcare leadership. Our programs are designed to meet the growing needs of the pharmaceutical, biotechnology, and healthcare industries, ensuring that our graduates are industry-ready and capable of driving innovation in these fields. Our activities: efficient recruitment strategies to enroll study participant with trial transparency and education with planning and design. Monitoring and analytics, utilizing data analytics to monitor recruitment progress in real time, allows for timely adjustments to strategies if certain approaches are underperforming. Monitoring and safety, adverse event monitoring, data security and regulatory compliance. Training, AI with clinical research, clinical data management and pharmacovigilance, medical coding, medical writing. Introduction of clinical research. It is a branch of healthcare science that involves the study of human health and illness through structured investigations aimed at improving medical knowledge, treatments, and patient care. It covers a broad range of studies, from understanding the mechanisms of diseases to developing and testing new treatments, drugs, medical devices, and diagnostic tools. Types of clinical research: observational studies, interventional studies, translational research. Clinical research requires collaboration across various domains, including medicine, pharmacology, bioethics, and data science, and is an essential component of improving healthcare. Introduction of artificial intelligence. It transforms clinical research, enhancing efficiency, accuracy, and patient outcomes. AI in clinical research. Data management and analytics. Patient recruitment and retention. Study design optimization, predictive modeling and risk assessment, medical imaging analysis, clinical decision support systems, personalized medicine, virtual clinical trials, real-world evidence generation, regulatory compliance, introduction of clinical data management. Pharmacovigilance is the science and activities related to detecting, assessing, understanding, and preventing adverse effects or any other drug-related issues. It plays a critical role in ensuring drug safety by monitoring the effects of medications post-approval, aiming to protect patient health and improve therapeutic outcomes. Data collection and validation, data cleaning and quality control, data storage and security, data retrieval and reporting, compliance with regulations. Introduction of pharmacovigilance, the science and activities relating to the detection, assessment, understanding, and prevention of adverse effects or any other drug-related problem. Introduction of electronic trial master file. Electronic trial master file, ETMF, is a digital system used in clinical research to manage and store essential documents generated during a clinical trial. Serving as a centralized, compliant, and accessible repository, the ETMF ensures that trial documentation is organized, easily retrievable, and audit-ready throughout the study lifecycle. It enhances efficiency, improves collaboration among stakeholders, and supports regulatory compliance by maintaining a comprehensive record of all trial-related activities and documents. Career in clinical research. Clinical research coordinator. Clinical Research Associate, Healthcare Data Analyst, Clinical Data Analyst, Drug Safety Associate, Pharmacovigilance Officer, Regulatory Affairs Specialist, Health Informatics Specialist, Regulatory Submission Specialist. Thank you.
Good afternoon, one and all. Some exciting news for all of us. The IICRM has a special lucky draw for all of us, and to conduct the same, may I please call upon from the IICRM, Mrs. Suman Menon and also Mr. Rajesh. We can clap for them. They're getting something really special for all of us. A surprise element at the NAPTICON 2024.
So good afternoon all. So this is a very good excited session from last 2 days and very well organized conference. I congratulate all of you and especially Ganesh sir and his team to organize such a vast and a wonderful conference. So many brainstorming session was there, so many academic session was there. But now this session is a very exciting session. I hope everyone might have write down your name in our lucky draw chit and you might have put over there in our lucky draw box. So here we are going to announce 5 lucky winners for a lucky draw, and I request Ganesh sir to please welcome over here and just yes. So just you have to pick up 5 lucky winners sir over here. Mike. Ganesh sir, anyone else you want to invite for a... Okay.
So, how many prizes are there? 5. Okay, all are here?
Yeah, all are here. All means prizes are there.
Okay.
The names are here.
Okay. So,
I would like to
I will...
request you to just give a big clap when... Yes.
And the first winner is Dr. Deepak Langde. हाँ लॉटरी दे देना
आवाज यहाँ सर
नेक्स्ट आने दो आओ दीपक सर
Congratulations sir.
कॉन्ग्रेचुलेशन दीपक पहली बार लगी The next winner is Dr. Atul Raut. Give a big round of applause. Yes, Sun Pharma. Sir is from Sun Pharma, Vice President. Congratulations, Dr. Atul. Congratulations. Next is Dr. R. Kavitha. Give a big round of applause. Hello. Okay, this one. Yes, sir.
Yes, sir, please. So everyone must charge.
The next lucky person is Dr. Nikhil
Mobile.
Munjbele. Is he here? Dr. Nikhil? Arey, wow.
Congratulations, Dr. Nikhil, and just give a big round of applause and clap for him, Dr. Nikhil. Congratulations, please. Yes. Congratulations, sir.
Congratulations, beta. Now we have last?
Yeah, the last.
Okay.
Please, sir.
The last winner is Dr. Chaitali Bajait. So congratulations, Chaitali.
Tali ma'am has just write down her name 1 year before, 1 hour before. And really, you are very lucky and congratulations, ma'am. Congratulations.
A big congratulations, Chaitali.
Please, ma'am. Please, sir. आपके हाथ से दीजिए.
हाँ, कोई बात. मेरे पापा फोटो के हैं.
So congratulations all lucky winners and thank you very much for allowing us for this lucky draw contest. Thank you very much. Thank you very much. Thank you.
Thank you so much. And heartiest congratulations to all the winners. We shall shortly begin with the pink cap ceremony and the spotlight felicitation, to be followed by valedictory ceremony. Request you all to please be seated at your places, and please inform your fellow delegates that we are about to start with the further proceedings in the next 5 minutes. Thank you.
Thank you all. Welcome to National Association of Pharmacology and Therapeutics. Thank you very much for being with us till the last of the NAPTICON 2024, the third National Conference of National Association of Pharmacology and Therapeutics, being organized by All India Institute of Medical Sciences, Nagpur. It is time to give the awards to the various winners, and before we can go to the awards, we have started a trend of pink cap ceremony. It is an old ceremonial message that is being followed all over the country. Pink cap denotes the responsibility. It denotes the responsibility. And it from one organizing secretary to another organizing secretary, in the presence, of course, of the President, Honorable National President by Dr. R.K. Dixit. So, Dr. K. Bhuvneshwari, please give a big round of applause. She has taken the big responsibility of organizing next NAPTICON 2025 at the city of Coimbatore, South India. Congratulations, madam, from the National Association and the entire member. Please come to the dais. I would like to request Dixit sir also to come on the dais, and Dr. Ganesh Dhakle, Organizing Secretary of NAPTICON 2024. So next NAPTICON venue, please come on the stage. Venue is the Coimbatore. It is being organized by PSG Institute of Medical Sciences, Coimbatore, and probably I feel Dr. K. Bhuvneshwari needs no introduction. She is one of the legend of the pharmacology, and moreover, she has taken the responsibility of next NAPTICON that will be organized in Coimbatore. So, I would like to request Dr. R.K. Dixit sir to please give her, along with Dr. Ganesh Dhakle, both, please felicitate madam, Dr. Ganesh Dhakle and Dr. R.K. Dixit with the pink cap. Congratulations, madam. Welcome to Coimbatore next year. It will be organized on 28th and 29th November. The theme of the conference is crafting clinical excellence with pharmacology and therapeutics. Thank you all of the members who are sitting in the hall, and hearty congratulations from National Association of Pharmacology and Therapeutics and the entire family of National Association of Pharmacology and Therapeutics. I feel she also deserves a standing ovation for this. Thank you, because it is a big responsibility. We have seen how Dr. Ganesh Dhakle have put the level of NAPTICON to such a level that for next and next team of NAPTICON, it is becoming difficult and difficult. And when it, please sit down, thank you very much. And we know it very well that with the passage of time, when it is becoming difficult and difficult, it is becoming very good sign for the rise of National Association of Pharmacology and Therapeutics. Thank you very much. Together we have seen a dream that pharmacology should reach to those heights where it deserves. I am quite confident that under the leadership of our current President, Dr. R.K. Dixit, Honorable President, National President, Dr. R.K. Dixit, who is leading the association in such a way that he is leaving no stone unturned, and National Association of Pharmacology and Therapeutics is doing excellent job under his leadership. He always available for guidance, and under the guidance we are blessed with the leadership of Dr. R.K. Dixit sir, our National President. Thank you, R.K. Dixit sir, our National President. And thank you very much, Dr. Ganesh Dhakle, our General Secretary, and the Organizing Secretary of NAPTICON 2024 for taking up to this stage where it is quite visible that I can say from this dais very confident, with very confidence, that pharmacology's future is very high, and it is going to be one of the favorite branch of the next generation of it. Thank you very much for joining this small ceremony. Thank you. So at NPT we are dedicated to promote pharmacology and therapeutics for a better tomorrow. Before I can conclude, I would like to request Madam if she can also say a few words around for the NAPTICON 2025 and her organizing preparations. Thank you, Madam Dr. K. Bhuvneshwari. Thank you.
Respected President, Dr. R.K. Dixit sir, the most dynamic Secretary, NAPTICON 2024, Dr. Ganesh sir, respected National Coordinator, Dr. Kamal sir, and most supportive Vice Presidents, Joint Secretaries, and Finance Officer of this Noble Association, Head and members of Variable Academic Board, Editorial Board, RS Factor, Clinical Research Board, and all members of National Executive Council, and our most dearest faculty and PG delegates. Good evening to all. This is my privilege and pleasant moment to accept this important responsibility on behalf of our August Association, National Association of Pharmacology and Therapeutics, as a secretary for the conference NAPTICON 2025, which is going to be held at 100 years old education legacy, PSG Trust Medical College, PSG IMSR Coimbatore. I assure that as a coordinator, under the support of the guidance of our president and also the support of a national coordinator, as a conference secretary, and with the support of all the members of the variable board members, state coordinators, I will do my level best with my maximum ability. And now itself, I welcome you all to join and participate and encourage 2025 conference proceedings and enjoy in visiting South India, Manchester, Cotton City, a place in India gifted with world's second tastiest water, ceremony water, a city which is popular for educational institutions, automobile industry, health and medical tourism, a traditional city which is known for unique Tamil culture and more diverse and cosmopolitan population when you compare with the other cities in Tamil Nadu, which is none other than my own city, Coimbatore, Tamil Nadu. And this moment, I really thank the National Association of Pharmacology for selecting our institution and my department as one of the supporting activity as a national secretary and also the organizing secretary for the forthcoming conference 2025 at Coimbatore. Thank you very much, sir. Thank you, President and Present Secretary and our National Coordinator, Dr. Kamal sir, and all the supportive board members. And definitely, I just want to say thanks to my family members. And my husband has come as a senior lawyer of Coimbatore, and all my faculty from my department. And very important thing is my institution, PSG IMSR Coimbatore, who's going to support a maximum during this conference. And I feel it is very much privileged moment for me in my career as a responsibility which is shed on my shoulder at this moment as a secretary for the forthcoming conference. Definitely under the support of the Nobel Association, August Association, National Association of Pharmacology Therapeutics, I will do my level best as a secretary, and thank you for providing this opportunity. Long live NPT and Jai Hind.
Thank you very much, madam, and congratulations from all the members of National Association of Pharmacology and Therapeutics for taking this responsibility. You can carry the cap also till next NAPTICON is being organized. And you may take the seat there. No, you can continue with the cap. And you need to continue with the cap. This is a bigger responsibility, and you are looking gorgeous in that cap. Probably you all have seen that our Lifetime Achievement awardee have also not removed the cap when he was standing over here at my place. Now next is the time for the next awards. Together we have seen the dreams of taking the National Association of Pharmacology and Therapeutics to the newer heights, presenting the National Spotlight Awards. And with these words, the National Spotlight Awards of the National Association of Pharmacology and Therapeutics has been introduced to highlight the profile of renowned life members of the organization who have made significant contribution in pharmacology, clinical pharmacology and therapeutics. Every month, National Spotlight Award has been instrumental in inspiring the next generation of the doctors to pursue pharmacology and therapeutics as an exceptional career path. This award fuels their ambition, encouraging them to refine their skills and make an impactful efforts to national and international level. The chosen profile is prominently displayed on the website of our organization and on the Twitter handle of the website. Twitter handle of the National Association of Pharmacology and Therapeutics. National Spotlight Award recipients are also being honored. It is my pleasure to invite Dr. Meera K. Desai, if she is present over here. Dr. K. Chirita from New Delhi, Dr. Saroj Kothari from Gwalior, Dr. Sangeeta Bhavra from Chandigarh, Dr. Deepak Kumar Langade from Mumbai, Dr. Prakash BM from Andaman and Nicobar, Dr. Meghali Chalia from Dibrugarh, Assam, Dr. Langde please, Dr. R. J. Lalita, Dr. Julius Anthony Voss, Dr. Sumana Sen, all can come to the dais to receive National Spotlight Awards. Bishwadeep Das, Dr. Bishwadeep Das. Dr. Julius Voss, if present, can join us. Dr. Meghali Chalia. Thank you, madam. Dr. Mukund Kenny, Mukund Raj Kenny Anybody is left whom I forget to announce, please come on the dais to receive National Spotlight Award who have been announced. Dr. Mukund Rajkani, Dr. Jaya Lalita, thank you. Congratulations from National Association of Pharmacology and Therapeutics. Dr. Biswadeep Das, Dr. Mukund Rajkani, don't leave the stage, please. Thank you all the Spotlight winners, National Spotlight Award winners from National Association of Pharmacology and Therapeutics, and congratulations. रिसर्च अवार्ड आप करेंगे? आप अनाउंस तो मैं करवा दूँगा, ये आपको पता है सब? ठीक है, वो पेपर है क्या जिस पे रिसर्च अवार्ड है? पहले मैं इसे कर लूँ, पहले इसे कर ले तो उसके बाद National Association of Pharmacology and Therapeutics is committed to advancing the field of pharmacology and therapeutics. It is a national initiative to start these awards, research awards from National Association of Pharmacology and Therapeutics, which are being given in various categories to be awarded during the national conference each year. These are the various award categories you are already aware of. National Research Award for Excellence, research publication with the highest impact factor by pharmacologists. Pharmacology, clinical pharmacology therapy, highest number of research publication by pharmacologists. National Therapeutic Research Award introduced this year for innovation, research excellence, remarkable contribution in the field of the therapeutics. National Award for the best publication in National Journal of Pharmacology and Therapeutics. So this is showcasing, proudly showcase your research last year these awards have been given, and these have been started with the Lucknow conference. I would like to request Honorable National President Sir, who have already announced these awards through the all over the country from Kashmir to Kanyakumari through video message yesterday also, but the award are being physically given by with the hands of the National President Dr. R.K. Dixit Sir. I would like to request R.K. Dixit Sir to please announce all these awards. So first award, so the National Research Award for Research Excellence, Highest Impact Factor, Senior Faculty, Dr. Bishwadeep Das, Professor, All India Institute of Medical Sciences, Rishikesh. Highest Impact Factor in Junior Faculty, Dr. Siddhartha Datta, Assistant Professor, All India Institute of Medical Sciences, Rajkot. Highest Impact Factor in the Consultant category, Dr. Shambhu Samrat Samajdar from Kolkata. Highest Impact Factor in the Resident category, Dr. Nalini Kumari, Senior Resident, Apollo Institute of Medical Sciences, Hyderabad. Dr. Bishwadeep Das. Congratulations, sir. Dr. Siddhartha Datta. All are requested not to leave the stage till a photography session. Dr. Siddhartha Datta. Dr. Shambhu Samrat Samajdar, congratulations, sir. Dr. Nalini Kumari from Apollo Institute of Medical Sciences, Hyderabad. Next, yeah, congratulations, sir. Dr. Datta. Dr. Shambhu Samrat Samajdar from Kolkata. Stay on the stage. There will be a group photograph also with these certificate and the awards. Dr. Shambhu Samrat Samajdar from Kolkata. Dr. Nalini Kumari, Senior Resident, Apollo Institute of Medical Sciences, Hyderabad. Next category is the highest National Research Award for the Research Scholar 2024 for the highest number of research publication by a pharmacologist. The senior faculty, the prize has gone to Puga Janathan Thangaraju, Associate Professor, All India Institute of Medical Sciences, Raipur. Junior faculty, Satish Eknath Behrekar, Assistant Dr. Puganathan Thangaraju, Associate Professor, All India Institute of Medical Sciences, Raipur. Dr. Satish Eknath Behekar. Next is Dr. Santosh Tor, please. And after that, Muhammad Akib Shamim. Dr. Satish Eknath Behekar, Assistant Professor, Department of Pharmacology, Government Medical College, Aurangabad. Congratulations, sir. Dr. Santosh Tor, Director, Medical Affairs, Vaccine and Digital, Pfizer Limited, Mumbai. Congratulations, sir. In resident category, Mohammad Aqib Shamim, Junior Resident. Mike, please. Mike. Mohammad Aqib Shamim, Junior Resident, All India Institute of Medical Sciences, Jodhpur. Mike, please. Please stay. Let me tell something. Dr. Dixit will also tell something about Dr. Mohammad Aqib Shamim. Is that he is the junior resident, he is GR3, and I would like his mentor to show her face to all the members of National Association of Pharmacology and Therapeutics. Dr. Sneha Ambani, she is the HOD of the department, resident GR3, and Dr. Dixit will tell you the story I have already told.
I was amazed to see the profile of this young student, JR. You are JR3, na?
JR3.
Real? And he has published around 50 papers. Now that's amazing. And continue, please, this journey, and your heights are above the sky. Okay? Good luck. Good luck.
Thank you, sir. Another important thing is, this guy who is having such a young age, you won't believe that our entire scientific committee and the entire award council is so impressed that he has bagged the award in both the categories. But due to the protocol, we have chosen one category and we have taken his permission also. But he has bagged the prize in both the category, highest impact factor as well as maximum number of research paper, and you won't believe it is exceeding to any consultant, any professor who have applied for these awards. And he is just resident GR3. Again, I would like to congratulate Dr. Aqib and head of the department, Dr. Sneha Ambani, for producing such a beautiful minds in the Department of Pharmacology and Therapeutics. These are the shining stars. These are the future of the pharmacology. I would like to request, there are two still more awards we have been, are still being announced. The National Research Award for Outstanding Research goes to Dr. Shikha Sood, Associate Professor of Pharmacology. I would like to request somebody if he can take on the behalf, if Dr. Vipul is here, he can take the award. Dr. Vipul Chaudhary. And the Best Therapeutic Research Award that we have started this year goes to Dr. Rima Shah, Associate Professor and Head Pharmacology, All India Institute of Medical Sciences, Rajkot. Dr. Vipul are requested to take award on behalf of Dr. Shikha Sood. Due to some emergency reason, she couldn't came to attend the award. She has communicated already. Congratulations. And Dr. Rima Shah, Best Therapeutic Research, Associate Professor and Head, Pharmacology, All India Institute of Medical Sciences, Rajkot. And this is because of these shining stars, pharmacology is also shining in the world of therapeutic and medicine. Congratulations to all the winners of these National Research Awards. Congratulations from National Association of Pharmacology and Therapeutic. Thank you.
Thank you so much. Heartiest congratulations to all the winners up till now. A warm welcome to all of you at the valedictory ceremony. Let us begin with the prizes of the oral paper presentation. And to give away these awards, we have on stage Dr. Dikshit, Dr. C.M. Kamal, Dr. Ganesh Dakle. May I please call upon our Scientific Committee Chairperson, Dr. Chaitali Chindarole, on the stage to present the awards for the oral paper presentation at NAPTICON 2024. Can we please welcome Dr. Chaitali Chindarole with a huge round of applause. We have been seeing her running here and there to make this conference a grand success. Thank you so much, ma'am, for all your efforts. Hello. The very first prize would be for the experimental pharmacology, and the first prize in this category for faculties goes to Dr. R. Kavita. Dr. R. Kavita, please welcome our winners with the loudest round of applause that you can. Dr. R. Kavita. Congratulations, ma'am. The next would be for drug interventional studies. And for the faculties, the first prize goes to Dr. Amrita Sil. Dr. Amrita Sil. तालियां बजती रहें. Do not stop applauding until our winners come to the stage and take away the prize. We have sir to take away the prize on behalf of Dr. Amrita. And for medical education in the faculties, the second prize goes to Dr. Ajay Khade. The second prize goes to Dr. Ajay Khade. And the first prize goes to Dr. Chitra Khanwelkar. Dr. Chitra Khanwelkar. Congratulations to both the winners. They would be taking away the prizes for the faculties for medical education at the oral paper presentation. The second prize for Dr. Ajay Khade and the first prize for Dr. Chitra Khanwelkar. Congratulations to both the winners. And the next topic, observational studies. Observational studies, and the third prize goes to Dr. Sara Banu, EPM. Dr. Sara Banu, EPM. The second prize goes to Dr. Sumana Sen. Dr. Sumana Sen. And the winner for the observational studies is Dr. Dhanya Sasidharan Pallapalil. Dr. Dhanya Sasidharan. Keep applauding, keep clapping for all the winners. NAPTICON 2024 के आखिरी पड़ाव में, और मैं चाहूंगी हमारी एनर्जीज पूरी तरह से हम यहाँ पर दिखाएं. जोरदार तालियां बजाकर हम हमारे विजेताओं को बधाई लगातार देते जाएं. Keep applauding, keep clapping. And for systemic review or meta-analysis, the first prize goes to Dr. Siddhartha Datta. Dr. Siddhartha Datta. Dr. Siddhartha Datta. Next would be for pharmacovigilance, and after Dr. Siddhartha Datta, we would have Dr. Bharti Karelia to take away the prize for pharmacovigilance. Dr. Bharti Karelia. Do not stop applauding. तालियां लगातार बिना किसी झिझक के, बिना किसी हिचक के बजती रहें, बजती रहें. Here comes the first prize winner, Dr. Bharti Karelia, for pharmacovigilance. Congratulations, madam. The next set of prizes would be for the residents. The next set of prizes at the valedictory ceremony of NAPTICON 2024 would be for the residents, and under experimental pharmacology. Under experimental pharmacology, the third prize would be taken up by Dr. Deepan K. Dr. Deepan K. The second prize winner would be Dr. Nidarsan Viswanathan. The third prize winner, Dr. Deepan K. Second prize winner, Dr. Nidarsan Viswanathan. And the first prize goes to Dr. Tejas M. Dr. Tejas M. Dr. Tejas M. Just after Dr. Nidarsan Viswanathan, we shall have Dr. Tejas M. Congratulations to all the three residents for receiving these prizes for experimental pharmacology. Next would be for the drug interventional studies. For drug interventional studies, the prize goes to Dr. M. Salguna Vivek. Dr. M. Salguna Vivek. Congratulations, sir. Next would be for medical education. For medical education, the prize would be taken up by Dr. Lily Joarder. Dr. Lily Joarder. Dr. Lily for medical education. आप तालियां जितनी ज़्यादा बजाएंगे, हमारे विजेता उतनी जल्दी मंच पर पहुंच जाएंगे. आपकी तालियों से इनका जोश दुगुना तिगुना होगा और ये दौड़ के, भाग के, डांस करते हुए शायद स्टेज पर आ सके. तो तालियां लगातार लगातार बजाते रहें. Let these thunderous applauses act as the background music for all of these winners to come on the stage dancing. And for observational studies, we shall have the third prize to be given up to Dr. Saksham Gulati. Dr. Saksham Gulati. Dr. Saksham Gulati की फैन फॉलोइंग बहुत बड़ी लग रही है. तालियों के साथ ढेर सारा शोर. क्या बात है. And the second prize, Dr. Anand Shukla and Dr. Shalini Thomas. Dr. Anand Shukla and Dr. Shalini Thomas. Dr. Anand Shukla and Dr. Shalini Thomas. जितनी जोरदार हूटिंग और तालियां आपने अपने दोस्त गुलाटी साहब के लिए बजाई, उतनी बाकियों के लिए भी बजाई. सभी आपके दोस्त यार हैं. भारत मेरा देश है, सभी भारतीय मेरे भाई बंधु हैं, दोस्त हैं, तो सबके लिए बजाइए, बिना किसी पक्षपात के, बिना किसी भेदभाव के. अरे बजाइए. And the first prize goes to Dr. Namita Dash. The first prize goes to Dr. Namita Dash. Congratulations. And now for systemic review or meta-analysis, the second prize goes to Dr. Revati Radhakrishnan, and the first prize for systemic review meta-analysis at the valedictory ceremony of NAPTICON 2024. The first prize goes to Dr. Nikunj R. Agrawal. Dr. Nikunj R. Agrawal. Dr. Nikunj R. Agrawal. Congratulations. हंसते मुस्कुराते थोड़ा डांस करते हुए आइए, क्योंकि हमने प्राइज जीता है, राइट. तो खुशी चेहरे पर साफ साफ झलकनी चाहिए, आपकी मोलर टू मोलर स्माइल दिखनी चाहिए. And for pharmacovigilance, the third prize goes to Dr. Gunjan Gayan. Dr. Gunjan Gayan. गुंजन जी के लिए तालियां गूंजेगी. Everybody, come on. तालियां रुकनी ही नहीं है, बार बार बोलने की आवश्यकता ही ना पड़े. ये आखिरी मौका है NAPTICON 2024 में तालियां बजाने का, अपनी हाजिरी, अपनी अटेंडेंस दर्ज करने का. डू नॉट लूज दिस अपॉर्चुनिटी. सबको पता चलना चाहिए, पूरे एम्स नागपुर को पता चल जाए कि हमारे डेलीगेट्स कितने जोशीले हैं. देयर सो मच ऑफ स्पिरिट एंड एनर्जी फॉर द फेलो डेलीगेट्स. द थर्ड प्राइज गोज टू डॉक्टर गुंजन गायन. द सेकंड प्राइज गोज टू डॉक्टर अरुण एस. And the first prize goes to Dr. Seema Gogoi. The first prize goes to Dr. Seema Gogoi. कॉन्फ्रेंस के बाद कोई मेरा नाम ताली वाली बाई मत रखिएगा, क्योंकि बहुत तालियां मैं आपसे बजवा रही हूँ, मगर आवश्यक है, ज़रूरी है. इट इज़ वेरी वेरी इंपॉर्टेंट एंड नेसेसरी, क्योंकि बहुत मेहनत की है हमारे सभी विजेताओं ने, तब जाकर वे यहाँ तक पहुँचे हैं. डॉक्टर सीमा गोगोई. After Dr. Arun S, we shall have Dr. Seema Gogoi. And just after this, we shall have the poster presentations. And to join us on stage, we shall have the very dynamic Dr. Mrunalini Karikar to give away the prizes. Now time for Dr. Seema Gogoi for pharmacovigilance, the first prize winner. And for poster presentations, for faculties, medical education, the prizes. For posters on medical education, the prize goes to Dr. Tadvi Nasir Ashraf. Dr. Tadvi Nasir Ashraf. Also, drug interventional studies, the prize goes to Dr. Nalini Kumari. तालियां बजती रहें, एक के बाद एक मैं नाम लेती जाऊंगी, आप तालियां बजाते जाइए. Observational studies, Dr. A. Uma Maheshwari. Medical education, Dr. Tarvi Nasir Ashraf. Drug interventional studies, Dr. Nalini Kumari. Observational studies, Dr. A. Uma Maheshwari. तालियां बजाते रहिए, बजाते रहिए, बजाते रहिए. Do not stop. And for residents, experimental pharmacology, the prize goes to Dr. J. Manoj John Paul. Drug interventional studies, Dr. Ria Ranjit Vedyar. For medical education, the third prize goes to Dr. M. Gautam. Second to Dr. Mansha GP. The first prize goes to Dr. Akshita Doshi. For observational studies and the residents who take up the prizes are, the third winner would be Dr. Urmi Jain. Second, Dr. Kritika Milind Dongre. Oh my God, कृतिका जी के जो दोस्त हैं, प्लीज़ वो लोग सभी के लिए तालियां बजाएंगे और शोर मचाएंगे. बड़ा अच्छा शोर आप मचाते हैं. डॉक्टर अक्षता दोषी. Then we have first prize for observational studies, Dr. Saro Meenakshi. Dr. Saro Meenakshi. And for systemic review meta-analysis, third prize, Dr. Lakshmi Hotkar. Second prize winner would be Dr. Prachi Tangde, and the first prize winner would be Okay, so we have Dr. Tadvi Nasir Ashraf here. She's taken up the prize. Okay, everybody please join us. Dr. Urmi Jain, if she's here. Dr. Ria Ranjit. Dr. Ria. Dr. Akshata, if she's here. Dr. Akshata. Dr. Manasha. Dr. M. Gautam. Dr. Saro Meenakshi. Dr. Kritika Dongre. Dr. Urmi Jain. Then we would have for systemic review, meta-analysis, Dr. Reflex Kumar Patel, Dr. Prachi Dangre, and Dr. Lakshmi Hotkar. And for pharmacovigilance, Dr. Ria and Dr. Dipanvita Sarkar. Requesting all the organizing committee members to please join us on stage. All the organizing committee members.
उषा डॉक्टर उषा किरण है क्या है सर नीचे बैठे हेलो मे आई रिक्वेस्ट टू डॉक्टर आवर वाइस प्रेसिडेंट डॉक्टर जितेंद्र सिंह वाइस प्रेसिडेंट डॉक्टर स्नेहा अंबानी वाइस प्रेसिडेंट उषा किरण काकीनाडा उषा किरण पी टू प्लीज come on the dais to felicitate the organizing committee members. Thank you very much.
Let us have a quick felicitation for all those who have worked day in and day out for the grand success of NAPTICON 2024. And first and foremost, I would like to call upon Dr. Avinash Turankar. Dr. Avinash Turankar, please welcome Dr. Avinash Turankar with a huge round of applause. He's not here, but तालियां उनके लिए जोरदार बजनी चाहिए ताकि वो जहाँ कहीं भी हैं कार्य कर रहे हैं उन तक हमारी तालियां पहुँचती रहे. Dr. Vandana Badar. Dr. Vandana Badar. Dr. Vandana Badar. Dr. Mrinalini Karikar. Dr. Mrinalini Karikar. Dr. Anant Khoth. Dr. Anant Khoth. Dr. Mrinalini Karikar, Dr. Anant Khoth, Dr. Chaitali Chindrole. Dr. Chaitali Chindrole. Dr. Mohini Mahatme. Dr. Mohini Mahatme. Dr. Kavita Jaiswal. Dr. Kavita Jaiswal. Dr. Vandana Badar. Dr. Vandana Badar. Dr. Snehalata Gajbhiye. Dr. Snehalata Gajbhiye. Dr. Sachin Hivre. Dr. Sachin Hivre. Dr. Khushbu Bisht. Dr. Khushbu Bisht. And last but not the least, very, very important, our leader who knows the way, shows the way, and goes the way, Dr. Ganesh Dakle. The leader who has put in a lot of efforts to make this conference what it is for all of us, the NAPTICON 2024 at the AIIMS Nagpur, at the Orient city of Nagpur, the tiger capital of India. Thank you so much, sir, for this wonderful, wonderful conference, which had a record-breaking registrations of more than 550 delegates, so many knowledge-sharing sessions, so many panel discussions, a beautiful NAPTI quiz, and so much more for all of us. Thank you for all the sessions, the great gala dinner that all of us had last night. Thank you for the music, thank you for the masti, thank you for all the blissful moments at the NAPTICON 2024. And now one for Dr. Ganesh Dakle. तालियां बजती रहे, शोर मचता रहे. लगातार लगातार जोरदार असरदार धमाकेदार तालियां बजाते रहिए, because organizing a conference of this class is not an easy task. Thank you so much to Dr. Ganesh Dakle for being the leader who shows us the right path. Thank you so much, sir.
My apologies for interrupting. Let me read this certificate of distinguished appreciation to Dr. Ganesh Dakle, Professor and Head Department of Pharmacology, AIIMS Nagpur, is hereby honored for his unwavering dedication, visionary leadership in shaping and successfully executing NAPTICON 2024. His parallel commitment and hard work has elevated the profile of pharmacology, clinical pharmacology and therapeutics all over the country, inspiring a higher standard of excellence across this field. Through this remarkable contribution, Dr. Dakle has been instrumental in establishing NAPTICON 2024 that has been held by All India Institute of Medical Sciences, Nagpur. It is a milestone event leaving a long-lasting impact on the medical and scientific community. Thank you.
Can we please have a quick group click of the entire organizing committee? Thank you so much and heartiest congratulations to all of you for the grand success of this conference. Thank you so much. May I please call upon Dr. Snehalata Gajbee for a vote of thanks.
Good afternoon, esteemed delegates. As we come to an end of this conference, it is with deep appreciation that I stand before you, I, Dr. Snehalata Gajbee, to extend our heartfelt thanks to everyone who made this event possible. First and foremost, I would like to express our sincere gratitude to the President of NPT, Dr. R.K. Dixit Sir, former President Dr. Padmaja Uday Kumar Madam, National Coordinator of NPT, Dr. C.M. Kamal Sir, and our very own General Secretary of NPT and Organizing Secretary, Dr. Ganesh Dakle Sir, for meticulous planning of the event. We would like to thank our Executive Director, Dr. Prashant P. Joshi Sir, for always supporting us in all our endeavors. We would like to thank our MMC observers, Dr. Vijay Modghare Sir and Dr. Meena Mishra Madam for supporting us. We would genuinely like to thank our esteemed resource persons, chairpersons, and judges. Your valuable insight, expert guidance, and engaging presentations and deliberations have truly enriched us. Your willingness to share your knowledge and experience has been instrumental in elevating our understanding. I would like to thank the organizing committee members who have made this event possible. A special thanks to our former medical superintendent, Dr. Manish Shrigivar Sir, for helping us with all the arrangement. I would like to extend my sincere gratitude to our working force, our senior residents, Dr. Bhagyashri, Dr. Cheta, and Dr. Kalyani, our postgraduate students of AIMS, GMC, and IGGMC Nagpur. A big thank you to the tireless efforts of our non-teaching staff members. I would like to take their names, Mr. Shubham, Mr. Shivam, and Mr. Neeraj, Mrs. Vidya, for their meticulous planning and tireless effort. I would like to thank the PRISM event management team and Richa for an excellent comparing. We would like to acknowledge the help extended by the support staff of auditorium security personals. Last but not the least, I would like to thank each and every one of you, all the delegates, for your active participation and engagement and enthusiasm. We hope that the knowledge and skill gained from this conference will empower each one of you. Thank you, and with this thanks, I conclude the conference.
आज हम सामने आ जाइए। अच्छा
मैं फोन पे बात करूँगा मेरा ये थोड़ा सा मेरा एक तमन्ना ये है वो तो मैं सब कर दूँगा मेरा एक तमन्ना ये कम से कम 100 लोग जो फार्मास्यूटिकल में काम कर रहे हैं उनकी एक मीटिंग सारी से भी बोला है और जी जी हाँ मैंने मैसेज भी किया